Okay. Good morning. Welcome, everybody. I am so happy to meet all of you here. And we are going to start the day. This is the last conference from after 6 years of a circle project. And so we will see our advance and results and innovation. Anyway, we can start now with a short video we made from our last dissemination event that has been carried out in Bologna, in the campus of our university in Rimini. And then we can start with our guest. And so let's start with the video, please. Okay, thank you so much. So we were enjoying a lot. So the idea is, since everything ended up in the sea, also our project finished in the sea as well. So on sailing boat, fortunately. So anyway, we can start now with the first session, innovative molecular strategy for sustainable food production. And we have two guests. One is Lene Lange, and the second one is Stephanie Compart. So we will start with our first guest. It's an honor for me. Lene, she's going to talk about the optimized circular use of bioresources, improving both human and animal gut microbiome. Lene is founder and owner of a PhD. She has a PhD, and she's founded and owner of an LLL Bioeconomy Research and Advisory, and she has a long experience from director of research position in both private sectors, more than 20 years, and academia as a full professor at the Danish University. And she is vice chair, scientific committee advisory of EU Commission, and now newly appointed as scientific committee of CBA-JRU. Member of the Danish Bioeconomy panel and advisory to the Danish government since 2014. So please, Leonard, take the floor.
Thank you so much. It's such an honor. I didn't know I was going to be the first speaker. I didn't know. That's fantastic. It's an honor and a privilege to be standing in front of you. The Circles project has been so famous in Europe for being very successful. And I've followed it from the Microbiome Support Action, which I headed by Angela Sesic, so I have been following, and I think we are on the same track in actually understanding, taking a holistic approach for the health and One Health. So, but let me go directly. Thank you so much for the introduction, by the way. But let us not use more time for this, but I believe I do have 20 minutes, correct? Yeah? Okay, that's great. So, circular feedstock for producing gut-healthy food. Remember that we are wasting so much. It's unbelievable. And I know you are learned in this area, so you know already, but let me repeat. We are having food waste globally, 34%. A little more than a third of all what we produce is wasted. Then we also have, and that is very often overlooked, maybe even in your circles, I'm not sure, and that is the side streams of food production and food processing. The residues from when you take the primary part of the crop can be used now with modern technology, opening the structures with enzymes and microbes. We don't more need have 4 stomachs to actually get something out of fiber-rich plant part of the crop. So also remember the side stream. The side streams are already collected in one spot. The logistic is paid for. It's already produced. It's already in the company, and pre-processed during the first separation from the primary food. And there, you can just upgrade that, and that is the less CO emission producing, the less logistics, the less efforts in more further processing, is if you start using what we already have in the side streams inside. And still no one almost talk about it. Even IPCC in the climate committee and UN and so on, they say in order to feed the world in the coming with 9-10 billion, we need to 50% increase our yield on each acre. That's the general. But no one mentioned in this context that if you just use better what we already produce and already have harvested, already have assembled, then we are almost there. I mean, it's amazing, and it's overlooked. So I will expand a bit more on this, but then I will first give you a little more. I think I have, but let me expand. Remember, it's not just agriculture and plants, it's also seaweeds from the marine, and it is cutout from fish, it is residues from forestry, it's all over. And then also, I think we should remember in our own yard, I mean, the biotech companies in the bioeconomy where we are producing biomanufacturing, where big microbial biomass bacteria or fungi are being produced. What are they being used for afterwards, after we have harvested that product? Biogas. And it's beautiful microbes. They are food compatible. It's proteins, it's vitamins, it is fibers, it's healthy components. We can do so much even with that. And I can speak on that. I had 20 years in Novo Nordisk and Novozymes. And I know that we, in the start, used it for animal feed. And the pigs loved it. And the farmers I was heading the green group of Novo Nordisk at that point in time. And the farmers came back to us, and we gave it for free. And said, we have never had a better pig feed than the yeast cream you provide as a leftover from your insulin production. But then, of course, and rightly so, EU said it is not acceptable to use a biomass from a pharmaceutical production. And they can have some good rights in saying that. But I'm just saying, maybe that kind of problem could be overcome. Maybe we could make sure it was okay. Maybe we can do something, because it's huge amount. Insulin for the most of the world, or half of the world. And then the other part, you also are talking about the microbiome. Are we ever talking about do we have good microbiomes in the residues, crop residues? Maybe some really good ones. Or do we have it in the processed side streams? We don't know. I think that we need more research on that. Okay. Unlocking the full potential, I think you already got my point. So I'll just say here, this is a little childish, kindergarten-like to make a color code, but I have been in this business of bioeconomy, in Novozymes, I mean, we started at the turn of the century to focus on this, in the better use of bioresources. And there, what I've been working on is designing the processes. The interesting part is this is not just a kindergarten color code. The yellow biomass, the recalcitrant straw and wood chips, etc., need pre-treatment, need much harsher because it's much more recalcitrant. The green biomass is more easily accessible and so on. So that need another process. The blue biomass, again, it's an animal, no plant cell wall, no plant recalcitrant fibers. The red biomass, blood and so on, totally different again. Gray agro-industrial side stream, I did introduce that. But let us say also the sludge. The sludge manure is not used well so far, and that's huge. I mean, the sludge, there's so many interesting, even from wastewater treatment plants. If we take care of safety, you can produce leather, non-organic, I mean vegan, whatever you call it, from this microbiome. You can make also from the wastewater treatment plants, the microbes there, the bacteria. You can have a phosphor enrich or recover the phosphorus, so you can use it as a soil improver. And then that's a brown one. The purple, I'm just adding it here. I'm not continuing to talk more about it. But please keep an eye on how much the fantastic methods now are producing food and feed from air. I mean, from CO2, from methane. There is a Unibio company in Denmark who have worked on making since the turn of the century, working on making from methane, grow methane-loving bacteria. And that is a fantastic protein-rich animal feed. And then the newest is that we can produce food from CO2. And that is a big Novo Nordisk Foundation, Melinda and Bill Gates Foundation, and Wellcome are going together. It's called Cork CO Research Center, CO2 Research Center, and they are. It is fantastic. And I could speak forever, and I know I'm using my time, and I have to go very fast over the list because you are such a small group, and I wanted really to convey the major messages here. And CO2, have you heard about how you can make food from CO2? One person have more, but let me just say, make sure you heard it from me. You can convert by a little bit electricity demanding, but you can go from CO2 to acetate, one step up. And both fungi and bacteria have a small, weak pathway in their genomes. For instance, that's the agaricus mushroom and several bacteria, where they can actually grow on acetate, make use of acetate, not grow, but make use of acetate and even producing protein. But it is not very good and not very high yielding, but with the new gene editing techniques, no GMO, no foreign DNA, the very nice just stopping, blocking a few of the metabolic pathways, so it only has the little acetate using metabolic pathway. And if you want to survive, you have to use that part. And that means that you can produce so much biomass and a fungal yeast, but also the mushroom, where you can make beautiful burgers from their mycelium with a good bite and umami taste. Even a meat lover can almost not distinguish the taste. So there is really something to keep an eye on. Yes. Target conversion of residual biomass can be done by enzymatic treatment. Prebiotic food and ingredients can be made even from the fiber-rich. Probiotic cultures can be grown, not just as most people do and companies do now, on sugar, refined products, but actually use the residue. Circle, 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 yeah? So you can convert already circular bio-based substrate to prebiotic, by enzyme treatment, making small, I will come back to that, but you could also make probiotic cultures. And you can ferment it all using fungi or bacteria. And you can even, for the fermentation, you will make anti-inflammatory compounds. And with all your focus on health, I think you know that prebiotic plus probiotic plus anti-inflammatory, that is the key to really have a stronger gut health with all combining all three. So here, the color code, and just to show here another way you can. By the way, you can have my presentation. Feel free to use all of this information in the drawings, and you may quote me or you may not. I want the message spread, you know. So and that is already published. So I think you got it here. So valorization of many types of biomass, unlocking the full potential. Same thing again and again. And then I have to use this one because I love this collage. Do not forget the fungi in the microbiome, both for the bioeconomy, I already said with the fungal enzymes, but also in the microbiome. Always, I mean, I have been now for so many years in the Microbiome Association, as we call it now. And I see so many papers coming out and review. If you're a reviewer, please stop if people conclude totally from only sequencing the bacteria of your microbiome, of the microbiomes of animals, plants, human, whatever. We have not the full picture of the function of the microbiome if we have not including the eukaryotes as well. And it's fungi, it's also protozoa, a lot of protozoa also in human gut. And they're also viruses and mobile elements and starship horizontal gene transfer. So many things goes on and we don't see it. And I'm not criticizing the ones who only sequence for the bacteria because it's so much more simple to handle and so on. But please don't, in your conclusions, draw conclusion like you have understood it all. By only sequencing the bacteria, we have concluded this and that. But remember, you have not seen the full picture. Of course, this picture was a picture of Denmark, and we are, I believe, the country in the world who get most out of fungi per capita. It is amazing. It is more than 20% of the wood, the good export. The export of goods from Denmark is from fungal-based companies. So many more can do that in many more countries in Europe. And I mean, I tell you, fungi is future. Investment, now I come with a little bit more EU investment, and you have to stop me if I am going too far. We had this, and you know all about it. We have BBI, joint undertaking. I was vice chair of the scientific committee there, and we had the cascading use and upgrading all that. And we ended up, and there are two references from the scientific committee, Lange, O'Connor, et al. We actually told the whole story. And at that point in time, in 2021, we were in lead in the world. Definitely biorefinery technologies, cascading use, full unlocking the full. But then drastically changes in international development, just 2022 to 2024. It's amazing. China, new 5-year plan, suddenly not just biomass to biofuel, bioenergy. That was the only thing allowed up to 22. Now it's everything. All U.S. Biden, the Inflation Reducing Act, fully blown bioeconomy using everything. Indian chairmanship, a strong document, 20 pages document in the G20 organization when they had the chair of G20. And India Modi made a biomanufacturing strategy in 2023 for all of India. Brazil is a new G20 chair, and they are fully blown also Brazil is carrying the torch in better use of the bioresources, many biosolutions. And Thailand and South Korea are really making the biorefining. And they are running so strongly. And this is beautiful. It's not enemies. We need it to happen all over the world. But don't get me wrong, I still think that we should do our best in Europe. We should speed up, because we risk to lose both talent and investment, especially for the fantastic climate of welcoming people and giving them good investment climate and good support for establishing and commercializing and upscaling. All the youngsters where we have invested in giving all the nice EU grants and so on, they are very appealing for them to move. We can hear it from Denmark. If we don't move fast now, it will happen. So, and I think we should move fast. I think I don't need to say that. So here EU in this spring, Ursula von der Leyen read the message and the Commission saw it. So they started a new and made a new dialogue strategy, EU, biotech and biomanufacturing. Microbiome hardly mentioned once. I'm happy it was mentioned. But we have to keep them on their tiptoes. I mean, really, they have to understand that there are new technologies. It's not just the old biomanufacturing. There are so many new things. The good news is that we will have news importance of microbiome and so on. And I will critical issues. And I advise, or I hope with so many influential people in this room, I hope that you will try to also be ambassadors for that the new types of technologies, the new knowledge we have to much better make the health out of also what we now wasted, to make sure that the One Health concept is understood, to make sure that we are looking across in a holistic way, but also really making sure, and this is my highest concern, that there are, all what I see, what comes from the rumors of the new commission and the rumors of which directions the new EU strategy will go in the bioeconomy and so on, I think they will welcoming more health to be included instead of being a very strong silo in EU. I mean, health is one thing, and the rest of us is something else when it comes to funding. We are taught in the scientific committee of CBE and PBI that we are not to make anything which is health, because that's another program, yeah? So this will be softened, that's good. But what I'm really concerned about is that healthy and health-promoting food and health-promoting feed is overlooked. Because then comes a strong farmer, one product, one compound, and one pill, and so on. But it is absolutely a necessity for a good, public health and good economy and so on, and also for it to be aware and robust for new pandemics, that we have more health based on what the food we are eating. And similarly for our animals. It is so important. So health-promoting food should be at the top of our mind. And I hope you will be ambassadors for this as well. Oh, this is just my wonderful Hall of Fames, the fungi producing biomanufacturing, insulin and enzymes and other nice things. Aren't they beautiful? I love fungi. Maybe you already knew. So, into gut healthy, I think I already preached this one, but the important part is that the fiber-rich pulp can be used. Because if most bioeconomies say, the fiber-rich part, we take out the protein, the fiber-rich part, we have to make for materials, chemicals, energy, and so on. No, please. The small oligos are pectin. The small oligos are hemicellulose. are fantastic as nutrient fibers, the gut-healthy fibers. Triple beneficial effect, probiotics, I also mentioned this as a very interesting one. Improved use, let us not forget the big perspective, all what I have been saying. If we carried on and did this, better use of the biological resources, we will favor a better use for the climate with less emission because we are producing more food, but not without making more all the time, starting from new production, etc. Food security is higher, public health is better. Biodiversity, if we produce more from what we now waste, we can save land and space for biodiversity. And it's not small acre. 75% of all arable land in the world is used for producing animal feed. 78% in Denmark, because we have 28 million pigs. That's a lot. We have 6 million people, so that's a lot. All this improved enzymes and enzyme blends are key. So my cases here are just to say enzymes are key in this, also for the fermentation. So, therefore, need for bioinformatics and analysis, we need new tools for prediction of function directly from sequence. Sequence is ample, it's all over. We have that by the truckload, but it's only data. And then if you are not an enzyme person, I'll tell you that the normal annotation is just to a protein family, and then many people believe then you have because it sounds like an enzyme function, and it is not. With each enzyme family, enzyme protein family, there can be 50 different functions. Or there can be 3 different. And it is crucial to be able to pinpoint the right function for designing the right type of processing. If you want, for instance, to convert the fibers into health-promoting oligos, yeah? So this function, because that's biology, that's applied direct to better know which way to make it good from a business as well, yeah? So don't fool yourself when you just have a protein family. Let me know if I'm too long. Yes, I am? Yes.
Maybe we can leave some space if you have any questions from the audience.
Yes, I will stop now, and then I will say, yes. Conserved unique peptide patterns, look it up, because that's with the function prediction, and automate it, and you can find it is there, and so on. And then I'll just say, share this, One Health. Ethics, remember when we talk about global food and global planet, remember, this is so important to share. Knowledge sharing in this area is key. Okay. Acknowledgement and thank you. Thank you so much.
Thank you very much, Lena, for the inspiring presentation. And I think that will be the room for discuss more later on the several points you were raising. I guess they are very important also for the future. And the second speaker is Stephanie Compat. Okay. Stephanie is a senior scientist at the AET, Australian Institute of Technology, and received his PhD degrees from the University of Rheims-Champagne-Ardenne, his habilitation from the University of Bordeaux, and was associate professor of microbiology at the Polytechnic Institute of Toulouse in France before to work at the AIT. His research focused on ecology, function, and exploitation of plant-associated microorganisms. He further used advanced microscopy techniques to study plant microbiome interaction and describe beneficial properties of models of action of microorganisms to develop biopesticides that can be used to tackle plant pests and diseases. The title of the talk is Addressing Fungi and Bacterial Interaction to Improve Plant Growth and Health. Please, Stephanie.
Thank you for the introduction and nice to meet you. Good morning. I'm Stéphane. Where is my presentation, please? No, the first slide? Yeah, okay. I'm Stéphane from AIT Austrian Institute of Technology, and today I would like to speak about fungal-bacterial association and if we can harness this interaction to improve plant growth and health. But first of all, if we check the plant microbiome, of course, we have different kind of microorganisms, protists, archaea, viruses, bacteria, fungi in different plant parts, the roots, the leaves, the flowers, the fruits, the seeds. But some microbes are present in the same niches of colonization. And especially if you take a look at the fungal and bacterial communities, there is some taxa present in the same niches, the same tissue of the plants, and some of them can interact. This is, for example, well known with mycorrhiza and some bacteria, but we have also several other examples. And can we harness this interaction, taking naturally place on and inside the plant microbiome? There is some bacteria, I just told you, that can help fungi. For example, bacteria helper of mycorrhiza. But there is also some bacteria inside pathogenic fungi. This is, for example, known with the good example of Rhizopus, pathogenic fungus, with some Burkholderia inside, responsible of toxin production. There is also, of course, some bacteria decreasing the growth of pathogenic fungi. But you see that there is different kind of function according to the bacterial and the fungal taxa. And there is some bacteria on the surfaces of hyphae. Some, they make biofilm. But some, I found this very interesting, some bacteria inside hyphae of fungi. What kind of taxa we can have? We can see that when we do metabarcoding, for example. But today I would like to show you some example. A beneficial fungus that can be associated with some bacteria, and a pathogenic fungus. The first one is Serendipita indica. It's a mycorrhiza-like fungus, stimulating plant growth, improving yield, improving tolerance to abiotic stress and biotic stresses. What is very interesting inside this fungus, it is that it hosts a bacterium inside the hyphae. And I thought, if this fungus can interact with a bacterium, maybe it can also interact with some other bacteria that can improve the growth of this fungus. And during a microbiome project, we were focusing on Serendipita indica, and the idea was to find some bacteria that can help the fungus. How we have done this? We took the fungus, put onto soil, where potato and tomato were growing, harvest the fungus, isolate the bacteria that can be associated with the fungus, put back the bacteria on the fungus, and check if the fungus was growing better or not. Here you have Serendipita indica alone. Some bacteria, they were killing the fungus, and some bacteria were increasing the growth on petri dishes of the fungus. You have the example here, Serendipita indica alone, the addition of one bacterium, another one, and also in liquid culture. Then we thought, if some bacteria can increase the growth in vitro of this beneficial fungus, we hope that this bacteria cannot increase the growth of pathogenic fungi. Fortunately, not. Again, Rhizoctonia solani, again, Fusarium oxysporum, some bacteria were deleterious for the fungi, and some bacteria had no effect at all. But now, what kind of taxa we got that were improving the growth of the beneficial fungus Serendipita indica? They are here. Some Momberg Xanthomonaceae, Mycobacteriaceae, Microbacteriaceae, etc. A lot of different kind of taxa, very interesting. But this, I told you, it was on in vitro assay in the lab. I told you that the beneficial fungus improved plant growth and yield. So now we move to the plant assay. Under high nutrition soil condition, if you had one bacterium or another one, or the beneficial fungus plus some bacteria, no effect at all. On the low nutrient soil conditions, there was some bacteria increasing the leaf area of tomato, Serendipita indica also, and some bacteria plus Serendipita indica were boosting the effect of the beneficial fungus. Then we were checking if there was an effect against plant pathogen, against Fusarium oxysporum, but I will not present this. I prefer to show you this example, again, Rhizoctonia solani. You have the mark here. A plant inoculated with Rhizoctonia solani, the pathogen, 111 days post-inoculation, all the plants were dead. When we put Rhizoctonia solani plus one bacterium T1210, a Trinicea species, previously known as Parabiocolderia, no effect at all. The pathogen plus the beneficial fungus, 40% of protection. And the pathogen plus the beneficial fungus plus one bacterium, maximum of protection. Very interesting. So you see, we select at the beginning some bacteria that can interact, improve the growth of the beneficial fungus, not the pathogen, improve the growth of the plants, but also improve resistance to pathogen like Rhizoctonia solani or Fusarium oxysporum. We know that these bacteria improve also the sporulation process on the root surface. Here you have one bacterium, Trinicea species, T1210, as red, the fungus as green. They were present at the same niches of colonization. Increase of sporulation, increase of aggregation of the fungus in presence of some bacteria compared to the fungus alone. Improved colonization process of the fungus en route, 10 weeks post-inoculation, 8 weeks post-inoculation. Then we wanted to know more about the putative mechanism involved in the interaction. We are planning to do transcriptomic analysis, not enough time for the PhD student, but we have done genomic analysis of the isolate, the bacteria. Of course, we found a lot of gene clusters related to prong growth motion, tolerance to biotic stresses. But I was very surprised to see a lot of genes cluster related to vitamin metabolism inside the bacterial strain. Some gene related to B1, B2, B3, B12 production. At that time, we didn't know if the vitamins were really secreted. Now we know this, and we have done also some experiment to put the bacterium on plants to provide B12, more B12 for vegan people. And the hypothesis was that these bacteria can improve the growth of the fungus by providing some vitamin. B1 is a thiamine. It's necessary for fungal growth. It's just the hypothesis. It was one example. If we can harness the interaction between bacteria and fungi to improve plant growth and yield and health. But now let's take a look at another kind of interaction. On pathogen. One pathogen I was focusing recently on, it's Formitiporia mediterraneae. It's responsible of white rot in grapevine, destroying the plants. When the plants, they have 10, 20 years old. And my friend from France, Patrice, told me, I found some bacteria isolated from grapevine. They increase the pathogenicity of the fungus. We know that the fungus is responsible of the white rot, but when we put the bacterium alone, there is no effect on sawdust of grapevine. When we put Formitiporia mediterraneae, there is a degradation of wood. And when we put the bacterium plus the fungus, there is more wood degradation. I made some genome analysis to try to find the putative mechanism explaining that. But I thought at the same time, if some bacteria can increase the pathogenicity of this pathogenic fungus, let's take a look inside the fungus, if we have something. And I found some bacteria. It's not mitochondria, it's some bacteria. Then the challenge was, do we have this in one strain or in several strains? It took me 4 to 6 months to ask people to give me strain isolated from grapevine. So I mean Formitiporia mediterraneae strain, the pathogenic fungus, from olive, from citrus. We made also our own isolation. We have circa now 200 strains from Europe, from South Africa, Formitiporia capensis. for Multiporia australensis in Australia. We have fresh sample isolated from ourselves, some fungus that were stored in freezers since 10 or 20 years. And we made beta-barcoding approach on the different strain, not on the 200 yet. And what we see here, we have different abundance of bacterial taxa, like some Rhodococcus, some Bacillus, Fungomonas, but also Cutibacterium. Very interesting. What they are doing inside the pathogenic fungus, we don't know yet, but they are present. We compare the diversity according to the cultivar of grapevine from where the fungus has been isolated. You have the result here. Also according to the host, grapevine or citrus or foliage, or according to the fungal species. But what we have seen, there is differences of diversity in some cases, but also conservation of the bacterium with some ASV present in all the species of Formitiporia, Mediterranean, Australiensis, or Capensis. Then the challenge was, can we isolate this bacteria? Thousands of petri dishes, nothing, or sometimes contamination, even when everything was sterile. And my postdoc thought it was a very good idea, but you know, if some bacteria are present inside the fungus, let's try to do isolation under anaerobic condition. And we got some bacteria, some Cutibacterium acnes, that were present in the metabarcoding. It was previously known as Propionibacterium acnes, some Brevundimonas, Georgian Apenic micrococcus. We are doing now some genomic and metagenomic analysis. We have cures of fungus, some strain. The physiology of the fungus look a bit different. We are doing new metabarcoding approach, and we don't know yet what are doing these bacteria inside the fungus, but they are present independently from where the fungus has been isolated. Is it symbiont? Is it parasite? We don't know yet. It is present inside the hyphae. We don't know inside the spore because in the lab we don't have spore production of this fungus. But now, what I told you about Cutibacterium acnes, why I found this interesting, because it was 10 years ago. At that time, it was still pyrosequencing. And there was some friend from Italy, from FMARC Foundation. They told me, Stefan, there is something very strange in the wood of grapevine. We found some sequence related to Propionibacterium. Well, what it's doing inside these plants, we don't know. There was no possibility of cultivation, but at the same time, they made analysis of rake A, GA, etc., and they found that it was a new species of Cutibacterium acnes. They call this, according to Frank Zappai. We submit this paper in Nature. It was rejected, but then they made a big news about our paper. So thanks for them. And yeah, and now I found this inside the fungus responsible of white rot inside grapevine plants. But I still don't know what the bacterium is doing inside. Yeah, take home message. I told you at the beginning, the title was Harnessing Bacterial Fungal Interaction to Improve Plant Growth and Health. But now I would like to ask you, can we harness this interaction really? With some bacteria, boosting the effect of beneficial fungi. But also, you know that there is some bacteria associated with fungal strain, pathogen, what they are doing. When we target the fungal pathogen, shall we also target the bacteria to reduce the pathogenicity of the fungus? Some fungus, of course, they can live alone without the need of some bacteria, but it's a eukaryote. There is also the bacterium of this fungi. What are they doing inside the fungi? How we can harness this interaction? If we can harness this, can we discover also some new metabolite of some bacteria inside the fungus? When we make genomic analysis of fungal strain, we clean all this data. Now I'm coming back on that. I would like to thank you very much for your attention, and if you have any question, I can answer. Thank you. if there is time for question.
So if there is no question, yes, Davide. Yes, of
Yeah,
course.
Davide, please.
I'm gonna shout.
Okay.
I don't know whether you mentioned, or probably I missed, is there any relatedness between the bacterial complement of a given fungus and the plants they colonize? Let's say if you isolate from grape wine or olive, can you discriminate the bacteria that are associated with those fungi? So can they contribute to the colonization of the plants, or is
Well, we don't know yet. We don't know yet. Yeah. We were just starting first to make metabarcoding of bacteria inside the fungus before to move back then to the plants. What we see when we compare the diversity of the bacterium of the fungal strain when it has been isolated from different plants, grapevine, olive, or citrus, there is the same diversity. There is also conserved bacterium. We think that there is a core bacterium, but also a satellite bacterium, from depending on the location from where the fungus has been isolated. What we see is that there is some ASV, they are always present. So they might have a role on the fungus.
Are there any other? Please, just because it's a streaming event, otherwise we cannot hear from the video, so we need to.
So I have a curiosity. In this case of symbiosis between bacteria and fungi, do you know anything about the abundances of bacteria and fungi in terms of cell numbers? Because they always grow at different abundances.
It was very hard at the beginning, first to make the metabarcoding, because it's the bacteria inside the fungus. It should be present at 0.0001% of the total DNA of this. What I see at the microscope on this Formitiporia mediterraneae, it's full of bacteria inside some hyphae, not in all the hyphae, but it's really full. In some strain, I have been focusing also recently on Trichoderma. We found also some bacteria inside Trichoderma strain. It's really depend of the strain. When the one from Serendipita indica, the natural symbiont inside Serendipita indica, it's a very low cell number, but it's still present. It's never sterile. It's an eukaryote,
Yes.
Fantastic talk, and I think we should start not being surprised about interaction between fungi and bacteria,
Yeah, thanks.
because that is nature. Fungi are interacting with all types of organisms. And I'm so happy you will continue as Serendipita, because the new work in India, they are believing that it will also be able to make the plant more, together with the bacteria, I think, more drought tolerant.
Yeah. Thank you.
It's amazing.
Thank you.
So if you don't have any question, we can move to session 2, giving the floor to Giorgio Palladino. Thank you, Stefan.
Okay, so we are starting with the session 2, streamlining development, implementing common procedures, tools, and initiatives for enhanced research. It's my pleasure to introduce you the coordinator of the Circle project and my colleague, Marco Candela. He is a full professor in chemistry and biotechnology of fermentation at the Department of Pharmacy and Biotechnology at the University of Bologna. And of course, he is the coordinator of the Circle project. And he is also a member of the bioeconomy thematic unit at the University of Bologna, and vice coordinator of the international PhD program, Innovative Technologies and Sustainable Use of Mediterranean Sea Fisheries and Biological Resources. The title of the presentation is going to be Food System and Environmental Microbiomes, a bidirectional connection to be protected and managed for the sustainable production of healthy food.
Okay. Thanks so much, Giorgia, for the presentation, and welcome again to everybody. So it's a matter of the fact now that microbiome are a kind of life-supporting system for the global biosphere, since providing a range of ecological services which are important for the planet health. And which are these ecological services? The most important are oxygen production, the role in carbon and nutrient cycling, the support of the global food web, but also when microbiomes live in association with animals and plants, they are integral to the physiology of animal and plants and are so important for the support of their health. So what about microbiome in food system? In food system, microbiomes that take the same action. And so they support life. And thus, they are so important for the food production processes, for the quality of food production, for the safety of the production process, for the sustainability of the production process. So they are inherent in food production. So, but the point is that in the Circle project, we made a lot of research to explore the kind of bidirectional connection that exists between natural microbiome and food system microbiome. And we believe that this is a key factor for the sustainable production of high quality, safe, and diverse food on our planet. So let's see what happens, some Circle example of the importance of natural microbiomes for food production in food system and how they are connected with food quality and productivity. These are two Circle research that has been performed in this here. So this is the first research in which we explore the importance of microorganisms living in the soil-plant interface, even including fungi, as a possible neglected determinant of the terroir feature, which explain the difference, distinct regional and local variation of the wine characteristic. In particular, we focused on the wine terroir in the production area of the Vino Nobile di Montepulciano, which is now protected as designation of, with the designation of origin, so it's protected as BDO. And this area has recently divided in 12 additional geographic units, providing a specific product. So they are called pievi, providing specific product with specific organoleptic features. So what we have done in the frame of the Circle project was to explore the regional specificity of the microbiome, the soil-plant interface in the production area of the Vino Nobile di Montepulciano, and also its local variation in the 12 different additional geographic units. So we have been successful in finding microbial genera and fungi that were specific for the area of production of Vino Nobile di Montepulciano with respect to other vineyards across the world. And also we have been successful in identifying some microorganisms that were specific for the different pievi or additional geographic units. And also this specificity, these characteristics were corresponding to specific functionalities in terms of plant growth chromatic function. So all this data has been now submitted for a publication under revision in Communication Biology. Anyway, without the take-home message from this first research is that the terroir microbiome showed declination at the regional and local level, and this declination at the regional local level can provide plant with adaptive features that respond to the soil specificity. And these adaptive features are also connected, and we have proven that with some very recent metabolic data that has been used to upgrade the paper after the first round of revision. These plant growth promoting features provided by the local diversity of microorganisms in soil are also connected with specific organoleptic features of the product. So the idea, what can come as a possible solution, a Circle solution from this story, is that of course we can include microbiome in the biotic component of the wine terroir. and we can implement management strategies for the preservation, for the protection of this microbial diversity in soil, which is so important for the wine quality. And we need also to protect this microbial diversity from climate changes and global changes. So we will see some action in this direction in the next presentation by my colleagues. So this is a second example in a publication from the circle process in which we were working on clams in the northwestern Adriatic Sea, and we were proving that the microbiome in sediment is so important for the clam productivity, since providing clam with health-promoting microorganisms. So clams growing from high productive sites were full of beneficial microorganisms that were provided by the microorganisms living in the sediments. And so also in this case, we can say that the environmental microbiome, the microorganisms living in the marine ecosystem, and in particular in the seabed, are so important for nurturing the animal that we are growing for as a foods, providing health-promoting and beneficial microorganisms. So now the other side of the coin. So we have seen now the importance of natural microbiomes for food production, and then we will see how producing food in intensive food system we can compromise, we can impact the natural microorganism. And here there are several research that has been carried out in the frame of the Circle project. At first we were focusing on terrestrial food system, looking at the direct impact at the local scale. And mostly we were focusing on the dispersion of antibiotic resistance microorganisms and genes from the terrestrial farms. And here you have a study we performed in which we were focusing on the dispersion of antibiotic resistance bacteria from pig farms. And then there is a second study in which we were exploring, second twin study in which we were exploring the antibiotic dispersion of microorganisms and antibiotic resistance microorganisms for poultry farms. And in the second study, we were also including human beings, so the workers, as a route of dispersion. So we will see more about these two studies in a dedicated session in the afternoon. So this is why I was running so fast. And then we were exploring the impact of the terrestrial food system at a more global scale. And the connection between the local and the global scale is the sea, that is the means for the globalization of the anthropogenic impacts of the food system around the globe. So in the first circle study, in this concept, we were searching for pathogens from farms in the marine water in the northwestern Adriatic Sea, and we were successful in finding Campylobacter ureolyticus, that is an emerging gastrointestinal pathogen of zoonotic origin, which is capable of destruction of filamentous microvilli, so it's quite dangerous. We were successful in finding these microorganisms in bathing water in the northwestern Adriatic Sea in the coast. And then we made a second study with this concept. So in this case, we were focusing on the Tyrrhenian Sea on the other side of Italy, on the other coast, western coast, and we were using Ballen waves as a proxy. As a proxy for the contamination of marine water with pathogenic microorganisms, so coming from the food system. So we were searching the presence of enteric protozoa parasite as Blastocysti, Giardia duodenalis, and Cryptosporidium as emerging enteric pathogens from farms in Ballen waves, free living in the Tyrrhenian Sea. And we were also exploring the feedback impact on their gut microbiome health. And so we were working on 36 Ballen whales belonging to 4 species free living in the Tyrrhenian Sea. And of course, we won't expect that, but 66% of the success whale were found to be infected with one parasite. So with no strong impact on overall gut microbiome of the balance weight, but with the increase of specific microorganism in association with the protozoa parasite can represent a warning on the gut health of the balance weight. So this is with this paper we were providing a kind of warning pictures of the impact of food system also on free-living marine mammalians using a flag species to assess for the contamination with enteropathogenic microorganisms from farm on the sea. And then we were exploring also the impact of marine food system at the local and global scale. For what concern of the local scale, this is the first research has been carried out in which we were proving that there is a direct passage of microorganisms from aquaculture to surrounding marine holobionts such as Patella cerulea, and then when we were focusing at the more global scale, we were demonstrating also other kind of impact. One very important is plastic pollution. So plastic pollution in the seawater is an emerging contaminant from aquaculture. So we decided to use carretta sea turtles as a flag species for monitoring the plastic pollution in the marine water. And so we were enrolling more than 100 carretta sea turtles, and we see that plastic debris were pervasive in the feces of the sea turtles, and also they were resulting in detectable change in the gut microbiome that was enriched in plastisphere microorganisms. So microorganisms that are enriched on the surface of plastic residues when they stay in the environment and include also pathogenic microorganisms. So finally, we were searching for what is going on when we produce food in a contaminated environment from the microbiome point of view. So we were concentrated on mussels in the northwestern Adriatic Sea. So to make the long story short, also this is another circle publication. In the mussel that were growing in the northwestern Adriatic Sea, we detected a complex dynamic pool of emerging pollutants, including antibiotics, including pesticides, including herbicides, all coming from food system. And what do the mussel do? In this context, they enrich resistance genes. So the microbiome enrich resistance genes against antibiotics, herbicide, and pesticide. And what do the mussel do? They filter and they release in the environment, in the marine environment, they release resistant microorganisms. So compromising generally the efficacy of our xenobiotic substances, and so further increasing the needs to increase the dosage of the xenobiotics that are detrimental from the environment. So what we can say, in summary, we can say that the current food system, intensive food system, impact the natural microbiome in terms of dispersion of antibiotic resistance bacteria and genes, in terms of dispersion of enteric parasite, and in terms of dispersion of microorganisms which are selected from the farmed animal or selected from food system contaminants. So all this data is being presented this spring at the Food System Microbiome Congress in Turin. So what we can do? I think that as a circle project we can preserve natural microbiomes from any anthropogenic impacts, and in particular in the context of food production, since they are so important, as we have seen in the first presentation, for the provision of high quality, diverse and healthy food. So how we can protect natural microbiomes? With three actions, very simple. First, we need to assess the microbiomes. So we need to assess the state and the conditions of the microbiome. And then, after understanding the microbiome, we need to implement microbiome sustainable management strategy to protect this microbial ecosystem. And if the microbiome are compromised, we need to take restoration action, microbiome restoration action. That is something that is very important, and you will see some examples. So in the frame of the Circle project, this 3 action has been implemented at the level of food system, and you will see the rest of the presentation today. And of course, for implementing this action, the ultimate result of the Circle project is the Circle box. That is a box of customizable microbiome technology that has been conceived for implementing the 3 action, so monitoring, protecting, restoring at the level of food system to achieve a certain endpoint in terms of improvement of quality, productivity, safety, and sustainability. We will talk more in the next presentation about the Circle box, and you will see some concrete example. I don't want to spoil or anything about the results connected with the box usage in a real-world food system, since this is demanded to other colleagues. But anyway, just 2 key messages. So with the box, we generally aim at the exploitation of natural microbiomes as life support system to replace chemical inputs with microbiome-based ones for a more sustainable food system. So do not use chemicals, but use microbes, natural microbes instead of chemicals. So this is the first point. The second point is that within the box, we have implemented a universal method for the assessment of the microbiome disbiosis that has been now applied for a patent. That is the only one patent we provided in the frame of the Circle project, and is part of the box, is central in the context of the box. So we will discuss, there is a dedicated presentation to this universal microbiome for assessing microbiome after me. So, but in the frame of Circles, there is a lot of dissemination action. So we were taking advantage of the dissemination action for doing something for protecting the natural microbiome at the global scale, not only at the ecosystem, at the food system scale. And these actions were conceived as citizen science activities, since we want to communicate and do something about the importance of microbiomes in the One Health scenario in a collaboratory environment. So one of our works in this direction is a citizen science campaign that has been carried out this summer, in which we were engaging citizens in monitoring the marine water microbiome in the coastal sites, just engaging citizens in promotion activity, and then provided them with auto-sampling kits that has been developed by Well Micro in the frame of the Circle project. This way they were able to sample by themselves the water microbiome, and then we perform a lot of training activities for teaching how to use the sampling kits. Then, so they were sampling by ourselves and sending back to us the sample for the analysis of the water microbiome. So we made a lot of effort in the promotion of this campaign, since we need the help of the people, and also this is the area under study, that is the Northwestern Adriatic Sea, and of course it has been possible only with the support of the local association and institution, no way out. Anyway, this is the pipeline, the operative pipeline. So there is the kit production by Well Micro, then the teaching by the University of Bologna, the kit distribution by local authorities, and then the kit collection after sampling by the people, by local authorities, then sent back to Well Micro from sequencing, then back to Unibo for data analysis, and then back again to the people, since this is for the people. So they need to know what we are doing and what is going on in the water microbiome. So And this is in this concept we made also a pilot in the frame of the Circle project. This is another publication. We made a pilot restoration action of since we were aware of problems in the local waters. We made a pilot restoration action by using an artificial reef to restore microorganisms at the coastal site. And so we get some good preliminary results, and this is a publication that has been carried out by Circle, proving that it's possible somehow to restore and to protect microbiome, even at the global level. So finally, human beings are so important. So even the microbiome of human beings are under a threat, and which is the threat is modernization. So according to the maladaptive hypothesis, a combination of several factors that are connected with the modernization, that is antibiotics, the use of processed foods, sanitized environment, xenobiotic exposure, and so on, is pushing for imbalances in our gut microbiota. And these imbalances has an impact on our health. And so this is why there is increase of several inflammatory disease in the Western countries, since they are due to imbalances in the gut microbiota that are connected with the modernization factors, since our gut microbiome is not adapted to this kind of challenges. These are very recent in our history. So it's just hundreds of years that we have these challenges. So the idea is to rewild now the human microbiome, so to let the human microbiome to restore all the health-promoting microorganisms and features that has been lost with the modernization factors. And there are several strategies for the rewilding of the human microbiome, but one, and for me, and also in the frame of the Circle process, of the most interesting is to try to restore the human microbiome by reintroducing health-promoting interaction between human and the natural microbiomes. This for the rewilding of our gut microbiome. And so we made a pilot. We made a pilot since we were believing that the contact with animals in a rural environment may be beneficial for the human microbiome. So we made a pilot with children from urban sites that were going in an educational farm, staying in contact with horses for 10 hours a day. So this is the study design of what we call a biodiversity intervention. In which we were enrolling 10 children with moderate to low adherence to the Mediterranean diets. Then the children's stools were collected before the interaction with the horses, and after 50 days biodiversity interaction with horses in the educational farms. And then, of course, for each sample we were collecting, we were assessing microbiome by 16S amplicon sequencing, shotgun metagenomics, and targeted metabolomics as well. And also we were assessing the microbiome of the 9 horses, since there are sink of microorganisms, so we are assessing the gut, the skin, the fecal microbiomes of the horses. And so I don't want to spend more on this story, but the paper has been now submitted and is under revision to one else, and it's another Circle paper. Anyway, we got a lot of positive feedback from the study, since after the interaction with horses, we have seen the increase of many health-promoting features in the children urban gut microbiome, such as the increase of several next-generation probiotics, the increase of microorganisms able for the bioconversion of plant polyphenol to anti-inflammatory metabolites, and also microorganisms such as the alister able to protect from metabolic information. And we didn't find any metagenomic evidence of transmission of virulence and toxin-producing strains. So only beneficial interaction. Of course, they were healthy children with a healthy immune system. So this data have been presented as an invited plenary lecture at 10 International Human Microbiome Consortium Congress in Rome this summer, and also this was another circle project. Of course, 10 children is not enough, so now we are doing a lot of effort, even in the frame of circle project, to enroll more children. Is not easy. It's not easy because these are all healthy children, and I want to play with horses, not to donate stool. Okay, so. This is why we have only 10. But anyway, we are doing a lot of efforts, even this summer, even now, to enroll more children. And we have many now to be analyzed. So what I want to say in conclusion, that in the frame of the Circle project, we provided the knowledge and solution for restoring and keeping the homeostatic balance between the natural, the food system, and also the human microbiome. And this has been possible only because we have the chance to analyze more than 10,000 microbiomes of different kinds. And so to see the system, its whole complexity, including nature, food system, and human interacting together. So, and this is our group, the Microbiome Science and Biotechnology Unit. There is here Simone and Georgia. I must thank my group. And have you seen many publications? And the key person for this publication are the people of this group. It's not me, and so they are all the leading author of all the publications that you have seen. Thank you very much.
So we're going to go to the next speaker. You can catch up with Marco during the break, I guess. So the next speaker is Simone Rampelli. He's also a researcher at the Department of Pharmacy and Biotechnology at the University of Bologna. He published a relevant contribution on human, animal, and plant-associated microbiomes and bioinformatic approaches for microbiome data. He has been awarded with the Seal of Excellence for the Marie Curie Action in 2020, and he was appointed as a lecturer in the fermentation biotechnologies at the University of Bologna, where he could pass down the students his experience and passion for microbiome science. His presentation, if we can have it on the screen, is going to be, okay, perfect, universal reporting of microbiome disbiosis. Please welcome Simone.
So, yeah. Coming back? Yeah. So thank you, everybody, and thank you also for the nice presentation. So I start by saying that one of the main results of the Circle project is the awareness that food production needs a eubiotic microbiome as part of the production system. And this comes together with the need of controlling food system microbiome for preserving a eubiotic configuration or restoring the dysbiotic shift as the new strategy in food production from farm to fork. And in the framework of the Circle project, we take advantage of more than 10,000 microbiomes from 7 different European food systems that comprise poultry, swine, grape wine, spinach, tomatoes, cyprine, and salmon, for setting up a new universal reporting. And when I say universal reporting, I mean a unique approach for defining disbiosis in such production system, but also in other microbiome that comes from different hosts. And this method is based on the Anna-Karenina principle that is here mentioned as all microbiomes are similar. It's dysbiotic microbiome, dysbiotic in its own way. This means that we can define dysbiotic microbiome by defining the absence of eubiosis. And in our experience, we found that all the microbes we analyzed in the Circle project, but also outside of the Circle project, are defined by a gamma distribution. What I mean? If we put the microbiome, the species, the taxa that are comprised in a microbiome in a decreasing order in relative abundance, this will describe a gamma curve. And this gamma curve is described by two parameters, the alpha that is known as the shape parameter and the beta that is called the scale parameter. And by varying this parameter, we describe different microbiome structure. And this all for saying that alpha and beta can be used for classifying the microbiome structure. And once understood that, we have to define the boundary between eubiotic and dysbiotic structure. And this can be done by this strategy. And this is the strategy used for the human gut microbiome, but we use that also for poultry and pigs microbiome, and also from dogs, but can be used for any kind of host-associated microbiome. First, we aim to reconstruct the gamma distribution, the gut microbiome, they are human, from a cohort of healthy individuals. And then calculate alpha and beta of each distribution for identifying the range for these parameters associated with an eubiotic microbiome structure. And then test this method on other gut microbiomes coming from other study of healthy and unhealthy individuals. And here an example for the first step. We calculate alpha and beta based on this 1,480 sample from healthy humans, and then we plot the alpha and beta of each individual in each microbiome in a B-plot, this for defining a sort of biotic area that constitute our reference for the further analysis. And then we test on other sample. So what we found that healthy individual microbiome from healthy individual map inside the eubiotic area. The majority of the microbiome that comes from an healthy individual affected by ABS, IBD, CRC map outside the eubiotic area. And when we try to map on the graph the microbiome that comes from other hosts, in this case gut microbiome for poultry of the Circle project, we found that the majority of these sample map outside the eubiotic area. This for saying that we can use a unique approach for all the microbiome, but we need specific reference for each kind of microbiome we are trying to use. And here a test on a dysbiotic microbiome, just for giving you an example. So here this red dot is the dysbiotic microbiome. They fall outside the biotic area. In this case, the microbiome show a lower beta and a higher alpha value of the parameter. And this means that the distribution is more peaked, so few genera in this case that are present at very high relative abundance, a lot of genera that are present at a relative abundance lower than 1%. And we can then try to plan a personalized intervention based on this result. And the strategy is first to compare our dysbiotic microbiome in terms of relative abundance of each genera that compose this microbiome with the reference. And we can identify some genus in this case that are present at a higher relative abundance respect to the reference range. And we call that as positive dysbiosis. And then we can find some genera that are present at a lower percentage respect to the reference, and we call that as negative dysbiosis. We can then extrapolate some information from a microbial network of correlation for enlarge the possible target, the potential target for a possible intervention. And then we can suggest a possible intervention strategy based on this result. And all this is combined into the universal reporting that is now submitted for patent by the University of Bologna. And thanks very much.
Thanks, Simone. That was very, very interesting. So how important is the ranking on the part of the curve that is, let's say, the low abundance one?
Yeah.
So if you swap the order of some, or have you look into your metagenome, are there examples where the order is swapped, or basically there is also an order very well defined in the basically the lower part of the curve for the genera that have a low abundance count.
So the method is independent from the order of the taxa. So we just need to put that in an increasing order. And then if we found something that is out of the alpha and beta parameter, we then search for some taxa that are outside of the range of the eubiotic references. So it's not important in this case.
Yeah, thanks very much. That was really stimulating for sure. I wondered, is there any enrichment in opportunistic pathogens in constellations where microbiomes would be outside of the eubiotic range?
What we found is not really an enrichment of pathogens, but of course an imbalance of the equilibrium among microorganisms. That means that some microorganisms can be considered as not really pathogen, but is present at very high percentage, for instance, and this constitutes a threat for the equilibrium of the microbiome in analysis.
And for those taxa, would you have enrichment, say, in virulence factors or antimicrobial resistance genes, for example? I mean, that they wouldn't be classified de facto as pathogens, I guess. That's
it could be, but we don't investigate that on this kind of result.
Yeah, sure. All right. Thanks very much.
All right.
Congratulations, Simone, for the great presentation. Has you patented the method of analysis? This would limit the utilization in practice or in scientific?
For the moment, yes, for the moment. But we have tried to develop, we have patented that for trying to develop a more in-depth approach, because this is just the initial step for getting something or more specific and effective on the production system, on the microbiome we would like to study.
But what is the final purpose? To create a tool that can be used from outside?
Yeah,
okay.
Exactly.
Thanks.
Exactly.
Congratulations, Simone, for the nice presentation. Nice job. Just because you used the data from circles, did you consider even the antibiotic administration to the animals? Because this can reduce the pathogenic bacteria, but is an artifact, is not a real eubiosis in that case.
At the moment, we discard this kind of data, so we use just the data, also from the literature, not only from the circle, that comes from hosts that are defined as healthy by the researchers. Yeah.
So in the future, you consider to include, for example, the genetic background of the animals. For example, the FUT1 or MUC4 genotype can be very different in terms of response to.
It could be an option. It could be an option for sure.
Or the dietary, or the dietary
we are
intervention.
just at the beginning of the story, so we have to implement several different applications for seeing if the approach is valid also on other hosts that we haven't tried now.
Thanks a lot. Congratulations again.
Thank you.
Thanks, Simon, for excellent talk. So Anna Karenina principle suggests that community composition varies a lot between the sample in dysbiotic state. So do you also consider in this case like distance between different samples? Somehow it accounts in your model?
So, sorry, say again, you consider difference in?
So community composition differs a lot following the Anna Karenina principle.
Yeah.
So beta dispersion increases. That's what it says. I'm just wondering in your cases, like did you also account in dysbiotic state how the sample distribution compared to community composition distribution?
So not really. So we just reconstruct the community. So there are several different communities, but we don't focus on the overall diversity that we get, but just in this, the single diversity in the specific diversity we get in each sample for depicting, for retrieving the alpha and beta value. And then once we get that the sample we are analyzing are all from healthy individual, we suppose that are in a biotic configuration, and we use that as reference. Then when we investigate with other samples that are not used for the references, we try to do the analysis blinded at the first, so in order to classify if the microbiome that we are analyzing are in eubiosis or in dysbiotic configuration, and then we get the statistic that they show you that for what concerns the healthy one, they are all inside within the range of the biotic configuration, and for what concerns the unhealthy individuals, the majority of that are outside the biotic range of variability. So this is the result that we get.
Okay, thank you.
Okay. Thank you.
Thank you, Simone.
Thank you.
Now I'm going to introduce the last speaker of this session, is Kati Rasanen. She's a research scientist at the Natural Research Institute of Finland, and she coordinates the sustainability assessment effort of the Circle project at LUC. She has extensive experience working on numerous projects involving life cycle assessment, and her work focuses on evaluating and enhancing the overall sustainability of product chains in microbial application. Her presentation is going to be about assessment of environmental and economic sustainability of the Circle product chain. Welcome, Kati.
Hello, everybody. Can you hear me? Yes. So I'm representing our group here. So, for example, thank you for the whole group, and thank you especially for Usman, who has calculated our environmental impacts. He's sitting down there, and thank you also Karetta, who is absent today, and she calculated the cost impacts. How the part? What's this? Okay, thank you. Okay, so let's go to see what was the aim of this research. So we studied environmental and cost impacts of the Circle's value chains. We used life cycle assessment as a tool. And life cycle assessment is a tool to study the environmental, social, or cost impact of different chains or different products. And so it's also based on the standards. And for example, when we calculate environmental impacts, so the most common impact category is the climate change, which is also called carbon footprint. Other things can be also measured, for example, eutrophication, ecotoxicity, toxicity, and so on. So in this study, we used Recipe 2016 and used this method for our impact assessment. We did the economic allocation for the big chain, not the broiler chain, and we also made some sensitivity analysis. So, as I already said, we calculated cost impacts with life cycle costing, and the same system boundary was used there also as in the environmental impact assessment. We got the data from the circles, poultry, pig, and salmon. We also got it from the spinach chain, but the results are not ready yet, and that's why we are not showing them to you yet now. So we collected the data from the chains with the Excel sheets, and a lot of data was obtained. Thank you for partners who collected that. So for example, the data was obtained from the feed, energy source, animals, animal size, and so on. And we filled some data cups with the secondary data sources. We used, for example, EcoInvent and AgriFood databases, which are very common used in LCA. And also some literature was used. And just remind you that our functional unit was 1 kilogram of animal live weight. That is a unit when LCA results are always shown. So let's go to the results part. So this is now the environmental impacts of the broiler chain. We can see on the columns here that in the middle there's a conventional chain, and on the right side you can see microbial treated chain. And in this case we can see that microbial treated animals, the chain was more environmental friendly measured with these indicators. And the reason of how the impacts came. So most of the impacts were affected by the feed production, and also methane and nitrogen dioxide emission played a significant role in global warming. We also get some environmental credits from the manure fertilizer and wastewater irrigation. According to our sensitivity analysis, we changed the feed between the chains, and according to that result, so microbial treated animals were not so environment friendly as they were in the original data. And in our other sensitivity analysis, we changed the energy source to be renewable energy source, and that was also good news for us that the environmental impacts decrease in all impact categories except freshwater eutrophication in both chains. Let's go to look at the big chain results. And here again, on the middle, you can see the conventional chain, and on the right, the microbial treated chain. And so there wasn't big change between the chains in this study. As broiler chain also, that the feed production was the major contributor of these emissions. And also methane and nitrogen dioxide emission played a role as in the broiler chains, and we also got some environmental credits, and that's why the columns are under the zero in some cases. And according to our sensitivity analysis, we could show that if we change the energy source to renewable one, so then the impacts are less than using just fossil fuel sources. Let's go to see our final chain, which was the salmon chain. And there the results were opposite than in the previous meat chains. So in the middle are the conventional, and the right side are the microbial treated fish. And there we could see that the microbial applicated fish, they were the environmental impacts were a little bit higher than in the conventional chain. And the reason was that the microbiome treated fish requires a little bit more feed, and the feed production was the main contributor in the environmental impacts. I'm going to skip the next one. So let's go to see the LCSA results. So this is the cost impact now of the poultry chain. So if we compare the conventional and microbial applicated chain, so we can see that the microbial applicated chain, the cost impacts were there a little bit higher than in the conventional one. And the bedding material costs were the main contributors there. But in the next slide, you can see the big chain results, and there were not so big differences between the conventional and microbial applicated chains. Let's go to summary and conclusions. So I repeat again that microbial treated broilers represent lower environmental but higher cost impacts, mainly due to reduced impact from feed. But there were not big changes in big chains when we compared the microbial and conventional one together. And feed production was the main contributor there. And if we change the renewable energy for the energy source, so then it decrease all the impacts except the freshwater eutrophication. So we success that optimizing feed and changing energy source to renewable one could improve the environmental performance in both chains. And in salmon chain, so conventional salmon fish production was more environmentally sustainable with studied impact categories than a microbiome treated fish. But I would like to highlight here that we haven't considered the health issues or anything about the quality issues. So our study is based on these indicators. So that's why it would be very important to also take into account other indicators and aspects concerning this fish. And the fish production was the main contributor. So optimizing feed with microbiome application could improve the environmental performance. Thank you.
Thanks so much, Kati, for the nice presentation. So, it's not my field. So, but as a take-up message, for what I understood, the point is that for improving the sustainability and the cost, we need to act on the animal production, but also on the feed production.
Exactly.
So we need to join two projects.
Yeah, exactly. And also the energy source. And yes. Okay, thank you.
So thank you, everybody. This concludes session 2, and we're going to have a short coffee break of 15 minutes. So let's be back at 11:10.
So let's start with the third session entitled Microbiome in Plant Food Chains, Innovation for Productivity, Quality, Safety, and Sustainability. So it's a pleasure to introduce my colleagues, Giorgia Palladino, with the presentation entitled Genome Mining for the Discovery of New Plant Growth Promoting Bacteria. Giorgia is a postdoc in the Unit of Microbiome Science and Biotechnology of the University of Bologna. He specialized in microbiology, microbial ecology, and molecular biology at the University of Bologna, and she focused on characterizing host-associated microbial communities and studying microbial-based responses to anthropogenic impact in natural ecosystem and food system. Giorgia, please, the floor is yours.
Thank you, Simone, for the nice presentation. So today I'm going to go a little bit more in deep in one of the topics that Marco already presented to you in the first presentation. So since this is a mixed audience, I'm going to explain very shortly what is genome mining and what are plant growth promoting bacteria. So genome mining is the systematic collection, analysis, and interpretation of big amount of data. And in particular, in our case, we are talking about whole genome sequencing data with the goal of discovering significant nutrient relationship pattern or trends. As for plant growth promoting bacteria, these are plant-associated and beneficial soil-dwelling organisms which can colonize the rhizosphere of the plant, so the narrow zone of soil that is specifically influenced by the root system. And PGPBs and their host can coexist in a mutualistic relationship where they both have benefits living in close proximity between each other. And in this case, we can apply genome mining for PGPB discover because genome mining offers the opportunity to scrutinize those PGPB and also looking for active principle mediated by the enzymes and secondary metabolites so that we can understand how these PGPBs can influence the health and the growth of the plant. So in this context, we explored the zonation of Vitis vinifera microbiome in Vino Nobile di Montepulciano, PDO area, as Marco already mentioned before. We, in this study, we describe the concept of terroir as the uniqueness of the relationship between the wine and its territory of origin that is composed by local pedoclimatic, biotic, and abiotic factors, and also traditional agricultural practices. In this context, the microbiome part of the terroir has been very neglected so far because everybody was focusing more on the agricultural practices and the abiotic factors, but we want to focus also on the microbiome part in order to characterize the different terroir associated with the territory of Vino Nobile di Montepulciano, with the aim of explaining the distinctive regional characteristic of the final product, that is the wine. This is the research paper that we produced in the Circle project. It's currently under revision at Communication Biology. And the territory of Montepulciano is composed with, I guess this one, 12 additional geographic units called AGUs. We performed a sampling in 4 time points, July, August, September, and October 2022. And we collected a total of 336 rhizospheric samples and 56 bulk soil samples. We characterized the microbial community with the bacterial community characterization through 16S rRNA sequencing, and also we characterized the fungal community with the characterization of the ETS2 region. And finally, we also characterized via metagenomics 1 rhizospheric sample and 1 bulk soil sample for each vineyard. I didn't add it to this presentation because this is very, very new, but very recently we also added the targeted and untargeted metabolomic analysis of the wines from 6 different AGUs. Okay, so what we found is that the differences in the rhizospheric microbiome were explained by the geographical distance between the different AGUs for both the microbial and fungal community. And this segregation pattern was robust to seasonality, agricultural practices and management, vine clone type, rootstock family, altitude, and soil composition. So the geographic component was the main factor influencing the rhizospheric microbiome. In particular, we found some specific microbial taxa, so 24 bacterial and 6 fungal genera that distinguished the 12 AGUs that we considered. And the relative abundance pattern can be described in a sort of a gradient in the Montepulciano territory from west to southeast or vice versa. So we superimposed the relative abundance of our taxa, both bacterial and fungal, on the territory. And you can see these gradients where red is a high relative abundance and yellow is very low relative abundance on the territory of Montepulciano. So what we found is that the AGUs of the southern eastern area show overall greater potential for phosphorus solubilization, while the AGUs in the western area showed a greater propensity for ACC deaminase production, whereas the nitrogen fixing potential was more homogeneous in the entire production area. And also we were able to reconstruct 37 MAGs, metagenome assembled genomes. 4 of them were belonging to core taxa that covered a broad range of function for soil fertility and plant health. And in order to confirm this result, the MAGs characterizing the AGUs in the southeastern side of the territory were equipped with metabolic function for phosphorus solubilization, whereas the ones characterizing the western part carried more genes for ACC deaminase production. So how can we apply the knowledge that we've discovered now for a practical application? So basically, we confirm the importance of protecting the microbial terroir and biodiversity to ensure the production of a high-quality traditional wine with specificity at a very local scale, in this case, AGU scale. But of course, we want to, or at least try to isolate the bacteria in order to verify the presence of the genes we identified through metagenomics and the genes that are responsible for plant growth promoting functions. And of course, this future practical application aimed at protecting the conservation of the microbial diversity of the area and so the quality of the wine production. So this is what we are going, well, this is what we did next, and we are still implementing this approach with more samples. So far, we collected the soil from the 12 additional geographic units during the year after the first sampling, so in September 2023. We performed some serial dilution and plating on a rich culture medium to facilitate the creation of a library of glycerinated microorganisms that are potential PGPBs. And we were able to identify 30 different microorganisms under aerobic conditions that we biobanked as glycerinates. And then we processed those samples for DNA extractions, and we processed the DNA for sequencing in order to sequence the bacterial genome of the isolates. So those assembled genomes were processed to identify the presence of PGPB genes according to a custom database. This is a preliminary data set, and this is what we have done so far, but we are also implementing this data set with more genes. And yeah, of course, I don't think you can read it, but the important thing is that all the genomes that we were able to sequence presented at least one, but a lot of times more than one of the PGP function that we investigated in our data set. So actually, it was possible to isolate AGU-specific PGPBs, and in this map, you can see that we put in each AGU that are on the background, the color dot represent the PGP function that we identified in the AGU and the bacteria that we were able to isolate for that AGU. So each set of AGU-specific PGPB isolate provided the whole set of PGP function. And these, of course, can also represent a possible microbial binocular to protect and promote the local quality of the wine product. In conclusion, as I said, we were able to isolate the 30 bacterial strain from the soil of the 12 AGUs in the Montepulciano territory, and all of these genomes that we sequenced encoded for PGP function, and they can be thus considered PGPBs. Of course, we can speculate about possible future application with the in vitro test, experiment on model plants, and afterwards directly on the vineyards. So far, these isolates are available in the Circle Biobank, and they represent a new potential AGU-specific circle bioinoculants that can be used to protect the local biodiversity of the wine territory and the quality of the wine production. And you can find all these results in the confidential deliverable, the 3.5 provision of at least 12 genomically characterizing plant growth promoting microorganisms. So this is the picture of our group. Marco already showed you before, so thank you for your attention.
Hi, great talk. Two questions, actually. One is, were you using any sort of selective culturing, given the fact that you only isolated 30 from bulk soil, I'm assuming? I would have expected more microorganisms to be cultured. Second is, in your results, you had Bacillus thuringiensis. So I was wondering if you had any sort of pesticide, biological pesticide application on your crops, given that BT, is one of the most famous biological pesticides.
Yeah, okay. So as for the, no, sorry. I wanted to go back. Okay, this one. Yeah. As for the medium, no, we didn't use any selection medium so far. So this is all generic culture medium. This is still a preliminary screening, so it is possible that we can obtain more strains in the future. But yeah, we didn't use any selection so far. Sorry, what was the second question?
BT, Bacillus thuringiensis in your selection of strains?
No. So yeah, it's difficult since we are interacting with a consortium that is very big and it's not very into the scientific aspect of the things. It's also a little bit difficult to understand exactly the agricultural practices they are using, but these are all standardized methods. So yeah, we don't have like very precise information, but it should be all organic crops. Simone, you can correct me if I'm wrong, but yeah, this is like all the information that we have. Yeah.
So, Giorgia, how much the rootstock, the type of rootstock, confound with the characteristic of the soil of the different zones? So I imagine maybe one zone has a, I don't know, a pH or organic matter content compared to another. They do have the same rootstock, or are they different?
It depends on the AGU. Yeah, I don't remember by heart because there are so many different types, but we do have the entire set of metadata for all the AGUs. They do have different type of rootstocks, and the soil has different characteristics, but we normalize those data for, well, we found that the geographic provenance of the sample has more influence on how the sample distribute on the PCOA compared to all other confounding factor, including the rootstock type and the characteristic of the soil. So the geographic origin is the main factor in shaping the distribution of the samples, much more than all the other confounding factors. So even if the rootstock is different, the origin is more important.
Thank you. So it's my pleasure now to introduce Taina Pennanen with the presentation entitled Wood Fibers to Feed the Soil Fungi in the Agricultural Fields. Taina is a research professor at the Natural Resource Institute in Finland, is a microbial ecologist, and her work is focused on soil microbial communities, particularly on the fungi in soil and plant-associated interaction from the perspective of host plant genotype variation and plant pathogen resistance. Thank you, Taina. The floor is yours.
Thank you. And here's the group we have been working together during this very nice and long project. And as you see that circles is about modulating microbiomes, but our approach is different. We tried to find out what kind of food we could provide to microbiomes to modulate the microbiomes, because in large scale, landscape scale things this might be, and soil aspect, this might be more persistent solution because microbes are very vulnerable to indigenous competition alike. But then what kind of food sustainable soil microbes like? And then we have to go back to the where do the most agricultural soils originate? They do originate from forests. And I love this old paper, Plowed Up Wood Wide Web, and they showed, and this still holds, that the biggest difference between agricultural soils which have been deforested from forest is that fungal mycelia has been broken down. Bacterial communities are more similar, at least in a higher taxonomic level. What else has been happening, has been observed during these years when lots of agricultural soil has been deforested, is that there's long-term carbon loss in agricultural soils globally, also in Finland, and that's why there is, you may know this international 4 per mill initiative, where we could turn the 0.4% carbon loss to gain in soils, agricultural soils. That would be really dramatic positive influence for climate change in combating. But as I said, in forest soils, organic layer, this carbon amount is increasing still. And why is that? At least I've been taught that the lignin of wood-derived lignin is the persistent and difficult source of food that microbes, it's persistent, like wood residuals are persistent in soil, but well changed roughly at the same time when these circles project started. So there started to become a new evidences that more or less 50% of the stable carbon in soil is not coming from plant residuals, woody residuals, but it's coming from microbes. So dead microbe is a good microbe because microbes eat, flourish, then they die, and then small mineral particles are persisted, stained in soil. And there's lots of evidences that particularly fungi are important. So from fungal tissue, most persistent carbon particles come. And in some other studies in boreal forests say that even up to 80% of the carbon stored in soil is coming from fungi and root-associated fungi. And quickly, the combination why carbon and microbes are important, because soil microbes are important for this, making these agricultural soils carbon-rich and also persistent, structure of the structure of the agricultural soil is depending on aggregate formation, which is quite largely made by microbial extracellular polymers. And so this is like a cycle of good between carbon and microbes, or we could say maybe circles of good. But how to increase these microbes in agricultural soil? Crop residues are not quite enough, particularly now when climate is changing and temperature is increasing. That activates microbes, and this is not quite enough, even though like permanent grasses and those, it's known that they are helping. Organic farming may help, and root biomass is helping because that's more tolerant for decomposition. But then, that's what we were studying, soil amendments. This is a study which raised my interest quite a few years ago. They put forest litter in agricultural soil, and what they found was that they found a decrease in really bad fusarium infection, which is very common nowadays in agricultural soils, and increase in biodiversity. But I didn't know if that was due to the substrate or due to the imported microbes. But like taking up forest litter and putting that in the agricultural soil, it doesn't make any sense. But we have some other side streams, because in Nordic countries we are making a big part of the paper in the world. We do have lots of these forest side streams. And so that's why we made a hypothesis that started to look at what if you take up these forest-based side streams and see how it activates, or does it activate the microbials in soil, and particularly we were expecting and hoping to get some positive impact of fungi. And we have two experiments, sawmills and sawmill side streams, microcosm experiment, and paper pulping side streams in the field. And then about this bark, just quickly, that when this process started, it was like 10% of the round volume is bark, and that was mainly combusted to generate energy, and which is really waste of resources. And that's why, because this is Circles project, we were not looking at the fresh bark, but we were also looking at the hot water extracted bark, where when from sawmills, bark is first extracted with water to take all the carbohydrates and make bioethanol, for example, and then the rest is subjected to anaerobic digestion to make biogas. And still there is lots of woody fibers present in these side streams. And shortly about this result, this was only microcosm experiment, but like a important sign for us. Of course, all the back materials increased microbial biomass, bacteria, but particularly fungi. But this wasn't very big surprise. But surprise was that there were big differences in soil type. That the increase was like in different kind of soil material about organic carbon percentages. And then I also show only quickly, you can imagine, of course, there were lots of community changes in microbiota, and in fresh and hot water extracted bark, which was mainly fibers there, lots of increase in fungal communities, and they different functional groups of fungi, also endophytic, mycorrhizal, pathogenic, but also very like a well-known beneficial fungi. They increased in this fresh and hot water extracted bark. Anaerobic digestion, because we combined it with manure addition, because that's the common in the agriculture, that affected more also bacterial communities. But then to the field experiment, which I think it's very relevant. And do we see similar results here? But now coincidentally, at our institute, researchers, soil researchers, which were studying leaching of nutrients from agricultural fields, they had established 2015 a field experiment where fiber sludge from paper pulping, this is like a, you can imagine a handkerchief is moist and then it's like put into small pieces, this kind of waste material from paper and cardboard process. And then there's two different kind of sludges which were like amended with nutrients. Another was calcium stabilized, and third one was composted fiber sludge. So I think the setup is really good because then there's pure nutrient, pure fiber, and then fiber amended with nutrients and composting process. And there's lots of these in Finland, and I cannot even imagine how much more if we think of the other Nordic countries. So, the same happened. Particularly, we were so happy to see that particularly fungal community was diversified. And for example, Arbuscula mycorrhizal fungi were found to be increased in the field, and some indicator fungi which have been found to be typical for organic farming. And also indications of a trophic level, increases in trophic levels of the soil communities. And then back to the carbon. Of course, we were measuring what is happening to the soil carbon, because that would mean the long-term change. We were measuring that during all these years, but these results are from last autumn. And these are much more, I think, important than the total carbon, because, of course, if you add a substrate to the field, you will see more carbon first before it's respired to the sky by microbial activity. And it wasn't, by the way, now I say that to prevent leaching, this is very important, effective. So it doesn't happen any leaching. So all the carbon which is lost by your carbon dioxide. But so this is like a measurement where we take into account the permanent forms of carbon. Particularly mineral associated organic matter is the organic, and carbon is the most persistent one. And unbelievable, in 7 years, there's a 19% increase in the POM and MOM fractions in the field. And calcium stabilized fiber 10% and zero fiber little bit, 1.5%. So, but this is because it is said that carbon changing in soil it takes at least 5 years. And this is very, to see any changes, and this is really logical. But who is doing this? And now we come to the metagenome and metatranscriptomic work. First I say that we have done only we have been sampling for metagenomes and transcriptomes, these soils, for 5 years after the first amendment to see how long this effect is lasting. And then last year, so it means 7 years after the second amendment, to further see that time effect, and also because the point would be the focus, because now we see change in carbon, and what kind of change in microbial communities we see, and then we could know better what kind of organisms are responsible, probably correlatively responsible for the increase in persistent carbon. Particularly this high carbon use efficiency functions will be important. But some results we do have. As I said, after 5 years, microbial biomass wasn't increased anymore, so all the biomass was respired to the sky. But some, as I said, some traces about this persistent carbon was increased in soil, and also some activities were there. This is a metatranscriptomic result. Some changes in activities, and there were lots of changes in bacterial activities. I won't go into those, but this is the only slide I show about this 5 years after the first amendment, and this is the only part which we have ready now, is that, and this is I think quite promising because lignin has lots of ester bonds, and esterases are higher, glucosides which are and glucoside hydrolysis which are connected to cellulose decomposition. So it looks like that even though microbial biomasses are leveled off, then there's still microbial activity going on, and they are probably decomposing still slowly these woody fibers. But as I said, hopefully lots of more results will be coming in the next few months, and we can focus on the taxonomic level of microbes and also functional level. As I said, carbon utilization efficiency will be highly interesting, and if we can focus that on the specific microbial groups, that's important. And then if we will hopefully know the names for the microbes, then we can also plan some other kind of ways how to increase these microbes to the fields. And what was interesting, I think, why this composted was so much better than the others. There must be, I hope I would be chemist now, because there must be happening something in composting the woody fibers, why it makes it so different from because my guess would have been it is a zero fiber, but so it's composting. Something is happening for the fibers in composting, which makes that even better for the carbon sequestration aims. Thank you.
Are there any questions from the audience? No? So thank you very much.
Thank you.
So it's my pleasure to introduce the last speaker of this session, that is Davide Bulgarelli, with the presentation entitled Structure, Function and Manipulation of the Horticultural Microbiome. Davide obtained a PhD in crop science at the University of Milan before leaving his native Italy for Germany to take a postdoctoral position at the Max Planck Institute of Cologne. In 2013, Davide established his own research group at the University of Dundee, where he is currently a reader. Davide's team aims at contributing to sustainable development by dissecting the genetics of plant microbiome interaction. Thanks, Davide. The floor is yours.
Okay. So, can you hear me? Well, okay. So, yeah, well, it's a pleasure being here. It's a pleasure being in a place where you don't need to cross a border when you land in an airport. And I would like to start by thanking Giacomo and Deborah for having set up this very, very interesting conference. Thanks, Simone, for the kind introduction. So let me start. No, I won't. What? That's it. Okay, great. So what I'm going to do in the next 12 minutes or so with you is giving you a very brief introduction on the plant food chain and the microbiomes that are there. What we have done in circles, in the interest of time, focusing on one of the focal species we've been working with. And then I will close up in highlighting where we would like to move next. Okay, so let me start with this slide. Probably in this room and even outside, people are very familiar with the microbes and the microbiome defined by our own digestive tract. What I would like to convince you of by the end of this presentation, that plants are not that dissimilar. So if you look at below ground, where is the... I can't move, right? If we look below ground, the plants, so this is the root system. So this is what plants have anchored to the ground. Georgia already illustrated that there is a thin layer of soil that is very special. It's called rhizosphere. This is changed by the plants in a number of ways. And this hosts a microbiota that is distinct from what is called bulk soil or unplanted soil, so a soil with no plants. And then microbes can colonize also the above ground part of the plants. Effectively, the way I see plants is a series of microhabitats for distinct microbiotas that can help or interfere with the capacity of the plants. So let me take a brief stop here, and let me wear my lecturer hat. So if anyone else in the future asks you the question, have you heard of biomass or fruit production derived from carbon dioxide? The answer must be yes. Plants have done so for the best part of the last 450 million years. So look at this beauty. So look at this plant. So it's, for the benefit of the people online, I'm pointing at a plant that will be 50 meters tall. This is the result of the conversion of carbon dioxide into biomass thanks to sunlight. Okay, so let's move on. Circles, I mean, one of the reasons why I was so excited being part of Circles is that the concept of lab in the field. So we were experimenting in actual food production areas. And for plants, those are farms. And I will focus on spinach, one of the focal plants of the project. And what we had is essentially two production sites in the north and the south of Italy that represent two different pedoclimatic regions, even within an individual nation. And what we have done in the first phase of the project is try to understand what is the composition of the microbiota inhabiting those spinach plants in the various microhabitats I discussed before. Right? And operationally, it works like this. So this slide illustrates what happens in one of these microhabitats. Let's assume this is soil. You have these different colors, highlight different microbes that are present there, and then you can name them. So this is the real scenario. What we try to achieve by looking at the DNA of those microorganisms is essentially reconstruct in silico the composition of the microbiota. Now, because of, I won't go and delve into what ASV means. It's simply for the benefit of understanding, so different bars means different microbiota. And these are as a quantitative value, so you may assign number of sequencing reads to a given unit, and then, of course, elaborate a quantitative analysis like this one. Now, if you are within the microbiome science, you know perfectly what I'm going to show you right now. For people that are not familiar with, this is probably one of the most abused type of graphical illustration microbiome science uses, and I'm no exception to that. So let me guide you through the figure, and then we'll discuss how we interpret it. So this is an ordination. Every shape in this figure represents one of the sample of the spinach plants that we have collected and characterized for microbiota composition. It's difficult to perceive, but the different shapes are highlighting the different microhabitats and then are color-coded according to the farms we sample it. Now, the key of interpretation is very simple. If the microbiome of two or more samples would be identical, the two shapes will overlap. Because you have distant shapes, so distances in this ordination means that there is something something that, from microbiome point of view, distinguishes those samples. And what we can see here is a so-called microhabitat effect. So it's difficult to perceive, but basically we are moving from the unplanted soil in this part of the graph progressively towards the leaves. It's like an ideal gradient from the unplanted soil to what the plants can host. So spinach plant hosts a microbiota that is distinct from the surrounding soil in the farm. And the second aspect is that, it doesn't matter. We have a separation between the northern fields and the southern field that you may argue, because your farms are more than 1,000 kilometers apart, this is certainly not a Nobel Prize discovery. What is relevant for us is that let's say the congruence of this distribution between unplanted soil to leaves is maintained between fields. This makes in the position to try to manipulate even fields that are far apart by tapping in what are their commonalities, and in particular the microbes that are sustaining this structuring. Okay, the way we have done is what Georgia told you before. Essentially identify relevant microorganisms, adding to the plants, and see what happens. What we have done, and this is a difference compared to what Georgia told you before, is that we use existing collection and we mine the genome of a number of microorganisms, bacteria in this case, that we deem relevant for our manipulation. So what you're looking at here is a phylogenetic tree, and again is a graphical representation based on the whole genome of those microorganisms. Now let's start to looking at the outer circles, the one colored. So the named circles that you can see here are the bacteria that we have sequenced. And then you have specific names here. And these are actually genomes that are present in the literature. So we want to make sure that what we have sequenced makes sense compared to what was already available. Then the tree, what let's say scientists call tree in the middle, is the relatedness of their genome. Again, if everything collapsed in one branch, means that their genome is identical. Because you have this separation in different clades, means that there is something at the level of what they can't do, in this case for plants, that differs. This manuscript is in review, and hopefully soon you'll read all about it. For the time being, let's have a look what we had in mind. So spinach cultivation is very quick, especially in the northern fields. Within 2 months, we move from seed, almost 2 months between seeds to harvest. And what we have decided to do is applying those microorganisms that we deem relevant for the spinach microbiota in order to sort of activate the spinach microbiota in case of something happens during this cultivation, maybe a stress, drought, presence of pathogen, and see again what happens to the plant. And this show you what we have done. So we have done a first intervention. At the same time, we distributed the seeds in the field, a second one at an early phase of development, and then we wait up until a week before harvest, before the harvest of the plants to sample the microbiota and see what happened to the plants, and then there is the harvest and you quantify the effect on the plant. Okay, so that's basically is the rationale of our hypothesis. So if we supercharge the spinach microbiota, it'll be better equipped to cope with environmental fluctuation. Okay, that's on paper. And it works? Yes. There is no audio, but that's what looks like in reality. I've told you, we have actual farms. So we need to be able to distribute those cocktail of microorganisms to surface that are, the minimum one was 1 hectare. And therefore you need an agricultural machinery. Now, the prediction of this slide is that whenever you develop something for application in microbiome, has to be compatible with farmers, with what farmers are already using. Otherwise, it would be an extra cost. Okay, that's it. So the tractor goes, these are spinach plants growing, and just wait because there is the label there, which clearly says Circles Project, right? I mean, it's not cheating. It's not that I pick up, as something from, it's coming. Look at beautiful plants. There we go, Circles. You see the logo? That's real. Okay, good. Let's move on. Now, first of all, the treatment, what you look at are the yield on the plant, because this is essentially what farmers are paid for, and that's what matters. Now, the two treatments, the one just at the beginning and the second application after the development, did not really differ between themselves in terms of average yield. But this is a yield difference. So this would have been the comparison with the uninoculated samples. And that's the only difference, right? I mean, we have a strip that was uninoculated, a strip that was inoculated with one treatment, and another strip in the same farm inoculated with the other treatment. And that's for the first time in my life, after having written numerous grant applications and papers that I say that what I'm doing will ultimately help farmers, that have a proof that this is true, because those are production sites, and in this base case scenario, there was a monetary gain. Of course, I need to warn you that is not always like this. There are fluctuations, and we have still figured out what the reason for. But this is a microbiome project, and we want to know what happens at the level of the microbiome. Okay, that's another graphical representation of the microbiome. Let me guide you through, and then we'll discuss what this means. Let's start from this part. Those are the control. Those are, let's say, the plants that were not treated. We are looking at the phyllosphere microbiome, so above ground, because this is what is harvested, so we were interested in see what happens in what you harvest. This is the south, and this is the north. There is a number of bacteria family here. I mean, at the moment, it's not very relevant what they are, at least for now. What is relevant is that this is the distribution within the control. So if our intervention had no effect, your expectation is that these panels should be identical to this one. They aren't. So there is a difference. And this difference is bona fide caused by the application of our intervention. Now, let's see whether this animation is still here. Okay, so the apparent paradox that I allude here is that if you now read to this list of microorganisms, there is no association with the type of microbes that we have applied. We know what we have applied. And our expectation is that we should have more in the applied plants compared to the control. We don't. That's a violation of the Koch postulate in microbiology, if you want. Okay? But what we have observed, and that's we think is probably the major breakthrough from our project, is that alongside the absence of what we wanted to see, we have a decay on certain groups, in particular Pseudomonas and other Enterobacteriaceae and Erwiniaceae, that are potential pathogens for plants. And they can cause this type of disease. So before arriving at a stage like this, where the plants are compromised, we speculate, I would say, that's the right word, that the application of our inoculants is somehow activating the plants, either the plants or the combination between the plants and the microbiome, to keep at bay potential pathogen. And that may be associated with the gain that we observe in terms of biomass. And that's what we are now mining our data to try to see whether we have an experimental support to this. And with that, I'm wrapping up. I show you that we can sample the microbiota in the field, and we have consistent patterns despite the diversity that you may have in different farms. Together, I would say, with Georgia, now we have expanded the farmer's toolkit. They have done it for a plant like, let's say, an orchard and let's say, a tree. We have done it for an herbaceous crop. And then we have now a way to try to understand how this benefit materialize in the field via, for instance, by improving the quality of the plant. But I must stress that the project will be over within, let's say, at the end of October, but there are still a number of questions that we hope to develop further, especially in terms of stability of this application across season for the benefit of farmers. And that's all from me. No. Sorry, it's not all. That's a snapshot. And I'm here talking, and I'm here talking because there is a number of people and a number of institutions that contributed to generate all the figures I show you today. And that's all, of course, my gratitude goes to them and the farmers. Those are real farms. They took the risk of experimenting, even when the experimentation didn't go according to the plan we wanted. So I'm very thankful to them, and that's really all.
So are there any questions? Yeah.
Fantastic talk. I think you are really onto something interesting. I have 2 points where I think make sure could be fluctuation, impacting fluctuation. One is, please remember to take the microbiome of the seed. Mm-hmm. Spinach seed is crucial, and it varies with the microbiome on its surface, and both concomitant and internal. The other part is that sometimes when you are adding microbes, like you did in foliar spray, you have traces of the substrate you grew it in. Mm-hmm. That substrate can actually be cutting away the Pseudomonas or benefiting the others and so on. So please remember that, because the feeding to the microbiome can sometimes be more important, as our Finnish colleague said, than to add the microbes.
No, absolutely, yes. So we are working on those points, but yeah, you are absolutely right. So the difference could be also, or at least in part, associated together with what we have had. No, you're right.
Thank you, Davide. Actually, my question is related. Can you elaborate a bit on how did you spray the spinach? Was it just the biomass filtrated directly from the cultures, or? Because that was my question, if there were other metabolites or residuals of the media.
I really wish our colleagues would be here, but I assume maybe Nicola can comment. So the liquid preparation is just a filtrate from the culture that we applied? No? No, the powder, the pellet goes together with the seeds. But then we had also this additional intervention where we sprayed. So the question, if you can elaborate how this preparation was made. Okay, okay. So something that certainly we need to iron a bit, yeah.
Other question? Yeah.
Maybe we can move to the panel discussion since we have
Okay. Okay, so I close this section. I leave the floor to Davide for the panel discussion. We have till 12:30. I mean, yeah, for the panel discussion. Thank you very much.
Okay. Ho bisogno delle biografie. Adesso magari chiedo a Deborah. Okay, just a technical issue. Give me a minute. No, less than a minute, because we don't have time. Okay, so I'll sort it. Hello again, long time no see. I'm still here because I have the pleasure of introducing now the roundtable about what we have seen in this last session and further development about what we have seen this last session. So I should have had the bio sketch for each one of the panelists. I don't have this piece of information. Okay. So I will let the panelists to introduce themselves to you before kicking off the discussion. Okay. Ladies first, Eva, please.
Yeah, okay, so I'm a bit an outsider here because I'm not really working on microbiomes in general, but in a very specific type of organisms, which are arbuscular mycorrhiza and ectomycorrhiza, which are fungi. So Lene, I totally agree with you, don't forget the fungi. And so since more than 10 years now, I'm working in Germany in a company producing inocula containing these fungi to help the growth of plants in agriculture and crop production and also in forestry.
Okay, so next, Michele.
Hi to everyone. It's a big pleasure for me to stay here. Congratulations for this conference. The topic is really important for the world in general, for agriculture. And I'm specialized on use of microorganisms in agriculture. And I'm an agronomist. I'm also a professor in the University of Timișoara in Romania. I'm Italian, but I'm professor there. And I have the opportunity to teach to the students the use of microorganisms in agriculture. And also, this is probably the most important things for all of you, that I'm also an advisor for LG Italia, that is a Italian company that produce bioinoculant. I mean, that use the microorganisms that for biocontrol, for biostimulation, such as of inoculum that Davide was showing us on spinach, very close, and it's really interesting that we are talking about microbiome, the soil microbiome, and now in the world, there is a big opportunity to use this technology now to cover and protect our crops.
Okay, and the last panelist has been already introduced. I'm very glad that he's here with us, Stefan from AIT in Austria. So let's do like this. Because I feel you may be shy, I'll start and I'll kick off the roundtable, and then we'll open up the discussion to the audience and people that are online. So the first question I have is, and it's a bit a provocative one, because I'm also in this field, right? I mean, we have seen in these three presentations that we start to, and also in the keynote introduction from Stefan, that we know a lot about the microbes that are interacting with plants. They know who they are, they know where they work, they know with whom they interact. So now the question is that why farmers haven't replaced all the chemicals with the microbial inoculants or capitalizing on the microbiome already now. So the question I have for you, I mean, is known that the, let's say, success of this application is not constant in time and may be fluctuating. The question I have for you is, what do you think are the trade-offs that are preventing a wider application of microbiome, let's call it microbiome solution, to agricultural production and crop production in particular. Who wants to start?
It's regarding, I think, first, the efficacy of the product in the field compared to chemical one, which was very, very active. And it's very difficult to reach an efficacy of a chemical product. So the farmer, what they see, okay, they have some yield improve, they tackle some plant disease, but they lose also a lot of money. Even if the product is sustainable and safe by design compared to chemical products, we don't have a good efficacy. Plus, sometimes some products are working in some terroir here under some paleoclimatic condition, but they have a lower effect in some other. And there is different product now on the market. Plus, I think we can discuss this later on about not only single microbe in the product, but some other, just to be short. What I think from the field, but I'm more scientist, so maybe you can say more.
Well, I'm also a scientist, but David will be even more provocative. This microbiome story is not new, and just people to know, the first patent for the use of microbes in agriculture was at the end of the 19th century with the Rhazorbia. So actually, maybe as scientists are not so good to convince people, or I totally agree with Stefan. We have difficulties to reach the efficacy level of a chemical product. There is also a big problem because these chemical products are easy, are accessible, and are cheap, and farmers tend to use them because it's easy for them to have access to these products. The effects are very visual, very immediate, and they're easy to use. And there is also, of course, the cost. The problem of costs. This is very variable between regions. In some regions, farmers will be ready to spend some money, but for some other regions, for example, they told us we cannot expect to pay more than, for example, 100 euros per hectare for something which we are not sure about the efficiency.
Yeah, I'll tell you my point of view. Really, it's not really the problem of the cost, in my point of view, and also it's not also the registration problem. Because now, in this moment, starting from the mycorrhizas product, bacteria and fungi, or some consortium, we have really, really good products that works very well. They have very good capacity, because we don't work with just one microorganism. We work in a protocol to try to help the plant to make this interaction with the microorganisms. The problem, in my point of view, is not in the technology, that now we have everything to make a good products. The products is not only I multiply microorganisms. It's a product, it's a technology. It's something more than only a microorganisms. It's to make something together that works in some direction. We are talking about to use the science in technology, But the problem is to change the paradigm. I mean that we have to teach the farmer that the soil is something different. Before we saw the World Wide Web, the interaction between the plants, the interaction, the endophyte between the microorganisms. I mean that the point is that we have to try to teach the farmer that there is a different way to make agriculture. That is possible. For sure, we have to write to the farmer starting from the university. I mean that all these things that we are discussing today, we have to try to bring to the students and after to the farmer. I mean, the big problem is to try to share this information. And the big topic is, we are talking about agriculture, we are talking about the soil. You show in the first your presentation, the first slide is incredible, and starting to see the soil that works like the humans, that microbial point of view. The point is there in my point of view, is because I'm working with a company that produces technology, but I don't have to convince the farmer that the product is okay. I make it wrong. I have to convince the farmer that this technology can make something important for all of you. I mean that we have totally to change the paradigm. The soil is alive. the microorganisms inside is part of the soil. You know, I'm soil scientist, and I saw some presentation about the soil, very nice, and it's important. But also, we have to try to change this paradigm. You know, the soil is not only. This is a big topic. You know, because most part of the farmer manage the soil fertility only using the fertilizers. But this is not the truth, because the soil is totally different. Because the soil fertility, the biological part now, today we saw in this incredible conference, as a consistent part. It's not only a part. This is probably the most important part to manage the soil fertility. And if we can teach the farmer that this part is important, and you have to manage how you can teach and to showing that there is a microbiome in the soil. This microbiome interacts with the plant. This microbiome can go in the fruit, can go in the human, can improve the human health. We can change totally the vision. And also, and just to remark, the problem is not the target, you know, the pathogen, you know. The problem is the environmental. I mean, I have to take care of the soil to take care of the plant. This is really the most, in my point of view, you know. You see, in my vision, I see it be a very great opportunity for the farmer to use these knowledges to make something good for the crop, but for us.
Yeah, but you know, when the farmers, they have a disease, they want to have a quick solution to prevent at maximum, because they are losing a lot of money. And if the products, they have not the same efficacy than chemical product. Of course, in organic approach, it will be fine. They will take organic solution. But also, for example, in the vineyard with downy mildew, exploring in Italy, in France, when they have a solution like copper hydroxide, which is cheap, they have a limited amount to use, they will still use this because you will have better protection than some biological product. And they will use the biological product not when the disease will be too high, because we know that the biological product will also protect disease when there is a low pressure.
Yeah.
And when we use some fertilizer, also most of the time we have good effect at the beginning, but with the yield we have the threshold. So we've just applied things, and it's the problem. The problem is when the farmers, they have real problems, they want a quick solution.
Yeah.
And you can teach them for IPM strategy, etc. But when the problem is there, it's too late.
Yeah.
Okay, I think that we can open to the audience. Please. It's you. Yeah. No, sorry.
I can speak up.
He's coming. Yeah.
I don't take all the responsibility on the farmer. We have been in an era of pesticides and chemicals and fossils for so long. So it's not just the farmer. What we need to look at is change management. How do we make the change? Let us look forward. And I'm I would like to ask the panel, what do you think about the new regenerative agriculture thinking? And that there are immediately many big food companies, Nestle and many others, who only would like to have supply of agricultural products from certified regenerative agriculture. Is that one of the drivers to go for, to get the biologicals in?
Yeah, say something else. I will try. I'm totally agree with you about the management. The problem is not the target, but management of the agro-ecosystem. If you work in agriculture, you know that you have a pathogen, but you don't have to fight the pathogen because it's part of the biodiversity, and you have to manage the problem. This is the big topic. I will tell you two things very fast about agriculture regenerative. One, a little bit polite. I mean, that is something important. You know, I love this type of approach, and we have to preserve. We have to preserve this idea, because it can be used from other company to sell something. You know, I don't want to be strong, but this is important. And I will tell you something, my point of view. biodynamics, biological agriculture, symbiotic agriculture, regenerative agriculture. The question is, we need to put some adjective close to agriculture, or we need just an agriculture that respects the plants and the soil? This is the point. If you want to make a marketing, I love the marketing. Probably it's one part of my job. But in my point of view is that we need an agriculture. The agriculture where the soil, probably, this is my love, is the most important things that you have to know. First, you have to know, because we didn't know, and you have to preserve. This is my point of view.
Yeah, from my point of view, as a scientist, of course, I want to preserve soils. Now, I'm also more pragmatic, and I agree that if big companies would go for this kind of product produced by respecting the soil microbiota, that would be a great incitation to go towards this kind of practices, because actually, for mycorrhiza, money do count. It's really something which is difficult for farmers to afford this kind of products. Now, if they know that they can sell the products to big companies, that would be a game changer. Definitely.
It's good that you provide some solution for farmers. We were talking about the cause first, why they don't use some biological product, why are the problem. It's nice that we provide some different solutions. There is a lot of putative solution to help farmers. And also to help for the biodiversity, for the One Health approach. There is a lot of different solutions. Then, what about the farmer, how they take this biological, yeah?
Yeah, that's another point.
And the mycorrhiza, they are working well in the field?
It depends. So, of course, it depends on all the interactions in the soil, of the chemical properties of the soil, of the crop. Yeah. So, it's a difficult point.
I will tell you something about the mycorrhizas. We are talking about one of the most important microorganisms that we can use in agriculture to increase the healthy plant. It's normal that sometimes it can work, sometimes cannot work. But this happens also in the chemical products. How many times you use the pesticide and doesn't work? The problem was not the products, because it's a chemical. The problem was probably the farmer that was not applying very well. Every product can have is not 100% efficacy. Doesn't exist. This is the protocol I can use. The mycorrhizas, for example, that I love, are really useful for the farmer. It's normal that you have to teach the farmer, this is the point. You have to give the products, or you have to explain, you have to teach the farmer why they are using this technology. This is the point.
That's a problem, because...
No, we have to do.
We have to do, but it's also the problem if someone will try a product without any effect, they will not buy it again. And of course, it's all a matter of education. I always try to explain when I'm talking with farmers that there are, of course, direct effect on the plants, biofertilization, bioprotection, so on. But there is also the indirect effects by structuration of microbial communities around the root system. But it is something that we need to communicate about.
Yeah, you have to spend time to explain, to change the paradigm.
And but I noticed also something that we discussed with farmers in the frame of several European projects. And they said that they somehow feel far away from these projects. So we kind of use them, or use the fields they have to test our products, but they are never really actors in the project. Yeah. And I can say, since 12 years I'm working in this field, I was just once at a conference where the farmers were here presenting results. And I think it would be really crucial to involve them as actors, because they are the key players in this change. We can do the best products, we can have the best knowledge about microbiomes. If nobody is willing to use these products, nobody will win. So that's important.
There is a question over there, Arne.
So do you have a comment on crop rotation in light of microbiomes?
I do.
Yeah,
I do.
You're a scientist here, yeah.
So it's very briefly, when it comes to herbaceous crop in particular, so the microbiome is rewired each time you put another crop in that field. So if you have wheat, there will be the wheat microbiome around the roots. I mean, talking about what happens in soil. Then the next year you have sugar beet, and then the sugar beet will reshape in the way the sugar beet likes. There are elements of commonalities. This is based on what has been researched and published by ourselves and others, but there is also an element of species specificity. Now there is, I mean, the common sense, or let's say the intuition would be, if we have rotations, then we'll maintain the healthy soil, because then the minerals and the microbes will be more uniform depending on the plants. There are exceptions to that. So plants do not have an adaptive immune system, but their microbiome may help them in, let's say, fending off potential pathogens below ground if you keep growing the same species over and over. This is a phenomenon called suppressive soil, and is very common on, for instance, cereals. So small grain cereal, often farmers do wheat after wheat, because this keeps at bay potential pathogen. And this is one of the phenomena that has been studied.
I want just to say something about, for example, the rotation or other techniques that can be useful also for regenerative agriculture. This is just to remark that it's not only the use of one product or one technology. It's to use new technology, but use also the best practice agriculture that we know from a lot of years. You have just to put all together, and you can, for sure, we can make something innovative for the farmer. It's something that we have to consider together, not only the inoculants, only the biostimulants. We have to consider all the management that could be a little bit different, and we cannot mix too much traditional. I mean, the use of pesticide with the inoculum to make something wrong. No, you have to make something totally new. This point of view.
I fully agree, but what do you think when there is some Italian people here? You know the situation of Xylella in Puglia, which is a catastrophe. There is no solution on the market. There is no solution at all. What do? We don't have the technology. We are developing, but it will come in the future.
Yes, I'm totally agree with you. But this is my point of view. The problem about Xylella, you know, that it's a really big problem. It's a political problem. It's, there was a very big misunderstanding there. But just to remark your point, the problem is not the target, or the problem is not a product. There is technology in this planet that we can use at this moment, but it's to change the paradigm. Because if we can teach the farmer that the problem is not only to destroy the fusarium in the soil, but to manage your biodiversity, probably we don't have this type of problem. But there, you know more than me probably, that there was something more than Xylella or other things. Yeah. But this is my opinion.
Is Taina over there raised their hands?
I think that the magical word is biodiversity, you know, because it's really important.
Just a
Sometimes
comment.
we forget this.
I agree with the panelists, all of you, but one thing came to my mind. Could it be that the problem of cost for the farmers, the problem is in the time perspective of the cost, because fertilizer and pesticides, they ensure the crop for the next year, but improving soil, which might be then more susceptible to bio-inoculation, also if the soil is in good condition. That takes time. That would be probably a political question to improve the time perspective in it, because it's only time. It's not the cost, because after all, the cost will be lower if this is sustainable.
Yeah, I totally agree. So I discussed with a couple of farmers who made the transition, but the problem is a lot of farmers are not accompanied to do this. So these two persons I discussed with were lucky enough to have enough funds to do it. And they told indeed that by good practices like no-till, protecting the microbiome, and using the right cover crops, using the right crop rotation, they managed to retrieve yields like with the chemicals, but it took 5 to 10 years, which is something which can be really too long for a farmer who is already very short in finances. But I agree that politically we should definitely accompany this movement and help farmers going to this transition.
Marco.
Yeah, this is our species, human being, is not able to program in long term. So probably this is why we are going to be extinct in few years. So anyway. And look at the missions. So they still go on since years and years and years. So we are doing anything at the global level, and I think it will go on like this. Anyway, the point is, this is not my field. My experience with the consortium Vino Nobile di Montepulciano is when we speak about that, they always say, I do like this, my product is perfect, why I do have to change something? And the point that we are trying to do is to say, okay, you are doing like this, and you have these microbes that since several years they are doing the work for you, but with climate change, maybe you lose your microbes and you lose part of your products. So we need to protect your microbes for the future changes. So it's nothing that you have to change the way you produce the food, but maybe you make the way you produce your food resilient to future changing, protecting your microbes. This is a different way, a different approach.
Yes, please. The gentleman over there.
Just a small comment, because I can completely relate this discussion with the earlier antibiotic use and AMR resistance problem. So there was the thing where, like, if there is a strong infection, you have to use the antibiotics. And it's the same case with plants and crops, because for farmers, it's their earning. So I think once we can tell them that what are the negative effect of that, and maybe they can use in more sustainable ways, use the chemical when absolutely requires and immediately requires, and yeah, to have a proper balance of the chemical versus organic and probiotics or prebiotic. I think that's the solution. Yeah.
Okay. Let's do an experiment to implement this microbiome solution. How many of you would be ready to pay 10-15% more for their food tomorrow if there is a label with written, this has been produced by replacing, I don't know, X chemical with microbiome application? Raise your hand. Yeah. Okay, let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we are 50-50. So I think that that's probably also something, no, there's zero, but the very last thing we're going to discuss. What this discussion has been is that the science probably is mature enough. Still, we need to learn a lot. Well, learn additional things, but the science becomes mature enough. Is the politics and probably the economy side. I mean, agriculture is an entrepreneurial activity. So when we speak about farmers, I work with them and I enjoy these 6 years. We were discussing about their livelihood. Okay, so if the inoculant doesn't work and my microbiome science doesn't work, that's it. I mean, I turn page and we try another experiment. For them, this hectare is gone. So I think if we look at the history also of the European Union, when it was built in the 50s, food production in Europe was basically on their knees. And thanks to the agricultural policy, within a reasonable number of years, was self-sustainable and more. Exporting led on to also distortion. So I think that we need to start thinking or lobbying at the political level to have a different approach and a different subsidy or vision for that. And thinks this is going to help. I mean, this is what I've, let's say, diluted from this discussion and what I've seen by working with farmers. So science arrives until a certain point. When it comes to livelihood and society, politics has to chip in at a certain point. Otherwise, this transition is unlikely to happening, at least in full. What do you think? What do you think?
I agree with you completely.
Okay, so
I totally agree with you.
Speaking about food, lunch is ready.
Thank you.
Thank you very much.
Sorry, we will have the lunch now. It will be at the first floor, so we need to take the elevator and go down. Okay.
Good afternoon. So we are going to start the section 4, microbiome and circular strategy in poultry and swine food chains. The first speaker is Yatkele Tenerife Makonnen. The title of the talk is a microbiome nutritional strategy to improve sustainability of the poultry food system, the Circle case study. Yatkele, I can pronounce it. Okay. Okay, it's not easy.
Yes.
He's a PhD student in food safety at the University of Bologna. Thank you very much. The floor is yours.
Thank you. Green one?
I think this green one is to go on, and this to go back, and this is the laser.
Yeah. I think it's not this one. This is not this.
Oh, nice. That's the backup slides of mine. So you need to go on. No, because quite some backup slides, so we should go farther. Go, keep going.
It's not going.
It's not going.
So what do you want to say about this?
No, yeah, if you click, there are already a few manuscripts that we managed to publish.
Poultry is around so fast. So it's difficult to catch.
Maybe shall I go to?
Yeah.
Okay, good afternoon, everyone. My talk is on microbiome nutritional strategy to improve sustainability of the poultry food system, which is part of the Circle case study. So as we all know, food is a basic necessity, and it is one of the components to build healthy and dynamic societies. But it needs to be nutritious, safe, and affordable. But currently there are different challenges going on in a global basis, which affects the safety and also the affordability plus the nutritional value of the food. So different strategies they are going on in order to tackle this. Among the strategies, employing of microbiome in the food safety in order to produce safe, quality, and also sustainable food is one of the topics. So from this point of view, the European Commission have the Food 2030 pathways, which include the exploitation of microbiome. And within this, the Circle project, the use of the investigation on microbiome is undergoing. So I will talk with the poultry food system in order to identify microbiome-tailored strategy to improve productivity, quality, safety, and also sustainability. This study was done in commercial condition, and within the food system of the poultry, we have different components, which start from the farm and transporting to the slaughterhouse, and also having a slaughterhouse process. But our case studies or the intervention is only conducted in the food, the animal food in the farm levels. So our approach, methodologically, we have three approaches we followed. The observational trial, which is part of, which aim to identify microbiome markers, which are associated with better performing poultry houses. And then after having the result from these microbiome markers, an intensive systematic literature review was conducted in order to identify nutritional interventions, those nutrients which can support the microbiome markers that we identified in the observational phase. Finally, after having this data, there is an intervention trial on those nutrition which is selected to be employed as intervention study. Then from the result of the observational study, what we found is there are a group of microbiomes that are correlated, mostly correlated with better performing poultry houses. Here we have two groups of poultry houses, those better performing and low performing. This is a relative term. So when we say better performing is regarded to technological application in the farm, some of the facilities which can improve the animal welfare and the animal health issues. So this is a term which is relatively differentiates the different farmers. So in this scenario, when in the better performing poultry house, we found the microbiome group Citrobacter and Ruminoclostridium 9, which is persistently correlated or represented in those better performing farms. Then there is another group which we can consider as a negative, as a negative actions. So the bacteroides, a group which characterized or represented in low performing farms. So for the next step, what we consider is Citrobacter and Ruminoclostridium to be considered as a positive action, whereas the bacteroides, which is more represent in the low performing one, to be as a negative action. In the systematic literature review was conducted, and a total of 518 papers are screened. And from these total papers, 213 in nutritional intervention was extrapolated from 145 original research articles. So these 145 original articles, which are dealing with the microbiome-related nutritional intervention, which can also modulate the gut microbiome of broilers. So these nutritional interventions, they can categorize as probiotics, prebiotic, phytobiotic, like that, and also enzyme. Then when we evaluate the impact of the microbiome-related intervention, so the groups of the intervention which can improve the digestibility and weight gain. There are interventions which is help to control pathogen and the microbiome modulation, which is our main target, and also improve meat quality and also alternative to antibiotics. Here, as far as the antibiotic control concerned, pathogen control and also alternative to antibiotics is main issues for this crisis in foodborne pathogen and also the emerging of antimicrobial resistance. In the modulation result, we see that there are a group of intervention which can enhance lactobacillus. There are a feed which can also, in general, a group level, can enhance lactic acid bacteria, and the other groups which is also more, which is can able to improve the microbial, multiple microbial community. From this literature review part, the paper is published out of it. That is the systematic review on microbiome-related intervention, which can help to control the colonization of foodborne pathogens. Then, as a bit of the conclusion from these two results, for the observation and also systematic review, and with the discussion of the partners of the project, like the poultry company and also the feed producer, finally we decided to use the enzyme muramidase to be used as an intervention, to be added into the nutrients and then to follow the production growth of the poultry, the broiler, and to analyze those data. So muramidase is a lysozyme enzyme which is playing a role in improving gut health and also innate immune responses of the broiler. This muramidase is acting on the cell wall of the bacteria in order to act on the peptidoglycan. From a study conducted, it is shown that the experimental trial, this muramidase can decrease Clostridiaceae, and also it enhances Bacteroideaceae and Lactobacilliaceae. And also the result from this study shows that it will also help to control, to reduce the abundance of Clostridium perfringens. It is one of the foodborne pathogens, and also Listeria monocytogenes. Then in the intervention study, we make a comparison or data from the animal performance, but in general, we found that there is no impact of the was observed between the two farms, the poultry houses, which is labeled as CM and ZDAR. In the categorization, ZDAR is the one with better performing, whereas CM a bit lower performing one. And the indicators like the mortality, conversion, and also the European production efficiency is not significantly changed before and after the intervention. And also we observed there is a higher level of mortality in one of the farms. The blood biomarkers are also analyzed before and after the intervention, and we saw that the significantly affected or increased blood biomarkers are observed during the intervention phases after adding the muramidase, which is those indicators are an indicator for stress conditions of the animal, and also those indicators can show the presence of inflammatory conditions that occurs. But this is not associated with, it may not be associated with the administration of muramidase, but it will be something else which needs to be investigated. So when we see the microbiome dynamics, it is shown that the nutritional intervention, which modulate or shifted the overall SECA microbiome composition of the animal in a low performing one, rather than the expected or the one which is selected is the high performing. Comparing to the Bacillota and Bacteroideta, they are among the beneficial one. We found that there is a significantly increase of Bacillota was observed in low performing farm, the poultry house, and also a significant decreases of Bacteroideta was observed in the low performing the field. But there is no any differences observed in those expected farms, that is the high performing one. The other group is Lactobacilliaceae. So we analyzed the Lactobacilliaceae levels, and we saw that there is significantly decreased level of Lactobacilliaceae in high performing farm, and also the genus Lactobacillus in low performing as well. So here, as we saw from the systematic literature review, Lactobacillus is the most modulated by most of the group of nutritional intervention, and also it is analyzed to have an impact, beneficial impacts on controlling of foodborne pathogen, and also helping for improving the gut health. Clostridiaceae, the other groups which is analyzed, and from this we saw that there is a significantly increase of the family Clostridiaceae. More specifically, we see Clostridium and Butriococcus. So the Clostridium, which is significantly decreased, whereas Butriococcus, one of the beneficial, which is also identified as a group in the high performing one in the observational study. The Citrobacter and the Ruminococcus level are analyzed, and we found that there is no significant change which is observed on Citrobacter. I don't know why the slides. But in case of Ruminococcus, significantly decrease was observed. We tried also, rather than or in addition to the beneficial microbes, there are also the analysis for those foodborne pathogen or a selected pathogen. This pathogen, which is selected because of the report of the European Food Safety Authorities, through it is One Health zoonosis report. There are a group of microbes, for example, Clostridium perfringens, Clostridium botulinum, and Listeria monocytogenes, which are significantly decreased in the high-performing farms. Of course, Clostridium botulinum in low-performing also. But there is a significantly high level of Salmonella enterica was observed in high-performing. But here, as you see, the relative abundance is not greater than 1%. So having the live cell is more or less depend on the level of the culture methods or detecting by other techniques. The other group of pathogen, Campylobacter, which is significantly decreased in both high and low performing poultry houses even after the intervention, whereas Salmonella, Escherichia coli, and Chlamydia are significantly increased in those high performing farms. So again, the level of the relative abundance is less than 1%. But still it needs the presence of live cell, which is very important as far as the public health and also the animal health issue concerned. As a conclusion, the selected intervention or supplementation of muramidase didn't improve the animal performance and blood biomarkers, but this it will be discussed with different reasons. For example, this study, it is conducted, the observational result is done in 2019. So now the intervention is done in 2023 after 4 years. So it will be difficult to compare those data. And also there is a change in biosecurity measure applied after the COVID-19 pandemic. So that is also the other challenges which may also affect us, the microbiome community. And the other challenge will be the commercial setting. You know, in the commercial setting, a large group of animals, handling of those all animal performance parameters will be challenging. And also there are maybe different factors undergoing which can affect the microbiome level, which in relation to the performance. The intervention addresses only one segment of the poultry food system, while it should be need to look at the other part of the food systems. Only, as I told you in the introduction parts, we follow the intervention only in the feed of the poultry, but there are different components which are found, like the transport, the slaughtering process, other. So these can be considered as one of the factors which need to be addressed and also need to be taken as a point for the future. The SECA microbiome was modulated as expected, even though there are some variation within the significant changes with the target population was undergoing. So I would like to thank my supervisors and also our collaborators, University of Bologna and also the European Commission for all and the collaborators in the company. So thank you.
Any questions? Thank you very much for the presentation. So, so there is no impact essentially on the intervention. The main impact you show is on a more beneficial microbiome structure in the animal, with much more lactobacilli and less pathogens, essentially. Yeah. Do you think that that can be relevant in when you go on with food processing? So maybe can protect from pathogens that can rise during food processing, food storage, so after the production. I mean.
So you mean having low level of the pathogens? So here we can consider different reasons. As I said, we can reduce the pathogen in a way of the production. Of course, it reduces the level, but there are different factors which can also associate it, which can be maybe contaminated after the transport and also in the processing or in the slaughterhouse and going to the distribution too. So these factors are there, but still we can say that having a lower level of foodborne pathogen in the animal, it will go a bit lowering. but still it needs a lot of study to make all the other factors. Yeah. Thank you.
So I think that we can go on with the next speaker.
Okay, thank you.
The next speaker is Paolo Trevisi. The title of the talk is Developmental Dynamics of Pig Intestinal Microbiota and Relationship with Health and Welfare Parameters. Paolo Trevisi is a full professor in animal nutrition and feeding at the University of Bologna. His specialities, he has specialties in animal nutrition and feeding at the University of Bologna. He is member of the Study Commission on Pigs and associate editor for the Italian Journal of Animal Science. Paolo's research is focused on the interaction between nutrients and host for the gut maturation in pigs to increase robustness, as well as dietary intervention to prevent antibiotic use. Paolo is co-author of 129 papers with an H index of 28. Thank you, Paolo. The floor is yours.
Allora. Okay. Thank you for the introduction. Good afternoon to everybody. So, yes, I'm not a microbiologist, I'm an animal scientist, so take care on the terminology, so sorry for that. So today I introduce to you the factor that can affect the gut microbiota in pigs, and it is a quite complex matter of study, and not all is understand right now. So you can see that there are several factors that can modulate the microbiota in pigs, like genetics, background, antibiotic treatment, diet manipulation, age, live weight, environment, and management practices. Okay. Smallest than expected. Okay, so the problem of the pig production is that we have several phases in their life, and so there are several movements from one place to another place. So this increases the confusion in study, for example, the natural development of the animals. We know that there are some waves of colonization in pigs from the farrowing through the growing phase, and the first colonization is through the facultative anaerobes, and there are other waves with bacteroides, Prevotella, and at the end anaerobe like Clostridia. So there are several waves of this colonization affected by different environment and the dietary changes that happen during the life of the pigs. The first point of contamination should be the intrauterine life or development, but this is something in which there are high debate. So there are some paper where seems, especially in human, that the first colonization starts in the uterus, while other trials, like even one in our labs, highlight that there can be some fragment, bacterial fragment in cordon blood, for example, but a real colonization or a real transfer from the mother to the fetus of microbiota is quite difficult. About newborn and suckling, so the post-farrowing phase, there are several factors that can modulate the gut colonization. The first factor is the vagina microbiota. So during the farrowing, this is very important in terms of, and you can see here that immediately post farrowing, the vagina microbiota is the most important contributors in the microbiota, and it remains till more or less the day 7. Sorry. Later on, start to be important together with the vagina microbiota, the slatted floor, so the environment in which the animal stay, and in post-weaning, the mother microbiota
seems something like a contribution, but we think that is more related to the change of the diet, and so that is more close the microbiota of the mother with the microbiota of the pigs. And anyway, this is one of our study in the framework of circles, where we observe, as expected, that the time have a strong effect in shape the microbiota, but even the mother effect is very important. So the contribution of the mother microbiota to the microbiota of piglets, at least during the suckling phase, is quite important. The weaning phase is a peculiar phase in animal production, because poultry don't have this phase, while pigs at around 21, 28 days of age is separated from their mother, and they experience several stress, like the change of the diet. They pass from the milk from the mother to the solid diet with other animal proteins and starch, for example, transport, separation from their mother, vaccination, change in temperature, environment, etc. All these factors affect the gut health of the animals. So increasing the inflammation, there is a sort of transient anorexia due to the adaptation to the new diet, and this implies a remodulation or a gut inflammation that at the end modify the gut architecture, and at the end we arrive through the modification or modulation of the gut microbiota, that is, in case of the presence of pathogen or real stressful weaning, can hesitate in diarrhea, so protein and energy waste. So it's something that we want to avoid. But anyway, really, weaning is a cascade of events that is, if is not well managed, can provoke a serious disruption of the gut microbiota, like in this picture. Okay, the most important thing is the increasing of the gut permeability, and so this implies the use of antibiotic, and so on, the modification of the microbiota. So where we focus our attention is in the postnatal phases, so from the suckling phase to the growing phase, and it's not just a matter of taxonomy, but even the function of the microorganism change over the time. This is just an example. I use circles data just as example for my presentation. So we have a lot of data from circles and are very interesting even to explain or to better understand the modulation of the microbiota during the pig life. So, as I said, not just the taxonomy is important, but even the modulation of the gut microbial function. And in this case, we were lucky because we arrived even to analyze the fecal metabolome, so to cross-connect the potential, the metabolic potential of the bacteria with the real metabolome of the animals. And even in this case, you can see that in the time modify the fecal metabolome. This data arrived a few weeks ago, so I have just some graph, but we need time to analyze it. But it's very interesting that there is this kind of time effect in any case. Again, the environment. The environment is a very important. Okay, is a very important factor that modulates the pig microbiota, gut microbiota. So, in the, as you know, probably circles. One of the objectives of the circles was to explore or to observe the microbiota in a typical rearing condition. In this case, so we designed a specific trial in collaboration with a group of Veronesi. Where from the same farrowing unit we split the pigs from the same litter in two different production chains. These production chains were fed with the same diet in order to isolate as much as possible the genetic factor, at least from the mother, and the dietary factor that we will see later that is an important factor. And so we want to check how or to verify how the farm environment and management can modulate the gut microbiota. The first point is the alpha diversity that is quite interesting because in the production chain 1, pigs were treated with antibiotics immediately post-weaning, and here we have a drop of the alpha diversity, and this is very interesting. After that, there is a sort of reassembling of the microbiota, at least for the alpha diversity, but never come to be similar or equal to the other production chain. This could be something interesting for your model, so I hope. About the beta diversity, very interesting is that the time point 1, so during suckling, there was no differences in the microbiota between the pigs previously assigned to the chew production chain, while when the pigs were moved to the chew production chain, the microbiota differ. Here are some data, again, from the observation on the taxonomy at genus level, so with the 16S analysis, because before we analyzed the 16S of all the animals, and later on we analyzed the metagenome in collaboration with the group of Marco. And you can see that there are some genus like Megasphaera or Prevotella like Erchinoliaceae that are quite stable over the time in the chew production chain. And this is something that we discuss later and seems quite interesting. We repeat the same analysis with the shotgun metagenome at the species level, and we have the same results. That is not something that falsely happened because the level of analysis is very different. So at the time point 1 during suckling, no differences in terms of beta diversity, while in the other time point we have a differentiation of the microbiota in the two production chains. And in this case, the species, we arrive to identify the species that can characterize the two production chains, and one was Megasphaera L. cederi, and the other one is Prevotella SP P592. This is interesting because Megasphaera is related even with a symbiotic effect with the lactobacilli, and we have even lactobacilli in, for example, in T3, that is the end of the weaning phase. Again, we analyzed the gut microbial function. I just show you the potential of this kind of analysis because at the T3, that is the time point after the antibiotic treatment in the farm, looking from the enrichment pathway, we have the enrichment of dark metabolism and metabolism of xenobiotics. So the antibiotic intervention, more or less 4 weeks before, maintain a different microbial function potential of in the pig's gut. The next factor, very important, is the diet. After the observational phase, in circles, the objective was to modulate or try to modulate the microbiota to be more closer for the microbiota, the best microbiota with the worst microbiota, even if we don't identify a real worst microbiota in this case. But anyway, we try to harmonize the microbiota between the two production chains. And thanks to the collaboration with DSM in one intervention and Silva team with the other intervention, we collect several data on the effect of the diet on the gut colonization. This is just an example on the intervention 1, where We utilize 3 feed additives, Enterococcus faecium, that is a probiotic, an acidifiers, that is benzoic acid, and an enzyme, that is amuramidase. Here, what is interesting is the interaction between diet and farm. That why I like this because what is very important, even in the practice, is that the same intervention frequently don't have the same effect in all the environment. So this interaction is very strong, and at least for pigs. And so is an intervention can be very farm specific, or you must clusterize or characterize the farm for some specific characteristic to be pretty sure that this what you expected happen. In this case, the 2 production chain were selected to be divergent for the production, so it's something that we expect that in some cases the risk was that we have different effect in the 2 production chain. And here is just an example of what the characterization of the microbial metabolic potential in the 2 production chain with the factor of the diet. But again, what is interesting is the fecal metabolome, where we observe that not just the age modulate the fecal metabolome, but even the dietary treatment. So control versus treatment, you can see that we have a different fecal metabolome. And what is interesting is that the ACO and LCO, so the production chain with high performance and production chain with low performance with the control diet are quite similar, while the treated group are quite different. Again, the interaction between microbiota and the host is very strong, and this can modulate the immune maturation or immune system activation. It's a very strong effect of the microbiota, and was the basis of the effectiveness of the antimicrobial growth promoters or the antibiotics. So in this case, when we don't use external factor that modulate the microbiota, really there is a strong interplay between host and the microbiota. In circles, is a lot of animation. Okay, a lot. Okay, we divided we checked looking for a cluster for the immune system. In this case, we have 2 clusters independently from the production chain on the number of the lymphocyte. And what we observe is that the beta diversity can affect the number of the cluster and the farm per lymphocyte cluster number. So there is a real interaction. In this case is just an example of the thousands of data that we have on the interaction between the microbiota and the immune system activation in pigs. Here another example are the significant correlation in L farm, so low performing farm, between the blood cell formula, white and red, and the potential metabolic capability of the microbiota. So all these interactions must be explored in the next month because they serve several quite interesting information. Here is a paper published in Animal with the results, side results of circus, where we're looking for the effect of the level of lesion on pigs, on the pig's physiology and pig microbiota. So we can see that L is low lesion score and high is high lesion score, differ for their microbiota, as well differ for the oxidative stress status and for immune system activation. So there is a strong relationship between the immune system activation, the oxidative stress system activation, and the microbiota, but especially for pigs is a very important topic related with the aggressive behavior in pigs that anyway change the animal physiology and so change all the environment. The last point is the issue of the core microbiota. That is something that is under debate, at least in our scientific community. And from our data, we observe that there are lactobacilli that are represented in different time points and in different production chain. So what we want to check was if there are a sort of core microbiota over the production chain. And so we used this approach where we included the taxon that are present at least in the 50% of the samples. And what we observe is that Lactobacillus amylovorus, Lactobacillus reuteri are the most present independently from the production chain in time point 2 and time point 3 that from our point of view were the time point in which the microbiota was really stressed for the highest use of antibiotic, for the change in the diet, and for the strong pressure on the immune system maturation. So just to some consideration to open hopefully the discussion, the vaginal microbiota is the first sources of microbes for the newborn. A time-related gut microbiota ecosystem is clearly demonstrated, as expected. The dietary intervention can modulate the gut microbiota profile and its metabolic potential, at even the real metabolome. The farm and the diet can modulate the fecal metabolism of pigs, and okay. There is a close connection between the welfare, physiology, and fecal microbiota profile. So even in the view of the animal welfare, it is important to understand if there are some specific microbial profile that can modulate the aggressive behavior. And the co-evolution host microbiota can be understood by using a multi-omic approach on different matrices. That is something that is very challenging. We have a couple of data to work on in the next month, and we hope to pitch some new information for the pig production chain. Thank you very much for your attention.
So when you showed us the ordination for the two production chain, I found very interesting that you separate the production chain on what is the y-axis, which should account for, let's say, less variance compared to the main, and let's say the shapes were almost aligned and mirrored each other. So the question I have, could it be that siblings, piglet siblings, are assigned to the different production chain, or this is not a scenario that can happen?
No, the sibling pigs were assigned to the two production chain.
Okay,
So
that's
is
okay.
that, yeah.
Okay, that's it. That's
Okay.
it. No, that's clear then.
Sorry.
I'm very impressed with your presentation. I have to say this was really such a massive amount of very, very thorough and important data. But of course I have a critical question also, and that is, you say adaptation to antibiotics was taking place, but I didn't hear, or maybe I skipped it, the antibiotic resistance, occurrence of antibiotic-resistant genes. In Denmark, we have MRSA. Even all of our farm helpers almost are latently infected with MRSA, which means that when they go to hospital, they have to be in isolation and all these horrible things. So this is a big thing. It's worker safety and so on. And also risk really upgrade, if the microbiome is propagating MRSA or antibiotic resistance genes. So did you include this monitoring of
Yes, this morning were presented the data on the spread of antimicrobial resistance in pig food chain, and was the data from the observational phase. So it's something that the group of, I'm not a microbiologist, so it's the group of Marco that analyzed the data. And so yes, there are, of course, we take into account the presence of antimicrobial resistant genes in our microbiota. There is another, because even this morning a little bit. Okay, so there is another presentation on that, really on that. The point is, okay, the antimicrobial resistance is a complex matter, so there are vertical or horizontal transmission, and in this case the antibiotic was amoxicillin, so it was not a critical antibiotic for humans. So there are several points to consider, but of course the work and the objective of this trial and why Group of Veronese is involved in the project is really to reduce the use of antibiotic in the farm system, because I work a lot with our colleagues in Denmark, like Charlotte Lauritsen, and if you don't use antibiotic, in some way the antimicrobial resistance disappear in some way. At least maybe not the resistance mediated from mobile DNA, but at least the genomic resistance can be strongly reduced. So it's a matter of discussion in the livestock production to reduce the use of antibiotics. Europe is quite in a good position compared to other production systems around the world. And but there are several challenges, because now we cannot use zinc oxide, for example, as you know. So it's not easy to maintain a safe production. So to maintain the food safety, when you want to reduce a lot all the factors that support this claim, that is something that we expected. Anyway, we work for that, and our expectation is to reduce the use of antibiotic in our production chain as much as possible, or to use in the proper way when they are needed. Thank you.
Thank you very much. So we move to the next speaker. The next speaker is Stefania Perez-Calvo. The title of the talk is Sustainability and Productivity, Two Sides of the Same Coin. Stefania is the principal scientist in animal nutrition and health at the DSM, and she holds a veterinarian degree and PhD in animal nutrition from the University of Zaragoza. She joined DSM in 2013, and her role, she is involved in an innovation project developing new feeds and investigating nutritional strategy for improved gut health and performances in a sustainable manner. Okay, Stefania, the floor is yours. Thank you very much.
Thank you for the nice introduction, and thank you for the invitation to here representing a company that was involved in the project since the beginning. It was not the case, it was some of my colleagues that were taking part of this project. And the name of the company is DSM Firmenich. Probably you have heard from the previous speakers, and probably you were wondering what is this company and what is it doing. So just a little bit of history about the company. Originally, it was a Dutch company working in the minery and then moving to petrochemicals. And in 2003, acquired vitamins from Roche in Switzerland. And then since a little bit later, was starting an alliance with Novozymes. It was already mentioned this morning. So all our feed enzymes catalog is coming from Novozymes. And recently acquired Bioeconomy, all the mycotoxin management. And last year was merged with the Firmenich, that's why the logo also changed. And now we have 4 different groups or categories of units in the company, and I am representing the animal nutrition and health, what is the last one. So saying that, and putting this a little bit in context, as probably you know, and according to the authorities it is to have a healthy diet, we have to balance in between vegetable and animal source. And the FAO is considering that for humans, adults, we need to take around 0.8 grams per kilo body weight per day. The origin of the protein comes from animal, but also from vegetable. I am going to convince for any of the choice. But it's well known that animal protein provide macronutrients like protein and amino acid and fatty acid, but also micronutrients like vitamin A, vitamin B12, was already mentioned this morning, and also some minerals like zinc. So, but what happened? That if the population is growing up, we are going to achieve in 2050 around 10 billion people, and if we want to still eating meat, we will need to increase by around 60% all the needs in animal protein production. And we know that has a cost in the environment and has a very impactful environmental footprint in terms of greenhouse emission, already mentioned this morning, but also antimicrobial resistance, one of the big topics that was covered in this project, and also land use, marine resource, and so on. We also heard this morning, so thank you for the previous speakers that opened me the floor for this, that in the business that the DSM-Firmenich is working is in the feed industry. The feed industry is having an impact in terms of economy. So for the animal producer, is in between 50 and 60% of the cost of an animal. And in terms of impact in the environment, is in between 50 and 80%. We already saw in the life cycle assessment that was done this morning that was the most impactful there. So we have something to do, and in the company or in the industry of the feed production, we are aware of this one. So we are trying to have putting some actions on that. This is very well known by all the consumers, for all of us, and that is having, as in the panel that I loved to hear before, is the impact that is having in all the value chain. Because of consumers are being more conscious and conscious in sustainability and all the problems that we are having, this is putting some pressure in some brands. And this is putting pressure in the economy part, what are the investors. And the investors are not investing money in companies that are not doing anything against this big issue. And, of course, regulators are listening to the public opinion, and they are also putting some restrictions. Already Paolo was mentioned about the use of antibiotics, growth promoters. Hopefully, luckily, in Europe was already forbidden for animals in 26. In China, recently was also starting to be forbidden. In other countries, come. So there are already a conscience, and regulators are already putting something there. So what we can do? So one of the things that we can do is what we are doing here in this kind of consortium is to talk about in between them, and the animal protein industry has to be shaped and has to be open the dialogue in between all the parts. I just wanted to represent here an animal in the middle, of course. Farmer is also the comment that was already mentioned this morning about the main person who are in contact with the animal, the farmer. An animal producer is who has to be listened and has to be present in these kinds of places. I wanted to represent here the academia that is also doing something there, but has to be a dialogue that in between them they have to understand each other, because sometimes it's a little bit far away, and around of them all the industry that are helping that this is moving there, so genetic companies, veterinarians, I put here the feed industry and also the manure management and all the other technologies that are around here. And so this is the dream of how from the industry we can work together and try to have a better way in this direction, and in the level of the company, what we are doing. So just to share with you a little bit what we are doing in DSM-Firmenich. So all of course we want to sell additives, but how the idea behind is that we try to categorize all the additives that we are having, the portfolio connected to some of the portfolio connected to the Sustainable Development Goals. And for this, we have 6 different business drivers. One of them is linked to emissions from livestock. Here we have all our category of additives linked to the feed enzymes. But also we have one additive called Bover that is linked to reduction of the methane emission in ruminants. And then in the area of the alternative, alternative antimicrobial growth promoters, we have our probiotics, prebiotics, but also some additives like muramidase that was already mentioned, and I will just talk a little bit more later. Feed and food loss waste. Here our catalog of mycotoxins, risk management, and other products linked to organic acid. In terms of making efficient use of natural resource, we have our feed enzymes, improving life performance. Here is more the area of the antioxidants, vitamins play a good role, and reducing our resilience on marine resource. We have one additive that is alternative of fish oil for fish production. And just a little example on this area, a link to the presentation that Kelly already saw for the poultry production, but also was included. This is one of the additives that was included here. Is an enzyme produced by Novozymes from Trichoderma resae, what is a fungi, and the target is hydrolyze the peptidoglycans for the bacteria cell wall. And as the title of the presentation was productivity and sustainability are the two parts of the same coin, one of the things is that this additive, what we need to show is that we have an effect in performance of the animals, and this is measured in body weight of the animals, but also feed conversion ratio. This, for people who are not in the animal production business, this is how much feed we need to provide to an animal to generate 1 kilo of meat. So if we reduce the number of feed, the amount of feed that we need to provide to an animal, this is something positive. So in this case, we can see a positive effect, but also okay, this is nice, but what else is doing this kind of molecule? And this is the most amazing part of my job as scientist working for a company in collaboration with academia, is trying to understand and to learn what are other potential effects that those kind of additives are having. So, for example, with muramidase, we have demonstrated and observed that there is a reduction in diarrhea and piglets, there is a reduction in gut inflammation, there is also an improvement in nutrition digestibility and absorption, and also modulates the microbial communities and microbial metabolites. And knowing those potential effect of the additive is when we classify in the different drivers that I already mentioned previously. So antimicrobial resistant or making efficient use of the production. And another example is the benzoic acid that already was also mentioned by Paolo. Benzoic acid is an organic acid. So what we are doing is reducing the pH of the gastrointestinal tract, and is very well used in piglets who are not able to reduce the pH there. So we can observe a positive effect in the body weight of the animals. In the body weight of the animals, but also in the feed conversion. And another potential effect of the benzoic acid is that has been demonstrated inhibition of the pathogens. As microbiologists in this room, when you reduce the pH, you modulate automatically the conditions for the microbiota. Then improve oxidative stress, inflammation, gut morphology, endogenous enzymes, nutrient, and the last one that is also interesting is reducing ammonia emission, because the property of the benzoic acid is metabolized in the liver and then is excreted in form of hippuric acid and reduce the pH in the urine, what makes that the ammonia is not volatilized. So we can reduce, and we have been demonstrated this effect from this part. And my last part is just sharing with you that we are not only working in products per se. We have recently also worked because in precision service that we call different tools, trying to make the invisible visible, because if we say that an additive is doing something, the producer or the person who is going to buy will like to evaluate and to know how much is the impact. So for this, one tool that is very useful is, as we have heard this morning, is the life cycle assessment. So DSM has developed in collaboration with Blanc, what is a Dutch company, Sustel, were taken into account, as was very well explained this morning, all the raw materials, the origin of the raw materials, the amount, but also take into account the feed additives that are included, the environmental impact that they have, and the effect that has in the animals. So if the animals are eating less, so it's less input, we know that that's why productivity is always an important part for the environmental footprint. Just providing you one example, we can say that we know that conventional pork production have an environmental footprint of 3.52 kilos of CO2 equivalent per kilo live weight. If we reformulate the diet and we reduce the level of crude protein, we can, because crude protein coming from soybean meal, which is coming from Brazil, and this is traveling a lot, and the environmental footprint is very big, we have a reduction of 3%. If on top of that we use include low footprint byproducts produced locally with of course, we include the feed enzymes because sometimes byproduct has very level of fiber that is not very useful to be digested. We need to help to this part. And if we add also the impact of having a higher productivity, so the animals need to eat less to gain the same part, we can improve 50% of we can improve the environmental footprint, what then in economical terms can be represented in a reduction in 4.60 euros per pig, what is quite interesting number. And the last tool that is still under development, and I think has a very nice link with this project and what you also are doing, we try to predict, or we can to use the power of the biomarkers, evaluate it at the level of the blood, saliva, feces, but also from the microbiota, with the idea of detect some imbalance linked to the nutrition, but also to try, the dream will be to predict that we are going to have an issue if we have a specific sequencing. And so the idea at the moment is to work also in this direction in collaboration with different universities, not is linked to circles because this is quite recent. And so yeah, this is the other tool that we are working on. And with this, just some key message about the animal production business, that animal source foods contribute to healthy diets, as I already mentioned, and have also an important environmental footprint. Because of feed is the most significant cost in animal production, have an impact in sustainability. So if we change the animal nutrition, we can improve performance, which can have an positive effect in the health of the animals, efficiency, and last but not least, reduce the environmental footprint, that everything can be measured in terms of life cycle assessment. And thank you for your attention.
Do you have any questions?
I loved your presentation. So much biology which really works and is being already implemented, and products and making progress. It's very optimistic. I have one question, and that's with the bovine. We have, and I won't take the long time. I'll just say it has been a huge part in Denmark because there was a whole national decision on you don't need to cut down livestock as single because you take bovine and reduce 30% of your methane in one go. And then suddenly something happened, and maybe you can shed some light that it is there may be no full understanding of what does it do? Is there a risk for laughter gas to happen after when it's metabolized in the soil? And the other part, what about the animal welfare? Because this is a kind of 3 times nitrogen, which it's never eaten before. So I just wonder, I don't know if we have time, but I just want to say it's a big discussion, and I think we should be aware of it.
Okay. Honestly, I work more in the monogastric part, so I'm safe. And regarding with the true and the experiments that they have done is that one of the questions was, okay, if we apply this one, then the effect is reversed, and the answer is yes. So we can apply this additive, the reduction is reducing at 30%, and then if we remove it after a certain of time, go back to producing this. So in terms of safety, I think that we are okay. Then the second question, the impact in the soil and so on, I am not aware of this part, but yeah, probably can just check with some of my colleagues on this direction.
If you can provide the data, it would be really great.
I will do. Thank you.
question? So we can move to the next session and whatever. Here's the next session.
Thank you.
Okay, so we continue?
Yes.
Okay, good. Hello. Now we are moving from the land to the sea. So this session is going to be about the marine and fish microbiome for sustainable aquaculture. So my name is Shini Pimlapas. I'm from Technical University of Denmark. I'm going to be moderating this session. So for the first speaker, Marco Basili, he's a PhD student from Italian National Research Council. His PhD focused mainly on exploring the fish microbiome in Mediterranean species in order to explore the importance of the microbiome and its function to the marine organism and to the environment. So, and he's going to be talking about exploring wild fish and marine microbiome for sustainable aquaculture. So, where is Marco? Yeah. Yes, please welcome Marco. Yes.
So, good evening. Thanks for the presentation. Thanks for the introduction. And this is my presentation of, the focus is going to be the exploring wild fish and marine microbiome to improve the aquaculture activities. So I guess here, yeah. So first of all, some statement. We are moving to the fish environment. So some numbers of the fish production in nowadays. On our planet, we are going to reach 10 billion of people by 2100, sorry, maybe before, because probably these data are not updated. So what is important thing that 70% of the global supply nowadays is provided by the ocean, and that's what we call food from the sea. And 80% of this food from the sea coming from wild fisheries, maybe even less now because aquaculture is improving in the last years, but for sure more than 50% is today coming from wild fisheries. And what is the important thing about this fact is that exploring this resource, we exhaust or kind of exhaust 90% of the global fish stocks that are fully or overexploited nowadays. So this situation makes it clear that wild fish alone cannot meet the growing demand for seafood in a sustainable way. And on the other side, aquaculture has enormous potential to reduce the pressure on wild fisheries. Sorry. So the main question that we asked ourselves as a researcher is: should sustainable fish production shift toward aquaculture from the wild fisheries? And the answer is yes, but with some concern, let's say, doubt, because sustainability challenges must be addressed by aquaculture itself. So it's not possible to change from wild fisheries to the aquaculture without taking account these challenges. And so what is the impact that what is the environmental impact that aquaculture has in the sea? First of all, aquaculture is well known that it's not a not sustainable environmental condition due to the high density of the fish in the open sea cage, because here mainly we are speak about the open sea cage farm. And another issue is the use of antibiotics due to the let's say to the pathogen that we can find and that are spreading pathogen and disease that can spread to these through this fish through the aquaculture. And the last issue is the organic enrichment that could affect the seabed in this area where a lot of these cages are placed. So what is the main question that we need to answer is: why do we need to know more on wild fish microbiome? Because we know a lot about the farmed microbiome. We know a lot about the farmed fish microbiome. There are several studies that explore the microbiome in terms of farmed fish, and just a few of them, studied and analyzed the wild composition of microbes in these fish. From these studies, from the few studies that also observe the wild fish, it is well known that there are different microbiomes, especially in the gut, because it is the most studied tissue from farmed and wild fish. So, do aquaculture practice have an influence on the microbiota because of these differences? Yes, and probably the microbiome change in wild and farmed fish could be an important knowledge, an important baseline for discerning the effect of farming practice through comparing to the wild fish, so comparing to what we can say the natural microbiome of the fish. This is a general state, not only in some species, so there are a lot of studied in different species all over the world. And another fact is that wild fish microbiome can inform about the potential healthy fish microbiome asset. So as I said before, we can assume that the wild microbiome can be used as a natural microbiome in which we can compare the farmed, that is, let's say, under some environmental effect, some environmental, let's say, changes. So can we improve the aquaculture practice? Yes, and exploiting wild fish microbiome for farmed fish modulation action. So we can use the healthy fish microbiome, the so-called healthy fish microbiome found in the wild fish. We can use it for the potential, for potentially improving the aquaculture practice using this modulation, this kind of modulation that I'm going to explain later. And another fact that is important for the wild fish microbiome is the aquaculture can be a common horizontal transmission route of pathogen in both sides, so from the wild fish to the farmed fish, and also from the farmed fish to the wild, because the environment is not a closed environment, so the open sea cage are connected with the environment, so it's not possible to close and to, let's say, control all the environment, all the input that we can have in the farmed fish and also in the farmed microbiome. And we can also use the microbiome shift from the farmed microbiome, from the farm fish, as an early warning bioindicator for the host health, that it could be a key for disease research and food safety, future food safety. So moving to the topic of this presentation, wild fish microbiome can be key to understand and improve aquaculture food chain and its sustainability. And moving to the Circus project, what we did on the Circus project, we work on Atlantic salmon and Mediterranean gilted sea bream in 3 different countries for the salmon, Norway, Ireland, and Scotland, and 3 different countries for the sea bream in Mediterranean, Italy, Greece, and Spain. And on both of them, we analyzed the farmed samples and also wild samples. And on this project, we are going to have a European perspective on wild fish microbiome for salmon and gilted sea bream. This is just a summary of the massive samples that we collected. You can see here the number; they are really huge. And thanks to Circus to help us to analyze, to sample, sequence, and analyze all these samples in order to make a comparison through the country, through the different environment, through also the wild and farmed fish. Then some result, moving from the topic to the result. This is the result from gilted sea bream. it is, as I said before, it is well known that wild and farmed fish microbiome are different. Here we can confirm this analyzing the Italian and Greek samples for gilt and cibrim, because we found also that we have also the difference in Italian samples and Greek samples in the wild samples, in the wild cibrim. And through these wild samples, we identify some core taxa, some core microbiome spread in all the samples. As well, we identify also a specific taxa from the wild samples comparing to the specific taxa that are well known for the samples, for the farmed samples. And in addition, we highlight a potential role for the host and the habit of the environmental farmed samples. And then at the end, we can compare the farmed and the wild as a potential to exploit core taxa for microbiome modulation. Here, a small focus on the Italian wild cibrim microbiome. Here you can see that we have different group of different group of samples. In the first plot, we have the visualization and the plot of the seasonal variation that we got from the wild samples, wild cibrim. So we were able to sample in 3 different time points. 2 of them were in winter, and 1 of them is in summer. Here in the first plot, you can see. Here in this plot, you can see that kind of samples are the summer one, and that kind of samples are the winter one. So we also see a seasonal variation in the wild microbiome, comparing to what we can see from the tissue, from the different tissue. From the cibrim, we collected different tissue from the gut, but also from the skin, gill, and fillet. And as you can see here, all of them are grouped together, and there is no clusterization, except for the gill that grouped together, all of them grouped together. But there is no variation in tissue microbiome comparing what we found in the seasonal. Here you can see. And then moving to the salmon fishes, to the result of the salmon, we still have wild and farmed fish microbiome differences. So we can see here also for the sample that we can see that there is some differentiation in the microbiome composition. And we can also see that the life condition and the country influenced the salmon microbiome. Here you can see in the color there is some clustering, there is some cluster, and especially in Norway, there is the posterior intestinal tissue from the gut that are more affected by the surrounding aquatic microbiota than the other tissue. Here you can see that are more spreaded compared to the other kind of samples. And as I said before, there is similarity between Scotland and Ireland, and not differences to the country. And then moving again to the salmon, some other result. Wild salmon exhibit higher diversity and richness than farmed sample, varying by country and sample type. Here you can see that in green we have the wild, and in blue we have the farmed samples. In all of the 3 countries, the wild samples show higher diversity compared to the farmed one, both in the richness and in the channel. Then again for the salmon, our colleagues focus on the main phylum that are abundant, the most abundant phylum in the farmed and in the wild microbiome, and there are some differences in terms of abundance. Again, the green is wild and the blue is the farmed. There are some differences in Firmicutes and Proteobacteria that can explain, but that can be the reason why the microbiome of farmed and wild are so different. So they show differential abundance of the major phyla, and also distinct differences in microbial community composition between wild and farmed salmon. Another analysis that we perform in the Circle project is also exploring new wild fish species microbiome. Here is an example. We explored the Solea solea, so the common sole microbiome that we were able to collect in Italy, and from another sampling activity. And we collect several samples from different area, and we analyze the microbiome of skin and the gut. From this kind of fish, we can say that the common sole is being largely targeted as a promising species for fish farming in the Mediterranean Sea, but the farmed activity is not, the circle, let's say, the cycle of the Solea is not closed yet. So the result of our analysis on microbiome is that skin microbiome is weakly related to the collection area. That is quite normal because Solea solea, the common sole, is a demersal species that live directly connected with the seabed. So it's normal that the microbiome of the skin, so the outer microbiome, is connected and similar to the sedimental microbiome. While we found out that the gut microbiome is related to the age, because on our sample we collect several samples, and all of them are spread through the age and through the maturity, through the length, and so on. So this is something that we didn't know before, that the gut microbiome is related to the fish. And the conclusion, the effect is that we can use the specific taxa to exploit the fish age in the common sole. Then moving to the environment, that is the other topic of this presentation. We need to understand the role of the fish farm environment, because can also affect the farmed fish microbiome. So it's important also to know about that. Some preliminary result of some preliminary analysis drive us to the analysis of the bioindicator that can be highly effective in understanding the impact of aquaculture and marine environment. So we use Patella cerulean, the limpet, as let's say bioindicator for analyzing the change in terms of in connection to the aquaculture. We analyze Patella for samples close to the aquaculture and samples that are far from them. Some of them present some differences. Of course, some effect about the environmental aquaculture. Then another approach is the exploration of the pathobiome. That is also another topic really important for the farmed activity. And farmed and wild sea bream samples harbor different potential pathogenic community. You can see here from the plot that they show some differences for some taxa, of course, not for all. And in the farmed samples, we saw that Vibrio community found out in different matrix, especially in the environmental one, including emerging pathogen series were found out in the samples. And Vibrio species found in adult gills or farmed species mirror those found in seawater, including the presence of Vibrio parahemolyticus, which is also present in fish and human pathogen, so can also be used as a warning for our health. And overall, farmed fish rearing in coastal water is probably the most exposed to potential pathogenic bacterial species such as Vibrio. And finally, we use some tests, we use some trial to try to improve the aquaculture environmental sustainability, and using some innovative tools in the mesocosm, we were able to find out some improvement in terms of organic matter in the seabed. As I said before, organic enrichment is a common problem in the aquaculture farm. So using some innovative tools, it can be possible to reduce this organic enrichment. So as a conclusion of this presentation, and as a conclusion of our wild microbiome analysis is that wild fish microbiome is key to develop a new knowledge on farmed fish microbiome and microbiome-based application. Microbiome can be studied or exploited to reduce the environmental impact of aquaculture, and also that Circle has expanded the knowledge on several key fish species, commercially relevant in the Europe and also in other countries, and on the links between fish microbiome and environment. I guess I'm finished with that, and I want to thank all the colleagues, all the partners that are involved in this project, and of course they are involved in the result and in my presentation. Thanks. Yeah.
Thank you for that very interesting presentation. Thank you for that nice presentation. I have a question, just really quick. You said that you can infer the age from the microbiome. What I'm wondering is how robust is it to basically getting the fish out of the water and then determining it, etc. I mean, you have different times of the fish being on the boat, etc. How does that affect the estimation?
Yeah. Okay. So yeah, it's not easy to deal with the microbiome for fish because, of course, as you well know, the sample should be managed in a as I can say not contaminant environment, and normally if you work in the farmed activity, it's easier because you can work directly with the worker there. But for the wild fish, it's not too easy because you can have to go directly there personally, or maybe you can go to the market. So we cannot say for all of them because some of them are catching from us, but we cannot say from all of them that they are out of contamination, of course.
We have another question.
Yeah, so just to follow up on that, I know for the Norwegian salmon samples, at least, they were dissected at the location where they were caught. I don't know about the other countries. But I think it's important also, when you show the results for the salmon, to understand that wild is not always really wild. So there is wild and there is farmed wild. So there's a lot of preservation activities ongoing for endangered populations.
Okay.
And some of those practices involve, for instance, hatching in more or less aquacultured systems, and potentially also a period of feeding so that they release juveniles into the rivers, which means that for the first part of their life cycle, the fish is genetically wild, but from a microbiome perspective, they are farmed. And they then gradually adapt to the wild environment as they live there. So there are a lot of these practices that we also need to consider as confounding factors when we analyze these data.
Yeah, of course. Thanks.
We have a question, right? Yeah.
Excellent. It's really interesting and needed. I have a question. To me, the most important environmental impact, which has to be changed, is the impact on too much nutrient is going down underneath the aquaculture, and that is devastating for biodiversity. And I didn't see you capture this part. And I think the microbiome is an option where you can, in the cultured one, that you can actually design the feed for improved metabolism in the fish, so that we have a better better use factor, conversion factor of the animal feed. Should be possible.
Okay, yeah. Thanks for the question and thanks for the clarification. Yes, I'm well aware that also the environment should be taken account for this kind of analysis. But for our point of view, for the wild fish, I mean, especially for the sea bream, which I'm directly involved, we found out that there are some, also if we catch the samples in the same place, we found out that there is a specific community. So the microbial community of gut and gill and skin are similar to each other from samples from the same samples compared to fish by fish. So for the farmed activity, yes, we need to take in account, of course, the environment, and we need to take account also about the problem of the seabed and about the problem of the environmental impact. But for the wild fish, it's difficult to connect the wild fish microbiome to the environmental microbiome. So yes, for the farmed activity, yes, we need to take in account, but for the wild, it's different and complicated. But thanks.
Yes, thank you. Okay, moving on for the next speaker is Kostas Komas. He is a professor of aquatic microbiology. He leads the microbial communities and habitats in aquatic environments laboratory at University of Thessaloniki in Greece, and he works on aquatic animal microbe interactions with special emphasis on wild and farm fish. So, and he's going to talk about ecology-based learning on fish microbiomes. Welcome, Kostas.
Thank you very much. As the day progresses, I realized how fantastic was the name Circles for this project. Whoever thought about it, Marco, because one of the features that a circle has is that every point in a circle can be considered as an end, but also as a new start. And I see a lot of new starts coming out from what I hear today. But I would like to start first from thanking the two other Circles people from our lab. Eleni is here. She did most of the work with all the fish. And our fish physiologist colleague, Elena, who was at that time at UTH, but now she has moved to another university. Let me go again to another start. When Circles was started to be articulated as a project, a nice paper came out entitled 50 Important Research Questions in Microbial Ecology. And when I went through this paper, I realized that several of these questions, a few of these questions, were actually at the core of the Circles proposal at that time. Primary mechanisms about host microbe interactions. What are the contributions of specific bacterial, microbial, fungal groups for the host? How do these microbial communities work? What do they do where they are there? These are actual questions from this paper. I suggest you go through. You will relate to a lot of these questions in this paper. What are the evolutionary mechanisms behind this host microbe interaction? What about the captive animals? Captive animals could be also be farmed animals as well. And if you want to go a bit further, maybe we have not gone that far to Circles, or the Circles project, but I see a lot of background knowledge for a systems biology approach in this project. So we all agree, we all feel that what we have done in Circles is like we confront the metaomes, any type of metaomes that people have worked in Circles, as independent biological entities and not just as numbers or sequences or whatever. And these entities have their own developmental processes, if you like. There are a few major ecological principles behind our work, at least the ones that we followed for the fish microbiome, the at least the Seabream, but also it relates to many of what you have done. First one is the concept of phylosymbiosis. It relates, well, in simple work, it says that closely related species are expected to have closely related microbiomes. There is a big debate about it. In our case, we did not compare so many different species, but we did compare, for example, as Marco said, that we have farmed samples from different genetic lines in Seabream, Greek, Italian, Spanish. Similarly, maybe for the salmon in the north. So we wanted to see if this actually applies to this. It means also that it's less prevalent in microbiomes with higher species richness, and that was the question that we had for the different species. As I told you, there has been a lot of work for the past 10, 12 years on this, and actually the approach is fairly simple. We screen microbiomes of different species and we compare with the numbers. The second concept is process ontology, and this comes from, not from biologists, but it comes from philosophers. Actually, it's a new proposal saying that what we see, we should actually consider as animals or plants, not as things, but as processes. It's not just the animal that grows, it's the animal that grows along with its microbiome. And actually this concept of process ontology was inspired by how microbiomes work. Fish are very appropriate to investigate this because they live in a microbial soup, that is the water. They're in constant interaction with gazillion of microbes from the moment they are born until the end of their lives. We have also invasive species that they carry their own microbiomes. We have captive, we have farmed, we have hybrids, you name it, it's there. So these are schematic, what it means. Just don't think about an animal as an animal. Don't think as a holobiont, a specific animal with a specific microbiome, but think an animal with an ever-changing microbiome. It is very important for us to keep this concept on mind. The third one, which is also very important for the fish microbiomes, is the concept of metamicrobiome. And as you can guess, what a metamicrobiome is a lot of specific microbiomes putting together and see how they interact. So in the case of fish, a simple metamicrobiome could be the microbiome of the water, of the feed, the provided feed, skin, the gills, anything in the gastrointestinal tract, feces, oral microbiome, you name it. I know that many of you recognize other metamicrobiomes for plants or other animals as well. Now, if you put a fish in the marine environment, fish consumes feed, feed has a lot of microbes, water has a lot of microbes, so you see that, laser pointer, you see that the relative abundance of bacteria in the water or in the feed, you name it, it goes through a process in the fish itself. The fish can swim in 3D. Sometimes they swim hundreds, tens of kilometers a day. So they ingest bacteria and they take them somewhere else. Some of the fecal material ends in the sediment. Some are flocculated in the marine environment and they can travel. You name it, all the processes. So there's a lot of mixture, if you like, of microbiomes there. We have used this approach fairly recently in one of Eleni's papers, and we realized it was one of the first papers actually for Seabream, that when Seabream is young, the first stages, they are much more affected by the microbiome of the feed than when they are adults. Okay, so when they are adult, the microbiome is shaped almost solely by the nutritional effects of the feed. But when you feed the young fry with algae or artemia or whatever, then you need to take into account the microbiome of this feed as well. The fourth one is the Anna Karenina principle. Somebody mentioned it this morning. I was very glad for this. And actually, it was inspired by a specific phrase in the book by Leon Tolstoy, saying that all happy families look alike, but everyone is unhappy in its own way. Well, we don't care how we're going to be happy. We don't care how. But when we are not happy, we want to know why we are unhappy. So the same happens with the microbiomes. It's called disbiosis, thing that we've heard also this morning. And it seems that dysbiotic animals seem to have a very different microbiome that we can fairly easily recognize. Marco said something about it. Of course, it has to do with resistance and resilience. I mean, when an unhappiness factor kicks in, maybe the microbiome, the host with the microbiome, the holobiont, will react to this. How much it can react, how much it can confront, this remains to be determined. It's very important to know these factors for the well-being of the farmed fish. Wild populations, as Marco also stressed out earlier, are considered the holy grail for the aquaculture, at least for the sea bream. And the rationale behind it is very simple. If the sea bream is so successful in the wild, and if the microbiome actually helps in this kind of success, why not try to mimic and reconstruct the wild microbiome under the farmed conditions? This dysbiosis is very important for the fish itself. It has been shown that it causes a cascade of negative effects. For example, in one of our recent work, we found it was an opportunistic work that came out after a major infection in one of the companies that we work with. There was a fish disease, Fungal-bacterial, infected all the fish. Most of them died. They tried to save, and they did save, I think 1 or 2 cages after antibiotic supply, but we monitored the microbiome after the antibiotic treatment. Of course, we found what we expected, that the microbiome after the antibiotic was quite different from the microbiome beforehand. And then we followed how the microbiome recapped after maybe month, actually 40 days. And we saw that in the fish that were treated with antibiotic, even after 30 or 40 days, they were heavily parasitized. Although the microbiome more or less started to look okay, actually it was not working okay, and it was heavily parasitized. No parasites were found in the untreated, but survived fish. This brings us to a more practical approach based on microbiomes for the aquaculture field, which is called microbial rescue. What do we do when something is happening and there is a microbiome disruption? There are 4 different choices. You can try to reconstruct the same taxonomic composition before the perturbation. You can actually not worry about the taxonomic composition of the community that you want to heal, and focus only on the functions. And the other 2 are a bit out of reach for the time scale that we have, the time scales that we have in aquaculture. Is the adaptive and evolutionary. I was very happy, Lena, when I heard you saying this the first thing in the morning. But for us in the aquaculture field, it took some time to realize. And this brings us to the other big thing, the core microbiota, the core microbiome. It's a big, big debate. It's my favorite thing to tease several of my colleagues and friends in the field, and usually turns to never-ending discussions. So let's see some of the rather fluid definitions of what is a core microbiome. Usually it's the stable, permanent, and often highly abundant microbial assemblages in any type of animal or plant. Another definition has to do with microbes that have high occupancy. High occupancy means that they are in high abundance in a lot of samples or in all of the samples of what you investigate. Instead of samples could be changing factors, time scale, anything, you name it. Another more ecological term is that the core microbiome is these microorganisms that win in priority effects. Who comes first and is established first against other microbes or other perturbations. Another more practical definition has to do with ones that are more difficult to modulate, to get rid of, or to change their presence in a specific microbiome. What are the core microbiome types? And what we did in circles, we focused to find the most common ones. That was done in the observational phase, as Marco said. We also did the temporal and the succession core microbiota. So we followed the whole developmental cycle of the sea bream and the salmon from egg to adult or commercial size, which has never been done before. We tried to get some of the ecological defined core microbiota. This was done also with observational, but also with the interventional experiments performed in circles. We tried also the functional with the metagenomics approach, as Marco said again, and some of the host-adapted. We tried to see if there are specific microbiota that are related to a specific developmental stage, maybe, or a specific intervention practice that we had. And now the question comes, should we sweat about the core microbiome, the core microbiota, or no? I have no clear answer. There are days where I say yes, and there are days where I say no, depending on what I read and what I think. So the functional core, the major problem that I have managed to spot to date is what we call the frenemies. There are several microbial taxa. I'm talking mostly about bacteria, sorry Lene, we'll think about fungi, that can act either as beneficial or at the same time act as detrimental for the same host. Okay, another term is called the amphibiomes, proposed by Thomas Bosch recently, where it's actually the same strain that in one case it acts like a pathogen, and another, the same strain in another context, it acts as a beneficial microbe. So actually the taxonomic core seems to be out of the table for now. So again, we're focusing again on a functional core microbiome. And this can be summarized in the fidelity of the bacterial, of the core microbiome, whatever you want to call it, to the host itself. So across the spectrum of the animal world, we have identified some animal species that seem to be doing better, living closer with specific microbes for the whole of their life, while others don't. And if we do this to the circle's context, it brings us to the transmissibility and transmission, if you like, of microbes from one generation to the other. So this is the whole range of fidelity between a host and a microbe or microbes, if you like. So on one side, we have the faithful side, and it happens mostly in aphids, where there's actually only one species, one bacterium that is doing all the job, and they keep this for the whole life, and several other insects do it. And on the other side, the more unfaithful one is where we are, the plants that we studied in circles. And regarding our sea bream, it seems to be mostly, on the unfaithful side. Unfaithful means that the selection of the host for the microbes is unlikely to drive adaptive changes. So actually this, in other words, means that each animal at a specific developmental stage, it has the microbiome that it better fits its purposes. So it's very different if you consider this faithfulness concept into an early stage, like say in the first feeding, okay, or in the egg size. Mark also stressed out the importance of comparing the farmed microbiomes with the wild microbiomes. I have put here, I won't read it, but you can have a look, you can take it from the slide. What is the reason? And the trend in dealing with microbiome engineering in aquaculture is what it is called the rewilding of the sea bream. Talking about the sea bream mostly, but it's happening mostly for other species as well. Not all rewilding attempts have been successful in terms of production improvement. There have been some detrimental effects as well. Recently, I was very surprised that in this journal, that they dedicated a whole, the cover, with this subtitle, Raising the Fish in a Fish Way. And most of the articles, they were around this rewilding process through metabolic engineering or microbiome engineering, if you like, mostly through feed-related or nutritional interventions. When circles started, another thing that occupied us quite a lot is that when circles started, many of the omics tools that we have now were not that developed, but now they are. So there might be some criticism if the 16S approach that we have been using a lot, for at least for the sea bream, is actually meaningful nowadays in the era of super omics. Actually, yes, because we have a lot of points to address this. It is very fast, easy, and cheap, and very insightful for what it offers. It is better testable. We can compare with older sets. We can compare sets between us with very little discrepancies. And the data that they produce, they can be tested based on macroecological theory, fairly easy and quite straightforward. There have been several papers that prove that many macroecological principles can be applied to this kind of data. Of course, it's a first thing that we can do to work on the core microbiome. Whoever believes the core microbiome exists. It's also very nice to depict isolation targets, how to move in a more biological approach, if you like, or it can target what is the best next omics tool that we need to know or sample. So it is a very good thing to do. It's a very rational first step, if you like, in the omics approach. And also it makes good use of what is known today. A big issue, especially for meta-omics, is the host DNA depletion issue. We get a lot of DNA from the host, and for us it is a problem because it masks the bacterial, fungal, viral DNA. We hope that we will do better in the future. So yes, we do have a taxonomic approach for the sea bream, but we are going to complement soon with the functional diversity. So it's not competitive. We're going to do the same job, the same insight, but from a different perspective. But yes, the target is exactly the same. And with this, I would like to thank you, and thank you all, Circles people, for the fantastic last 6 years. I hope to see you in the future again. Thank you. I can hear you.
This is extremely inspiring. I love it.
Thank
And I could come into many different things, but I would like to just suggest one approach.
Yes.
I think we need to look at what gives the fitness for the host pathogen, seen in one.
Okay.
Because that's what drives evolution.
Right. Mm-hmm.
And that also drives when it restores, when you rewild and so on, what is evolutionary pressure, select for the best fitness. And I have an idea for what should be done. And that is to focus, not answering everything, but I think we should focus on the secretome composition, treating the microbiome as one organism. Don't care about the names of all of the organisms. Mm-hmm. Just look at the metagenome of the entire microbiome. And there we have the conserved unique peptide with COP method, where we can have the enzyme function easily for a whole microbiome. Mm-hmm. It's automated. You can find it online. And then if we use AlphaFold, I know this is going to be complicated, but as at least a hint of the metabolites.
Okay.
Because this is what impacts the connection between the microbiome organism and the host is what comes out of the cells, and that is the secretome.
There is some work done with the cazymes so far, and the context that you are for fish, for seabream.
We have done too. Yeah.
Is it the secretome? I mean, no, I'm not questioning the nature, but what about microbial associated molecular pattern? So by the fact that you have a microbe sitting there, it's not secreting anything, just a piece of the cell wall, which often is linked to the taxonomy.
I think it's related, the crosstalk, also between the microbes, but also sharing public goods. The concept of public goods, it's also very, very important for the host-microbe interaction as well. And there has been some indications that the use of public goods, I think somebody showed it, I don't remember who, that, was it you? I think it was you, that one metabolite or anything that is excreted from one microbe is used by another and is transformed to another one, that it can affect either some host function or some other microbial function. So it is
that is all captured with what I suppose. But what you are suggesting is exactly what also should be done. So what I'm suggesting is just one approach which gives us a new perspective, but it's not the entire truth, of course. Yeah. But I think we need it, and then we have it's more clear to take out what you are, then we can distinguish it from what I'm suggesting. And that would give us this evolution and evolutionary pressure of fitness has not really been taken up for the microbiome.
You're right. I think I agree with you, Lena. Good.
Let us start a new project.
You see? Thank you. Thank you.
Okay, so now the last speaker for this session before the panel discussion. Wasim Udin, he's a senior scientist from Norwegian Veterinary Institute in Norway. His work focuses on improving human, animal, and environment health by integrating high-throughput sequencing and advanced bioinformatics with the aim to understanding of host pathogen microbiome interactions. And his talk is going to be about insight into the dynamics of Atlantic salmon gut microbiomes. Welcome, Wasim.
So thank you, Shiny, for kind introduction. Okay. So I know it's a kind of a late evening, but I hope you bear with me. So I'm going to extend the salmon work, which was introduced briefly before by Marco. Atlantic salmon is a very interesting species because it transitions between the marine phase and the freshwater phase. And to achieve this, it has to do drastic adaptations, physiological adaptations. This is where its normal territory is. So it's also one of the species which consumes a lot for food, and that's why its production is continuously increasing over the years, and Norway has a major player in that. And you're all listening about the microbiome since morning, so I should not introduce, but just to set the context of my talk, microbiome evolved with us with several millions of years, and it served important functions in the body, including weight gain, etc. And we wanted to see how host microbiome interaction in the salmon works, considering that it has these interesting phases, life stages. It does go to ecological changes, and all these aspects. Specifically, we want to ask how the gut microbiome of wild and farm Atlantic salmon across countries. So we collected the salmon from Norway, Scotland, and Ireland. We wanted to see how the factors such as life stages, diet, and environment shape the gut microbiome of the salmon. And further on, if we have this knowledge, can we use this knowledge to improve aquaculture practices? So with this, I'm just jumping to the results. So first, the wild and farm comparison. What we saw here, by applying the generalized linear mix models, what we found that wild salmon microbiome show high diversity compared to farm salmon microbiome, but it also differs according to tissue, like we collected digestive and intestinal tissue. So it differs according to tissue, but also according to country. So overall, the three-way interaction is significant. So when you are talking about wild and farm, you have to also consider different factors to have their mixed influence. And here, the 5 major phyla, which is like 0.1%, more than 0.1% abundance, we applied the same models, and what we saw, at least these 3 phyla here, which significantly differs and are highly significant here. So to explain why we are getting these differences. Similarly, beta diversity, which shows the, how 2 samples are far apart, how similar they are. It also shows that wild and farmed are quite different, but also you can see that digester samples are clustered together and in comparison to intestinal tissue samples. So this is the just quick, and we wanted to further explore how at the species level, like ASV level, what's the differential abundance, like which ASVs show differential abundance for each country, and we had the different figures for each, like it's in hundreds. But what we were interested in to look at the common pattern across countries. So what you see here, there are these genus which are common across country, and they are carrying these ASVs. So mainly you see that these 3 Mycoplasma, Alivibrio, and Photobacterium, they are continuously increasing in the farmed salmon, and 2 bacteria, Brevinema rickettsia, are more in the wild fishes. So that's interesting pattern. Talking about the next life stages, salmon life stages, how the microbiome changes. These are the life stages salmon microbiome has, and I'm just showing, so there were discrepancy in sample collection in some of the stages, so I just compared these stages, and that's why I wrote early, mid, and late. And here, just showing the results. So what we saw here, that in all 3 countries, the diversity decreases with the age, and that's very interesting. You do not see this pattern in mammals. Usually the diversity increases with the age, but here it's decreasing. also for the beta diversity, it shows very much circle-like pattern. So it started from the T0, T1 stages from here. It goes up T3, T4 stages and comes back to T8, T9 stages here. So it's completed the circle as the fish itself is completing. And to dig further, what are the major phyla which are showing the changes? So as you can see here, these 5 major phyla, Firmicutes is specifically increasing and Proteobacteria decreasing over the lifetime, and that's interesting. And we further interested in actually at the species level or ASVs level, what's going on. So I ran some deep analysis to run the linear models for ASVs, and what we found that there are different, so these are the significant ASVs or bacterial taxa which were increasing, decreasing, or mid-peak rising following the life stages of the salmon. For example, here you see Mycoplasma is quite drastically increasing here, and they belongs to these 5 phyla, but you also see that there is a major decrease in the ASVs over the time period, and that's why you see overall decrease in the salmon microbiome over in different life stages. So going to the third questions, how the microbiome is shaped by the environmental factors or diet. So here you see the diversity of the different sample types. And you can quickly see that freshwater, seawater are more diverse in all the cases, for example, in comparison to diet or in comparison to salmon microbiome, salmon gut microbiome. But what is more interesting is to know how these digestile intestinal tissue, what's the source of this microbiome? And I ran the source tracker analysis to understand that. And here what you see there, it differs according to country. For example, in the Norway, if you talk about digestive samples, these bacteria are mostly coming from diet. And you can also see that in some cases, it's like in the first 2 stages, which are freshwater stages, it's coming from the freshwater, but also from the seawater in the later stages. So it differs according to country, how the microbiome is shaped by different factors. And again, it's just showing the beta diversity pattern of these differences. So here are the environmental samples, and the diet samples somehow immersed in Norway, case of Norway, and this is digestive intestinal samples. So by having this knowledge, how we can use this knowledge? So this was previously done by our partners. They have added some probiotic bath in the fish ponds, in the farm fish, and showed that enhanced growth and decreased mortality, which they later confirmed that it's mostly because of fish are having less sea lice infestation. Sea lice is the major problem in the salmon industry, and this specific probiotic Vibrio species can decrease the sea lice infestation. So, but they wanted to understand what's going on at the microbiome level, how the interaction is working, host probiotic interaction is working, and in general, what's the picture. So we repeated the same experiment with the control as well as probiotic treated samples, and here I'm just showing the results, and from the different tissues, I have included skin and fillet as well because it works. It's the probiotics added in the water, and it mainly works on the skin as the sea lice attached on the skin. So here you see that in the red color, it's more spreaded kind of compared to normal control. But more, what we saw that these bacteria which are getting increased in the treated cases, you see that we received this Vibrio, which is quite a lot increasing, but also in combination with other bacteria. But you also notice that there is one Vibrio ASV which is getting decreased, and this forced us to look the complete picture of Vibrio. And what we found okay, this is not on the right spot, but what we found that we recovered all these 3 strains, but in general there are 29 different strains only of L. vibrio in the salmon different places. So yeah, there is a lot of work has to be done to understand how these L. vibrio species, for example, some are just popping up in the C1 phases, and how they are and how the overall system is working. So yeah, of course, it raises more questions and further explorations. So in summary, what we found that microbial diversity and community composition differs in wild and farm salmon, but also vary by country. Life stages, diet, and environment all together shape the gut microbiome of farm Atlantic salmon. And specific bacterial taxa such as L. vibrio can be leveraged to improve the salmon aquaculture. Yeah, with this, I would like to thank the team of the Veterinary Institute and all our collaborators, and I'll thank you all for your listening and collaborating with us. Thank you.
Okay, one question. Yeah. Okay, so I can ask one.
Yeah.
So I'm just curious, the result you show for microbiome data is from only thickness or also with the metagenomics sequencing data?
No, it's a 16S.
Okay, then the next question is, if you do metagenomics that you already have the data, do you think you will see the same pattern?
Yeah, we are curious what we'll see here. First of all, it's we want to see the function which are missing from this study. So we hope that we will find more information. And also, as you see in the last slide, there are interesting strain of L. vibrio and there is mycoplasma, etc. So we want to see the full genome of these, like how they are evolving with the host. Yeah, so that's also important information. So we will see that I think some part of will be repeated, but we'll get in addition more information with the metagenomics data.
You haven't tried to binning yet?
Not yet.
So from the sea bream community people, they try to bin, and they have absolutely nothing that can be bin from the fish sample. From the environment, yes.
Okay.
So the fish sample has very little biomass and very hard to do binning. So I want to see if you can. If you can, it's very good.
Yeah, sure. So I joined very late in this project. It's just my first year, so that's why I was just started. It's very fresh results.
Yes. Yeah. I think we need to move on to the panel discussion. Yes, please.
So it's Anton is sitting there, Rosa in the middle, and Ricardo in the blue chair. I have my cheat sheet here because I'm sitting for Grazia Quero, who was supposed to coordinate this discussion panel, but unfortunately she could not make it, so I will do this job. So before that, small introduction for our panelists. So starting from Ricardo Vicenti he's been working for the Italian feed and food company Veronesi since 1990. As swine nutritionist within the R&D feed team. Ricardo is working a lot with, he collaborates a lot with research and institutes and universities for this, for what he does in Veronesi. Rosa Domenech Matta, okay, I got it correct. She's the leader of the Industrial Biotechnology Department at ITENE in Valencia since 2022. She has a PhD in biophysical studies at Miguel Hernández University in Spain. She's now working a lot on the Tribiome project, which is on the study and modulation of rhizosphere microbiome in wheat crops. And last but not least, Anton Alberti on the far left for me, right for you. He's an associate professor at the Global Institute at the University of Copenhagen, where he leads a group of quite a few people, I would say, on the impact of gut microorganisms on ecological and evolutionary and applied aspects on vertebrates. So he likes animals. He's the coordinator of the EU project 3D Omics, while he coordinated the other big EU project, the HoloFood, and also the Earth Hologenome Initiative. So now, moving here, but I will keep my cheat sheet if you like, because I don't remember my questions. So I have some questions to each one of you and some more general questions. And when somebody from the public wishes to chip in, just wave your hand. So I will start with Ricardo, which is closer to me. So the first question to Ricardo would be, what kind of information do you expect to get from animal microbiome, but beyond taxonomy?
Yes. First of all, thank you to the organization to be here. This is the first time, as I said previously, this is not my job. I'm not, I am only a swine practitioner, not microbiologist or researchers.
We're a very inclusive group
in other fields. Anyway, as a end users, which I love to call me, we, as a swine nutritionist, we try to collect all the possibly useful information coming from the field, of course. This is our first impact on a daily job, but our interest is much more than in the past to collect data coming from the science and the project. And this is why we decided to very interestingly approach this kind of project. Which kind of information? This morning and also this afternoon, we have heard about the interesting information coming from the research made by the authors and participants. And as a swine nutritionist, of course, I'm much more interested in the results collected by University of Bologna in the swine field. I think that we have seen several results which show the interaction between the microbiome and the gut. And one of the things that I love more is the concept feed the gut. Since the recent past, this is the phrase, this is the sentence that I love very much, because really, and Paolo showed before and discussed before, how different can be the lifetime of the different species. Fortunately or unfortunately, I am involved in the swine business, and swine, as Paolo showed before, is represented by swine lifetime is represented by different phases, and in each phases, the real target for us, just to try to answer to your question, is to make it smoother. The impact from the one phase to another. So the transition between the sow farm and along the nursery period up to the weaning phase, post-weaning phase, and later on growing, finishing, considering that at least, and I can say, I can repeat, fortunately or not, in our pig business in Italy, the lifetime of pigs is longer and longer than in the other countries, because just this morning we have seen the results of pigs reared up to 170 kilos. So every phase and every period is represented by different situation in terms of microbiome, in terms of gut health. And this is why you ask me how should we get from this project is to really to find out the most suitable solution just to face and to overcome this troubled transition period between the different phases.
Thank you. Thank you, Ricardo. Well, now moving to Rosa. Rosa, you're coming from the plant microbiome world, okay,
Yeah.
and you work with wheat. And then wheat is used for as feed, you know, for animals or as humans. So how do you interconnect this tripartite, I would say, association, microbes, plants, and then higher organisms?
Okay. So first of all, thanks for the nice presentation, and thanks to all the organization for inviting me to join the final event of Circles. So, to get everybody into the scene, I will explain, or I will give the objective of the Tribiome project. So, the Tribiome project consists of searching and modulating the microbiome of the rhizosphere of wheat crops. And then what we want to we are modulating to see to improve the crop productivity, to get more nutritious food, to reduce the greenhouse gases emission, and what we will see regarding what it took me to this panel. So the project wants to clarify, or aims to clarify as well, the interconnection of ruminants microbiome with the crop-based food components. So to that aim, to see this interconnection in the project, there will be performed different in vitro assays with modulated wheat as feed of animals to see the variability of the ruminants microbiome, and also we will see how it affects to the digestibility, to the fermentation, and also to the methane production, as this has very impact in the food system and also for the management of livestock. So these assays, in vitro assays, will consist on determining to assess the variability of the ruminant microbiome with this improved wheat. And what we will check by metabolomics to see metabolites that are linked to this methane production, and also we will search for microbial strains which enhance the digestibility, reduce the emissions of gases, and also improve the nutritious content of the grain. So, why is this important? Is this crucial to understand this interconnection? Because it can reduce feed costs, it can mitigate the greenhouse gases, and we can obtain more nutritious food. So, and then contributing to the food, to the secure food system, and also to assess the malnutrition in the developing countries.
Which ruminants are you going to work with, just out of curiosity?
We are going to extract the rumen of cows.
Okay, good.
Yeah, okay. And also, if I'm not wrong, also with sheep.
Okay, good.
Yeah.
Thank you. Well, Anton, well, it seems that we've heard a lot about animal microbiomes today, the animals that we included for the investigation in Circles, you know. But it seems that a new aspect in animal microbiome science is the micro-scale differences and the localization of specific microbes in the gut system or anywhere else. So do you think that a spatial arrangement should be, included in future studies for animal health? And if yes, how?
Short answer, yes.
Okay.
Before the long answer, thanks a lot for the invitation, and it's a pleasure to be here. And the second thing I will say before the long answer is that I am an imposter here in this field. I am an ecologist, evolutionary ecologist, but an animal evolutionary ecologist who transitioned into microbiome research about 10 years ago. But still keep the frustrations of being unable to implement a lot of the theory, a lot of the modeling that I was able to do back in my old times at the macro scale with these microorganisms. So I found very, very enjoying to reconstruct genomes and to try and find different functionalities and model metabolite flows, etc. This is really fascinating for understanding how microbes interact with each other and how these interactions could be affecting the animal themselves. But for many years, I was finding that we were lacking something, and that something was the space, because we know how important space is for ecology in general, for interactions between different organisms, but we were treating microbiomes as one unit of millions of organisms that are interacting with each other in a scale that if we give the size of a human to a microbe, the space the human microbiome spans in the human gut, for instance, would be as large as the entire continent of Europe. Subcontinent of Europe. So we are basically trying to understand how a system works, melting and blending together elements that at the macro scale would take the area of an entire continent, trying to answer questions that that require another level of resolution from my point of view. So this is why I embarked in this let's say dream of trying to reconstruct the spatial arrangement of microbes within the animal intestine. And this is what is the European project I am coordinating is all about. And I also think that it's the future. It's the future because we are revealing interactions that are not possible to be detected at the macro scale. I think in the future, as the technology develops, we will be able to develop not just, or generate not just shotgun metagenomic data, but also gene expression data, metabolomic data that has this spatial component. And we will realize that probably we will laugh at ourselves of how we were interpreting the results that we were getting from the fecal samples when we see how these interactions are changing, depending on what scale we are analyzing them and which intestinal section itself we were detecting those interactions.
It sounds that it involves a lot of methods development, okay, and maybe techniques development as well. Could you elaborate a bit on this?
Well, we spent 2 years and a half in the European project just pursuing a dream without getting any results, basically. So we were blind, and it was just full of faith, and it was a lot of people participating, and I was the one that were trying to, come on, we need to continue, but I must say that the day when I saw the results, and those results were making sense, and those results were revealing things that we never saw before, that was when I took a lot of weight out from myself, because it was a huge relief. And but still, there is a lot to do, because that requires not just generating the data, it's also generating the samples themselves, because we get intestinal samples, but then you need to cut them with different type of cryotomes, etc. Then you need to laser microdissect them to get really tiny samples. We are talking about samples that have between 50 and 2,000 bacterial cells in them. So we are talking about 500 square microns with that realm. And that requires a lot of development because the existing tools, the existing kits, existing extraction methods, etc., do not work at that scale. Because you cannot really measure anything until you get the sequencing data. So I think there's a lot that needs to be done, but I am very, very motivated to continue working on this, and perhaps in a few years to normalize all this and these techniques to be much more applicable into a broader range, also including in plant microbiomes, etc., etc.
We'll keep an eye, many of Circles people, we'll keep an eye on the project, definitely. So now I have a couple, we don't have a lot of time, we have like 13, 14 minutes. So I will just pose a few general questions, and whoever feels, that they have an answer, please do. So my idea is, what do you think about the potential, any potential, if there's any potential, on microbiome engineering, regardless of the method used, from your own perspectives, from your own field? I'm talking about from simple, nutritional, small experimental interventions, like changing one ingredient in diet, for example, to organoids, to gut simulators, you name it. Should we put more effort towards this, or should we put more effort, like Anton's project, for example, on new approaches and see what is happening where in the gut or so? From your own perspective, this is an open question, of course, to all Circles people. But let's hear our guest first. You can go, Ricardo, then let's follow the same sequence.
It's a very challenging question. Yeah. No, theoretically talking, I think that it is possible to implement this kind of technology. Honestly, I'm not so much involved, so my question is very partial.
Yeah.
But theoretically, I think yes.
Okay. Yes. Rosa, Anton?
Yeah. Go ahead.
You go, Anton.
No, I think I was a bit surprised in the morning when I heard that. So the science is there, that we kind of know enough now to go ahead and do policy and implement this in the field. I have the opposite feeling. So I feel that we know nothing. That's a very honest answer. So anytime that I am analyzing a new type of sample and a new host, it's a new world. So basically, it's a new world, and I also have the feeling that whenever we try to do in vitro experiments and we put together more than 3-4 microbes, we have no idea how they are going to react to that. So sometimes I feel that's how do we want to predict how a complex ecosystem will going to respond to some kind of treatments if we are unable to predict how 3 strains put together and with 2-3 different carbon-nitrogen sources will react to that, because there are so many spontaneous interactions that happen that we were expecting, and so on and so forth. There are so many genes that we are unable to know yet what they are doing. So you mentioned the Earth Hologenome Initiative. In that project, we have analyzed microbiomes of over 250 vertebrate species, and we have just annotations for half of the genes that we predicted from there. So we have no idea what's going on with the rest. And those that are annotated based on model species that are easy to culture and we can experiment with, etc. But there are so many bacteria that are uncultured so far, so many bacteria that are extremely weird, some that are so small, some that are so big.
Not to mention fungi, eh?
Well, it's bacteria because it's easy to work with.
Yeah. True.
But then when we expand to viruses, also plasmids within the bacteria, and we go from the chromosomal DNA to mobile elements and so on.
It's getting there.
That's why I think we are. My feeling is that we are still quite far away to be able to properly implement this kind of modulation strategies. Of course, some will work in certain environments, and we heard this previously that something works in one farm, but that it doesn't work in the other one, because priority effects also have a huge impact, and what comes first then determines what's going to happen later on. So, of course, there might be some specific conditions in which these modulations can really exert a huge impact and have an effect size that is big enough to have an impact. But I'm afraid that still we are a little bit far away to implement this at a global scale.
I am really tempted now to make the question that Davide did and ask who in this room are in favor of microbiome engineering and who not, but I will not answer this question. You can think about it yourself. Yes, Len. Can you, the microphone, please?
The reason why I'm suggesting secretome is because then we have a tiny part of all what's in the metagenome. That's one part. But to your question, that we are facing most likely the emerging of genome-edited plants. Mm-hmm. That is a discontinuity where the microbiome research community can try to see if that is, for instance, in genome edited, which have improved nutrient efficiency, so we don't pollute the water surrounding so much, and need less fertilizer. There we could try to design an improved microbiome for the rhizosphere. Mm-hmm. Of course, including both bacteria and fungi in the right...
And viruses.
And viruses and starships, and you name it. But the point, no, proteus and, yeah, maybe. But my point is, that is a discontinuity because we have an opportunity that there is a new type of a plant host where we could even make it even better by improving the nutrient efficiency by penicillium supplying the phosphorus, and etc. Can you see? And I think we should really consider if that is a time where we should move in, because then we are not so far away from the foreign genes and foreign DNA and no GMOs, etc. But still a new host microbiome interaction possibility.
Seems like another level at the host microbe relationships that, maybe it's the next step. Yes, you're right. Thank you. I have any other questions on this? Yes, Davide. Or comments?
Yeah, I think if Anton stays for dinner, we're going to have a very long discussion. But I will cut short to the benefit of the round table. Yes, I have this tendency of being fast and furious when it comes to research and exploitation. But there is a reason. So I do agree that probably there are many facets that we have not yet understood. The problem is that the time that we don't have.
It's evolutionary. He's thinking on an evolutionary time. That's why.
I think that there is the need of finding a common ground between the understanding of a biological system, and I concur what you have just said, and the fact that we've got to deliver in terms of, let's say, benefit for society. And we may not have the time to, let's say, acquire all the knowledge that is required. At a certain point, we need to, let's use it maybe a strong term, gamble on what we have learned so far and try to implement it. So what do you think about that?
I think that I haven't found myself a case example that is strong enough that drives me to lead to the conclusion. So I guess that's it. Probably you have other experiments where you saw really big effect size and say, okay, then this is clear. A leads to B. Therefore, if we do this here and there, we will get what we want. Unfortunately, I haven't found that in the animal systems that I work with. So it was poultry, salmon, cattle, swine.
Spinach.
Yeah, so there are spinach. So I might need to switch to spinaches. But yeah, that is more the feeling of being still in the infancy of understanding these ecosystems. And but I do agree in the sense that sometimes we need to react, and we need to react with what we know at the moment. And then probably in 10 years we will laugh about ourselves again. But that's how science works.
Yeah, I mean, this is a different approach. Davide is mostly interested in increasing productivity, but in your case, productivity is not a major factor.
It's more the
growth, yes,
of understanding
but not,
processes rather than and it is. I think it is a question of whether you model for classifying or you model for it's a predictive modeling or it's more a
intervening.
causal modeling to understand how a system works.
Okay.
I probably am biased towards the
Yeah.
the mechanistic approach, and that's
Good.
Yeah, that's a systemic bias.
I have one last question. We have to be very brief in your answer because we're running out of time. But it is related to the future of the people here involved, you as well. About if it is possible and what will be your strategy to persuade any type of stakeholders, funding agencies, industry, research institutes, educators, about moving on in the context of circles. I mean, if you are from the industry, what would you ask from the universities? Would you ask to be educated on bioinformatics? If you are from a research center, how would you persuade the funding agency, to support your future work in the context of circular microbiomes? Same for you. I mean, whoever wants to answer. You cannot answer if you don't want to. Huh? I mean, no answer is an acceptable answer here. Yes.
So for this, to assess this challenge, I would go on. So we need intimate collaboration of all the stakeholders of in my case, for the animal feed chain, no, because I explained of producing feed for animals. So we would need all this engagement and collaboration among all these stakeholders, and to persuade, we could focus on the problems for each stakeholders. For example, for farmers, to mitigate the greenhouse emissions, how to solve that, and for end users, for example, how to provide evidences that the food, that the products that they are consuming are secure and have quality. So with this, and design strategies, and for sure research dedicated to each problem of each stakeholder of the value chain.
For you, Anton, what would be the number one challenge to persuade somebody to?
I think I mentioned what the challenge is. It's complicated. So to me, one of the biggest fears is that it is so complicated that we end up oversimplifying something that usually it is oversimplified by companies and by journalists. And it is very difficult to find that common point in how to express something that it is extremely complex and sometimes it's simply uncertain to people that need certainty. And I feel quite uncomfortable in that position, and it is.
So the challenge for you seems to be a rather deconstruction of, what we want for the future, right?
Yeah, I think it is also a cultural thing. That's how society wants to get knowledge, and there is
an important factor.
No uncertainty. This is the fact, and this is how it should be. And when you are unable to provide that, then it looks like you're not valid in that sense. So to me, that's the main challenge. So how to ensure that we transmit what we know, while also transmitting that there is a lot that we don't know. But that's the natural thing. That's how it should be. And we are in the way to get this. But it's oftentimes challenging, mainly with companies that, of course, need to sell their products. And for journalists that also need to sell their product. So that's...
Well, we have now the opinion of a company. So what would be the challenge for you?
So the challenge coming this from this project and from other projects we have carried out in the past, because this is not the first one. In the recent past, we have carried out similar project on the same, more or less the same road, just to get as precisely as possible information coming from the study of the biomarkers of modulation microbiota across different projects conducted with the DSM in the past 2016, if I remember well, Stefania, or together with the Unibo. So what I mean is that at the end of all, and this is what we found it quite difficult, is to translate in the same word the project and the scope of the project up to the farmers. As a supply chain, as Veronese Group, as AIA Group, what is really difficult is that at the beginning, even in this project, we found the farmer quite skeptical. Because they don't look at a good result or good perspectives in terms of profitability, in terms of redditivity. And this is what probably is one of the, I don't mean the only one, but the main target of this kind of project. So not to separate what is really sustainable, what is really environmentally positive, environmentally good, with the profitability, because at the end of all, at the supply chain, the farmers has to earn, and we are obliged to let them to earn in the right way, of course. But to be very honest, to be frank, one feedback coming from this project is that I can see, and we can see, some guidelines to be forwarded, yes, in terms of sustainability, in terms of health, and in terms of profitability. When the animal is healthy, probably it's really good at well performing. Yeah.
Thank you. All of this I would like to thank, and please applaud our guests.
If you have any question, we need to let Rosa leave, because
Rosa has
she
to catch planes
has,
and trains and everything.
She has trains and everything, sorry, and maybe our applause to...
Thank you, Rosa.
Sorry. There is any question for
yes,
for the
just in
speakers?
case.
Okay. Otherwise, we can move to the break.
Yes.
Okay.
And Davide, jump on him.
Please, just keep 10 minutes, because we will manage to...
All right, everybody. The last people coming in. So it's my pleasure to start session 6 now after everybody has hopefully been re-energized from taking some coffee, sugar, and or nuts. And the session now is about tackling antimicrobial resistance in food systems, from surplus food chains to environmental impact. And our first speaker is Marco Candela. I will not reintroduce him again. So Marco will talk about rhizostome dispersion from surplus food chains to the environment. And yeah, the stage is yours.
Yeah. Thanks so much, Cedric. Yeah, okay. Okay, this is the correct one. So we are going to talk about the dispersion of antibiotic resistance bacteria from food system. So in the frame of the Circle project, we were exploring two models. So the first is the dispersion of antibiotic resistant bacteria from pig farms, and the second is from poultry. So we will see the data has been produced, the results, and so the publication. How we assess for the dispersion of antibiotic resistance bacteria. So we applied a metagenomic assembled genome-based approach. So essentially, we use the shotgun metagenomic to recreate genomes and then look for microbe dispersion across the food system. So this is the model for the pig food chains. So the microbiome has been sampled longitudinally in 2 commercial farm systems. And we got a total of 300 shotgun samples that has been analyzed by shotgun metagenomics, 5 time points across the entire production chain in 2 different farms, including 99 pig stools, 12 booths, air sample, drinking water, wastewater, and soil sample outside the farm. So we try to provide the whole picture of the farm system. So from the shotgun metagenomes, we were reconstructing the metagenomic assembled genomes. Then they replicated in single genomes beans. And this was done for studying the dispersion of the microorganisms crossing the farm biomes, and for assessing the antibiotic resistant genes that they were carrying around. So we have been successful in the reconstruction of around 3,000 high-quality metagenomic species that were replicated in 500 single genomes beans. The 31% has never been detected before, and this is the clustering of the sample according to the compositional structure in terms of single genome beans, and you see a clear clustering depending on the sample sources. And what is important is that we detected around 300 single genome beams, so microorganism that has been detected in the pig gut microbiome that were distributed across the entire farm system. And 135 of these single genome beams were belonging to host-restricted taxa. If you can see, here there is the total core rhizostome from these 309 single genome beams that were shared between the pig gut microbiome and the external environment, and that way of providing resistance over several antibiotics genes, 18, and 14 of these 18 antibiotic resistant genes were of critical importance from human health according to the World Health Organization. So among the 300 single genome beams, there were 11 single genome beams that were pervasive and present in all the farm system, in all the sample we were studying. And these pervasive single genome beings were carrying the entire core resistome. And within the pervasive single genome beings, there is one that is an E. coli, so an enteric microorganism that was multi-resistant, so providing all the resistances against the antibiotic, including the core resistomes, including the 14, which are of relevant importance according to the World Health Organization. So what happened in the farm, in the poultry farm system? The model is exactly the same, but here we go further, since at the end of the production cycle, so at the time point 3, we were including also workers and their respective families. And so we were sampling the workers' coat, skin, stools, and then the cohabitants' skin and stools, and also some surface from the family houses, such as the kitchen surface. So also here we were applying a metagenomic species based approach, reconstructed around 200 metagenomic species, then replicated in around 300 single genome beans, 22% never detected before. Sample cluster according to the ecosystem. And here, with the same approach, you see the 180 poultry gut microbiome single genome beans that were detected across all the ecosystem in the internal and the external farm environment, but in this case also including the workers and the cohabitant metagenomes. And these single genome beans include 70 species that were from known poultry origin and 21 belonging to host-associated non-spore forming genera. So here there is the total resistome from the 180 single genome beans that were sharing between poultry, farm system, and the workers and the family microbiomes. And you see that is huge. It's called resistance for 25 different classes of antibiotic compound, 15 of which are recognized of clinical importance according to the World Health Organization. And it is important that 20% of these genes were detected on mobile elements, so theoretically able to move from microorganisms to microorganisms, so able to dispersion between different microbial taxa. And regarding the core, 24 core antibiotic-resistant genes have been detected, so shared by the 118 single genome beans. And within the 24 core antibiotic-resistant genes, 4 were predicted to be mobile, since included in mobile element and also of clinical importance, so able to perform dispersion interhost with high efficiency. And then focusing on these 4 antibiotic-resistant genes that were present in all the study environment in the farm system, including workers, and also including their family and family surfaces, and that were of clinical importance, and also that were on mobile element. We were looking for their dispersion at the level of population. So we were searching for this core mobile antibiotic-resistant genes in the world population. And what we found, of course, that these genes were highly prevalent in the circle population, of course, because they are the workers and the family members, but then their prevalence was higher in the Italian, that's Sweden, USA, and then essentially was high in all the Western population, whether the prevalence was very low in all the rural population from Tanzania, Brazil, and Peru. So it seems that there is, it suggests a dispersion process still going on from the farm system and going to the human population and to the local population, and in particular, since we are speaking in the Western world, to the Western population. So the take-home message is that human beings, workers, are a route of dispersion of antibiotic resistance bacteria and genes from the farm systems. And this dispersion goes from the workers to the family and maybe at the level of population. And so this requires a more monitoring of the workers' microbiome with respect to the carriage of antibiotic-resistant genes as an action to protect the workers, the family, and the human population for the dispersion of antibiotic resistances. Thank you very much.
Thank you, Marco. We might have 1 minute or two because you were right on time for questions. Antjan?
Thanks, Marco. That was great. It is so scary, but that was great. I have one question about the replication of the genome. So which average nucleotide identity did you use? And can you ascertain that it was the exact same strain that was carried across the different systems? Or
in some cases, yeah. So we applied the same approach that Nicolas Agata were developing for sharing. And I don't remember exactly from which strain we were able, we got enough reads to check for the presence of the same strains, so the correspondence at the strain level between the different microorganisms, different hosts. And so in the paper, there is this detail, and now I don't remember. But in a way, when it was possible, we did it. But you need more reads.
So then if there are no further questions, let's switch to the next speaker. I'm more than happy to welcome Shini Piplapas on stage. Shini is a specialist in genomic epidemiology, bioinformatics, and food safety, as well as antimicrobial resistance. With over 90 publications in these contexts, and her work includes developing machine learning tools for bacterial analysis to significantly advance food safety, as well as bacterial genome analysis. And next to her research activities, she's also teaching at DTU as well as online courses. And yeah, looking forward to your talk, which is entitled Resistome Dynamics in Aquaculture, so another important dimension of circles. The stage is yours. Thank you.
Thank you, Cedric, for the introduction. Yes, hello again, everyone. So I'm gonna back to the sea again, but now I'm not gonna talk about the bacteria. Yes, still something in the bacteria, which is the antimicrobial resistant genes. And when I say the aquaculture here to see the dynamics, I only mean one animal here, which is salmon. So I'm going to show you the dynamics of the AMR in the salmon. And yeah, you have heard a lot of things about the salmon and why we work with it, and it's how important it is. And we so love the salmon in Europe. It's the highest consumption of the salmon in the world. And in circles, besides we looking at the microbiome in the food chain, we also looking at the antimicrobial resistance and dynamics in the food, especially in the salmon. So to do that, of course, we need to have samples. And the sample that we got the sample that included in this result is from 2 countries, Norway and Ireland. And this is number of metagenomic sample. So they are all shotgun metagenomics, and they're from different stages of life. They're also from the fish, and they also from the environment and the food that the fish ate. So, and you might wonder why we talk about metagenomics a lot, or the shotgun metagenomics. If you study only the microbiome composition to know what it is, maybe it's only enough to do sickness, you know what it is. But in order to know what it can do, what is carry, you need to do the shotgun metagenomics because you need to sequence everything in the sample, DNA, in order to get what they carry. And the one of the things that I focus, what they carry is antimicrobial resistant genes. So I just talk a little bit briefly on how I process the metagenomic data until I got into the abundance or diversity of AMR genes. So first of all, once you have the raw reads from the shotgun metagenomics, you need to process the data. You need to map or align to the databases of genes. In this case, is the AMR genes. We have the AMR genes database called Breastfinder, and we also need to map to the bacteria, even though we do the AMR genes. I will tell you why. Because later stage, we need to do a normalization in order to compare between the sample, compare the AMR abundance. In order to do the normalization, that what we do in our group, we need to take this, let's imagine this, each of the columns here is the number of reads that map to each of the AMR genes. So we normalize by the number of reads that map to the genes by the size of the gene, because different AMR genes have different size. And then the gene came from the bacteria. So we also normalize by the reads, total reads that map to the bacteria. So normalize by the size of the gene, normalize by the total reads of the bacteria. And that we come up into this unit of AMR we call FPKM, fragments per kilobase reference per million bacterial fragments. So when you see the term FPKM, for you to know it's a unit of the abundance of the AMR. So the higher FPKM, the higher abundance of AMR. Okay, let's have a look at the result. So this is just the total abundance of AMR, just sum up the total abundance of AMR in different samples. So this is the farm samples. This is the white fish, white salmon, and the red one is from Ireland, and the yellow one is from Norway. And you can see that we only got Ireland. We only got the feed sample, but in Norway we got also the feed and environment sample. From here, you can see that the abundance of AMR in feed, it varies because it varies by state of life, it varies by the type of the sample. But in the lowest abundance of AMR, it go to the environment. So it looked like the environment, the water, the seawater, the normal water that they stay there, it has lower abundance of AMR compared to the fish or to the feed. But in terms of the diversity, it looks different. Even though this is the environment, they are here. Diet, the feed, the freshwater, seawater, they are here. They have lower abundance of AMR, but they have a highest diversity of AMR. Also, the environment here, they also show the higher of the richness of AMR. So environment in the aquaculture for salmon, lower abundance of AMR, but high in diversity and richness. If you look further into the different classes, so now I show you the proportions, abundance, relative abundance in the different classes of AMR instead of sum them up. So I flattened them down into different classes. Different colors here is different classes of AMR. And you can see it from the feed, environment, and it's the fish. This is only Norway because Norway has 3 categories of samples. And if you just look at the fish, this is quite small, but just for you to know, the beginning of the plot, the bar here is the early stage. The eggs, the larvae, the fly, and so on. So from the early eggs and early stage into adult. And you can see that the composition of AMR classes from the early state, the egg and early state of the salmon, it look different. Some classes are the same, but some are not tied. Also the abundance of the classes. So it looked like when they're early life, they have different composition than when they become an adult. But this picture is even clearer if you look at the genes. This is the class. Now you can look at the genes. I cannot show you all the genes. I will show you only the most abundant 15 AMR genes. That is it. So the same order of samples, but the colors here represent the genes. What you see, the green one here, because that are the genes, that the most 15 abundant genes, okay? And you can see clearly that the most abundant of genes, I mean, when they become a baby fish, early state of life, they have the different set of the most abundant AMR genes compared to when they become an adult. And my next question, where are those genes come from? If you look at this when it become an egg, and then when they start to eat, some of the genes that they didn't have before, it look like they come from the feed that feed them. And when they're growing to be adult, some of the genes that they didn't have previously, it looked like it also came from the environment. So in these results, it's indicating that yes, there are some of the AMR genes present in the aquaculture of salmon, but where they're from, this might indicating it could from the feed and the environment surrounding the fish. That environment, I don't mean the use of antibiotic, because they don't use antibiotic when they farm the animals. So that might be something else that already in the environment and contribute to the transmit or moving of these AMR genes coming into the fish. For the islands, we don't have the environment sample to compare with, but just for you to see that the early state of fish also has different composition of the most abundant AMR genes compared to adults. So but I don't see the environment of transmit of gene because you don't have the sample, but just see the pattern that early life of fish have the different set of AMR gene compared to adult one. So let's get to the conclusion of this study. So what I can see here that you can see that the environment samples has a higher resistome diversity and also the richness. And then the fertilized eggs are only set of fish, of salmon, has different resistome gene pattern. And resistome or the AMR genes could be from the environment and the feed which is provided to the Atlantic salmon during their growth. So, and that's it for my talk, and I would like to thank so much all the partners in the Circles project that so contribute very much to this study. And that's it. Thank you.
Thank you, Shini.
Yes.
Very nice. One question that I would have right away, because that puzzles me, next to the audience. The question is, do you have the data, or do you do what is known from the literature about this? If the AMRs comes from the feed, how does this basically translate into being persistent in the gut over time? So if you were to switch the diet, would you think that the AMR patterns would then also switch? Do you think these are kind of early colonizers, long persisters in the gut? Or how do you think about this?
The feed.
Well, the AMR genes which are in the gut, do they come consistently from the feed? So do they need to have basically this constant feeding in order to keep these bacteria that host these genes? Or do you think that they, once they're there, engrafted, let's say?
I cannot say that until I know the degree. So what I show you, I only know where it's from, but I don't know the degree yet. Maybe it's not only just the feed and environment, maybe it's also something else that I cannot foresee from the results. So in order to answer that, I think we need to know the degree of these factors that contribute to the AMR that you see in the feed.
Oh.
Just one question. Did I get you right in because I think in one of your slides you said that the yolk sac larvae got their AMR genes from the feed, but the yolk sac larvae are not fed. So that is before they start feeding them. So I don't think that would be the explanation in that case.
It's not only the yolk sac one, but it's also the later one.
For the other one?
Yeah.
No problem.
Yeah.
For the yolk sac larvae, there was a
But what about the environment? It also has some of the genes that is similar to environment. I don't have the pictures now.
Yeah, thank you. Excellent presentation. Just following the same what Arnie mentioned. Also in the fertilized eggs in the wild, if it is coming from environment, I would expect that fertilized eggs in the environment, they should have more AMR. But I see, if I understood correctly, that in farmed, they are more compared to wild. So do you?
No, we
No,
don't
but
have
it's
that sample.
fertilized eggs?
Not
no?
in the wild, no.
Oh, okay. Yeah,
It's
sorry.
only the
Maybe
farm.
Okay. Okay. Yeah, sorry, my misunderstanding.
Costas.
Thank you, Shini. Is there any idea or knowledge where the AMR in the feed are coming from? I mean, is it microbial? Is it from the plant, animal materials? Is there any estimate on this?
I just discussed with Arne during the break before coming here. I also asked him the same question because he's the one that would know what the feed is, mainly plant. And if an organic plant that use fertilizer from, animal manure, that also could be, transmission factor already there.
I'm asking because as a nice control, it would be nice if it was possible to have an AMR-free or less feed.
There should be
that's
not only the
right.
animal feed, but also human feed.
Thank you.
Thank you very much. Thanks for the nice presentation. So I was just to say that we observe the same behavior in human beings. So in human beings, as you age, up to the extreme aging, so I'm speaking about ultracentenarian, you increase the diversity of antibiotic resistance genes in your gut microbiome. And this is consolidated. And the other point is that these approaches are still unspecific. So maybe some of the resistance you get provide also other functionalities, so are very generic from the functional point of view, beside providing the resistance to a given antibiotic. So maybe there is a secondary function.
Thanks for the presentation. Just a curiosity, the feed that you analyzed were medicated feed or not?
Agne, do you know the answer?
We know very little about the feed.
Okay.
Yeah.
No, because in
We may be able to find out more about the feed, but
okay.
I don't know. Yeah.
Okay, because in pigs is quite usually to include the antibiotic in the feed, and so this can select microbes directly in the feed.
No, but in that's not the case.
No.
Not the case, okay. Okay, sure.
Good. So then in the interest of time, I would suggest that we move into the panel discussion. And thank you again, Shini. Have a seat.
Okay.
Sushi, Itagele. All right, so it's my pleasure to be now sharing also this session on panel discussion on lessons learned and actions combating AMR. Yeah, we had now 2 talks on antimicrobial resistance, and it is a problem which is obviously of global spread, and I don't know if some of you have seen this publication that came out recently, and I haven't fully read it yet, but I'm sure there's some truth to it, which is about spreading of microorganisms across the troposphere, so at the planetary boundary layer, without any human intervention, right? There's nothing we can do about it. They just travel. And then they settle somewhere. So they settle with everything that they have. Now, we have all been through a pandemic, and AMR is also referred to as the silent pandemic. And within a pandemic, surveillance is key. and which is what I would like then basically to start with actually is we have now seen several ways of tackling antimicrobial resistance and surveilling it. And Shini, maybe you can share some thoughts on other ways you can think of AMR surveillance could be done beyond what we have seen, for example, in circles in other environments, other approaches, to basically make this possible at scale.
Okay, you start with me.
Yeah.
When you talk about the AMR surveillance, it's something that our group has done a lot. And if you, I don't know if you heard about the sewage surveillance of the AMR from the wastewater. So in the other EU project called WEO, we have sampled the sewage samples from around the world, 700 samples from 100 countries for 4 years, like sort of 16 to 19. Like 20 years, 4 years. So that's like a spanning of time and across different countries throughout the world of the sewage wastewater sample, and then we shotgun metabolomics of all of them. And what we want to see, not only to know the abundance between countries, and you can see that, of course, Europe and Northern American has a low abundance of AMR compared to African or Asia, for example. That's something that is not so surprised. But something that is more surprising me as well is that now we want to know what is the contributor to the abundance of AMR. Why some country has higher abundance of AMR than the other countries. So what is the reason, the cause of that? So we did some of the association study, machine learning kind of thing, comparing the abundance of AMR from the metagenomic of sewage with the socioeconomic data. Every country had these socioeconomic data, like population, how many people, how many doctors, how many people died, so like thousands of variables in socioeconomic data. And of course, the usage of antibiotics. That even though it's not a perfect data, but we still use it anyway. And we found that we've been talking a lot about the usage of AMR, the usage of antibiotics, and trying to reduce the use of antibiotics. But what we found, the degree of contributing to the antibiotic resistance from the usage of AMR, antibiotic is less than the socioeconomic data. Some of the socioeconomic data that contribute to having a higher abundance of AMR is sanitation. If the country invests more in sanitation, the density of population, especially in the slums. So there are more and more of the factors that related to transmission factor. I call it transmission factors. That AMR might be already there, but how do you stir it up and transmit it? If you have the factor that help them to transmit to people, animals, environments, that may be that the reason causing the problem of AMR than just the usage of the antibiotics. And another study is coming out from my colleague. He did way more analysis, statistic analysis, look at the different classes of AMR. Now it go down to classes of AMR, clinical, whatever. And he found out, yes, the transmission factor, socioeconomic factor, has way impact on antibiotics than the usage. But what is interesting, if you have like a line here, and if you have like a negative association or positive association, and you list all the socioeconomic data, including usage, the socioeconomic data can be negative and positive to the AMR, but the usage is in the middle, meaning that the usage of antibiotic doesn't contribute to either negative or positive of antibiotic resistance. It's meaning if you use more of antibiotics, but you have control very well of those transmission factors, you might not have AMR problem that much. On another way, if you reduce antibiotics, but you still have the AMR because you still have the other transmission factors, that's why the intervention way, the trying to stop the usage, it might not work in some of the countries because if they still have the other transmission factor, they will still never get rid of it. That salmon, they don't use antibiotic at all, but they still have a little bit from the environment. You see what my point? So my point of this surveillance of AMR using the sewage is it tells us that, well, if we focus only trying to reduce the antibiotic, not using it without taking care of the other factor that contribute to the animal, to the people, you still have the antibiotic problem. There is one farm in Denmark, my colleague told me, because they take very good care of the animal. The animal is not sick at all, and the farm and everything is good. That means the transition factor, they erase it. And once they had to use antibiotic from time to time because the animal is so sick, they didn't have antibiotic resistance coming out from the farm because the transmission factor is already erased. So my point is that maybe we should talking about the transmission factor, the well state of animal, human, and also environment, rather than talking about the usage of antibiotic alone. Yes?
Cedric, if I might add to what Shini said.
Oh?
Sorry, can you hear me? Can't hear me.
Oh?
Can you hear me now? No? Hello? Yeah, okay, great. Sorry, yeah, so to sort of to add what Shini said, I think AMR is a naturally occurring phenomenon, right? It's an ancient phenomenon. AMR exists in microbial communities, plant microbe interactions, irrespective of antibiotic usage. And I think it's important to recognize when you talk about surveillance networks, the key aspects of understanding AMR in natural ecosystems, whether that's rivers, whether that's wild animals, so on and so forth. And several countries have done this. I currently am in the UK. I know of examples in Europe, in the US, where people are looking at rivers, where people are looking at soils, where people, in essence, are looking at wild animals and so on to understand primarily what are the reservoirs and where are the potential pinch points where this might now explode into the human ecosystem. Because at the end of the day, we're all worried about ourselves and human health. So I think as a surveillance network or surveillance mechanisms, it's important to identify the human interaction-oriented natural ecosystems, whether that's lakes, where we like to go on our summer vacations, or rivers, or marine ecosystems, for that matter of fact.
And how about pristine environments?
Yeah. Incidentally, we did work on this, right? So I worked on glacier-fed streams back in the day, and we found AMR in glacier-fed streams, environments where you can't easily reach. These are above 5,000 meters in alpine ecosystems, and AMR exists. This is just a natural occurrence of competition among bacteria, viruses, fungi, of course. People always forget azole resistance in fungi being a primary factor, and we don't talk about it as much, because obviously it's easy to reconstruct bacterial mags and easy to reconstruct bacterial genomes. It's hard to reconstruct fungal genomes when we do metagenomes and so on and so forth. And this is often neglected. There are citizen science projects. For example, there's one in the UK that has looked at azole resistance from different environments. And of course, air has been a very transmission factor, has been a mediator of this. Glacier-fed streams, we find AMR sometimes that is associated with antibiotic usage, resistance against synthetic antibiotics. And you would think, well, is this a natural evolution of resistance, or is this something that actually just dispersed over time from anthropogenic or urban environments into these glacier-fed streams?
So this also links a bit to what you presented on basically monitoring different aspects of the food chain, right?
Yeah.
What do you think are specific technologies that would help to better surveil this? What do you think would be needed in order to improve this AMR stewardship dimension? Because surveillance is one of these.
I think that the technology, which is the shotgun metagenomics, which is reported by Professor Marco, is the one. Because in this study, we also see there are unexpected high level of some contaminant, like the chlamydia, for example, we found in the poultry carcasses. So in the SECA, there is less abundance of chlamydia, but we found in the carcass. So there are some contamination, which is other than the poultry or the farm, but it is related to the transport and also the processing. So I think having say type of technology which can be used as a routine diagnostic tool will be one. And the other is confirmation based on the target method, like the culture. Because when you are doing this shotgun metagenomics and others, it is more or less with untargeted. That is good to have a general idea, but if it is complemented with some of a routine-based method, I think it will be help to detect those pathogens and also the transmission of AMR.
Okay. I mean, in this context, so these are all mostly examples of human environments, except for maybe the pristine environments. And one of the aspects of AMR stewardship is also hygiene. Now, obviously, this is a double-edged sword because you can excessively be hygienic, and that comes with its own problems. Your immune system will not be sufficiently trained. But it's something which is relatively easy to control in a human setting. But how can we control this in a non-human setting? So, for example, what is also important is animals, domesticated animals. I mean, we have tracing of what you mentioned, antibiotic use. Does this also include, in your case, domesticated animals? Yes.
Yeah.
Okay. Cool. But in that context, how did you get to that data? So you had veterinarians that then shared this.
The usage of antibiotics. Yeah. Yes, every country has. I mean, not every country, but at least in Europe, you have this data. So how well they collect and how well they represent, that's another thing. Okay, but they have the data. In Denmark, we have even in the farm level.
But that's on the farm level. But I meant domesticated animals, dogs, cats, birds.
Oh, I mean, sorry, I misunderstand that. And I think I don't know about, but I think in Denmark they recorded pretty well, but I don't know about the other countries.
Okay.
I think in a majority of the developed countries, you do have very nice records. And it's funny, we don't talk enough to veterinarians. I used to work with veterinarians in the past. I think we as researchers sometimes are siloed. And we tend to stay in our community. But if you talk to veterinarians, they will tell you, oh, we've been recording antibiotic usage for the longest time. And I can tell you exactly which dog got what and how many doses and what kinds of antibiotics, because that's something we don't talk about. We just say, we found AMR this, we found AMR that. But the kind of resistance you develop depends on which class of antibiotic you've been prescribed and so on and so forth. So I think it's just a matter of talking more with other people outside of your field, which Circles obviously has done, but I think it just needs to be expanded a little more.
And so we discussed this before, or you mentioned this briefly before. So this is a global phenomenon, right? Yes, Kostas?
Do you want to ask questions for some time now?
You seem to be very excited, so by all means, please. No, go. No.
I'm excited from what Ginny said, and I'm wondering if this discrepancy that you described could be facilitated by putting the AMR into a biological context. These are genes, fine, but genes, belong to biological entities, to organisms, right? I mean, for me, it would feel different if I get the 10 AMR genes, in a strict endosymbiont, or if I get the same 10 AMR genes in a bacterium that lives on the fish skin, for example. It is very, very different in terms of a risk, if you like, okay? Or I would worry in a different way if I was getting these AMR genes on my skin, or on my gut, or in a bacterium that lives inside my gut, and it's very, very important for my health. The first one is this one, and the second one is, can we actually do it? Can we actually append these AMR genes to specific microorganisms from the samples that we analyze, or no? Can we have access to data of these genes? Are they actually expressed? They are swimming there, but are they ever expressed? Do we have cases where they are expressed? I mean, it's to anybody. You are the
What do you
expert.
mean they express?
Express the enzymes that the genes, the AMR genes.
That's
Yes.
why the antibiotic doesn't work, because they express.
Yes, but in this case, it's just presence, right? So it's just DNA.
Oh, you
correct?
talk about...
Yes, about the data.
I can maybe answer that question.
good.
So, in fact, we did this study in a wastewater system. We followed samples across a 51-week time point. We had metagenomics, metatranscriptomics, metaproteomics as well to go along with it. And I'm glad you asked that question, right? Because metagenomics, you see this long list of high abundance of AMR. But the moment you look at transcriptomics and proteomes, it's not there. There's very few genes that are expressed, first of all, to begin with. You can, yes, track which organism they came from. There are methodologies to do this in silico, and you can confirm this. And then when you go to the actual metaproteome, and yes, it might be a methodological issue, whatnot, the half-life of the product, degradation, etc. Metaproteome is virtually absent, right? So you can take it as you will. This is published in eLife. I was a postdoc at the time in Paul's group. And I'm just saying, this has been done for certain ecosystems. Obviously, it hasn't caught up. There's a cost associated with doing these kind of studies. It hasn't caught up with other fields, if you will, in human microbiome studies or even animal microbiome studies and so on and so forth. But the expression is sort of a sore point for me when anybody talks about, we found so many genes and we found this abundance of genes, please show me the expression data. Yes, I'm guilty of doing the same. All my papers are, we have all these genes and we have all these taxa that encode these genes. Rarely do I have transcriptome data to back it up. But that's the reality of it, right? You're a postdoc, you're a PhD student, or if you're a PI, you have to graduate, you have to get a job, so exactly.
But it might not express right away where you sample it, but it could transmit to somewhere else that it has a chance to express. But if you have a pool of genes so a lot here, it has more chance that it can go to where that is properly expressed.
Yeah, I agree. So I would not be worried so much about the expression, because the expression happens when the pressure, the antibiotic pressure exists. But in the case, if there is the resistance and there is the antibiotics, and so the resistance will be expressed, and so the phenotype will be provided to the microorganism. So the warning is present. And then, of course, if there is the pressure, the antibiotics, the genes will be expressed. So this is so important. The other point, so I want to say, we can match, we can match the antibiotic resistant genes with the taxonomy and so on. We did by reconstructing the metagenomic species. So in the other two works, we were exploring the dispersion of bacteria, so metagenomic species, and the carrier gene. The problem is that in that case, it's not possible because it's not possible to reconstruct metagenomic species because of difficulties in shotgun metagenomics with the fish. The other point is, in your when you said that there is no direct connection between the antibiotic usage and the antibiotic resistant genes that you observe in some countries, maybe there is a problem of the incredible use on unprescribed antibiotics in the third world. And in particular in Africa, they are using a lot of antibiotic without any prescription and without knowing anything about what they are using and why. So, like a magic, I would say. So this is a huge problem. So the usage of unprescribed antibiotics that can be mapped.
That's what I meant about the usage data is not so complete. So there are a lot of unhidden numbers that is not included there. But I can say an example in one of the countries, Thailand. Thailand has a lot of AMR problems and also a country that you can buy antibiotic in pharmacy. So the data of usage doesn't represent what it actually used. They realized this problem, and there had been done a lot of effort reducing the usage of AMR in animals, pigs, whatever, cow, whatever that they raise. Of course, in the hospital, they also reduce. So both human and animal, they reduce it a lot for many, many years. But the incidence of antibiotic resistance never decrease. It's going up constantly. So that's what I meant about the transmission factor. If you try to reduce the usage without taking care the welfare of human and animal environment, you never get rid of this antibiotic resistant problem. Yeah.
Which is a very important point because, because if you adopt this perspective of One Health, where basically the health of humans is linked to the health of the animal and linked to the health of the environment, and when you couple this with pollution, which again is a transmission factor, like dust and whatever else you have that can travel across countries, doesn't stop at boundaries or doesn't give a damn about what you think it should do. So what is your experience maybe on the on environmental pollutants in AMR spread?
Yeah, here maybe the point you raised before regard to the cat, dog, these domesticated animals. So here you have to bring the points of One Health. Because in our university, we have in the veterinary clinic, there are a clinic for small animals, which is come, and we are also detecting those antibiotic resistance bacteria, those which expressing a gene for beta-lactamase and also carbaminase resistances. So now we saw from the study, which shows there is the transmission from poultry house to human. So I think there is also the reverse way can happen because of the cat, dog, and also the human interaction. So this is one of the One Health approaches which can increases also the transmission dynamics.
Any comments on the pollutants dimension of it?
I mean, microplastics, right? Microplastic is one of those things that, yes, it's catching up now. I think we saw some evidence today of that in Marco's talk as well. This is, I think, a key area, primary pollutant. You obviously have industrial runoff going into rivers, oceans and so on. So this is no surprise. They do tend to select and add, whatever pressures are required for selecting for certain resistance genes. It's co- and cross-resistance. These are mechanisms that are inherent to microbiology that we tend to forget often, that when you have a certain metal being dumped, whether it's in soil, groundwater, or rivers, it potentially is going to co-select for other resistance genes that are usually encoded on the same operon or in a bacterium. Any sort of pollutant could, in essence, drive it, but I think the big thing to focus on at the moment is microplastics.
Thank you. We actually have approached a lot of questions that I had on my list, so that is really great. And Marco can read mine, so he had brought up one. One that I would basically like to close this panel with is, I think we are now getting to a point where we are, as a scientific community, maybe larger than it was before, because obviously it is known for years that antimicrobial resistance can be a problem. But more generally, like towards the public, but also towards the next generation of scientists, what is maybe one key point, key advice that you would like to give them in that direction? Maybe starting with you, Itagele.
Maybe in the control option, you mean?
Whatever, in the context of antimicrobial resistance and combating it.
I think what I will suggest is really the using of microbial modulation aspect is one of the important, because as I indicated from the review, I saw there are a lot of nutritional intervention which can modulate the gut in order to fight the foodborne pathogen, and also there can be used as alternative to antibiotics. So these are data available, because most of these drug tested already from the literature which I saw done on broiler it indicates that there are some of these WHO indicated clinically important antibiotics can be replaced by those nutritional intervention which can modulate the gut. So as a recent science, I can say the modulation strategy can help to control.
Shini?
Back to what I already said, that usage of antibiotic is not only thing. So I think the surveillance system in different countries should also put a bit of focus on from the antibiotic usage, try to reduce it, but to try to reduce the other transmission factor. Before that, you need to identify what is or are the transmission factors that contribute or transmit the AMR in your country or in your farm or whatever. So maybe you need to identify the transmission factors that contribute to the AMR. And that could mean that also the research funding should also be more on the environment, because in AMR, all the some of the E. coli that that reason normally come from the clinical samples or from the animal samples, but not much from the environment. So that because there's no funding on the environment microbiology to starting AMR. So that would be another thing that I think the funding also should, focus more on the transmission of AMR in the environment.
Sushil?
I think two key things for me. I mean, I would tell the next generation, focus on the ecological drivers of AMR, right? And this is something we tend to forget. And education is key. The kids these days are much smarter than I was back in the day, and I think start at the grassroots level, start at the schools, start talking about AMR at a really grassroots level, and it'll stick. I think we are retroactive at the moment, we do things in hindsight, and I think we should be more prospective going forward. And I think education is the best way to go about doing it.
Thank you. And just one little note, actually, I've been talking about retrospective and prospective. I actually forgot to introduce Sushil at the beginning. All the others had already the pleasure of being introduced. So I would like to do this now before closing, actually. So Sushil is a molecular ecologist at the UK Centre for Ecology and Hydrology in Oxfordshire, England. He has a diverse career, including leading organizations such as Google, as well as Imagilin Technology, which is a probiotics company, and academic roles at University of Missouri and University of Luxembourg. He has a PhD in molecular immunology and microbiology, with a research focus on studying microbial ecosystems across various environments, including humans, animals, and pristine environments. And he's interested in better understanding the ecological drivers of antimicrobial resistance. So now you maybe better understand why this panel has been conceived the way it is. With that, thank you very much.
Okay, now we'll have, I'm Giacomo Sini from UFIC. Now we'll have the last session of this final conference. We're going to present briefly the results of this scientific research, which is the circle box. We'll have 3 speakers. The first one may surprise you, is Marco Candela. And then we will have Andrea Castagnetti and Carmen Bianca Socascio, that is going to present the business model we make for this circle box. But first we have the video that maybe we can project now. So, yes.
Introducing the Circles Box. The Circles Box is a tailored solution to help farmers, livestock owners and aquaculture farms to assess and modulate the microbiome of their food chain in order to improve the safety, productivity and environmental sustainability of the food production process. The Circles Box will be available for 4 food chains: spinach, tomatoes, swine and sea bream. Steps. 1. The the end user can sign up to a virtual platform and enter the details of its operation. 2. Based on the inserted information, the end user receives a tailored circles box, which contains a self-sampling kit. 3. The end user samples the microbiome of the crops or animals using the test kit provided by the circles box. 4. The end user sends the samples to a lab which will analyse the microbiome of the crops or animals. 5. Based on the analysis, the end user will receive a tailored selection of natural microbiome modulators, as well as custom-made protocols for their use to improve the food chain's microbiome. The Circles box offers several benefits for the end users. For plant food chains, this includes higher productivity, improved plant health, less need for chemical fertilizers and pesticides, and less environmental pollution. Within animal food chains, it enables higher productivity, increased nutritional quality, as well as a lower need for antibiotics. Follow at Microbes for Life or visit www.circlesproject.eu to stay posted about future updates about the development of the Circles box.
Okay, and now I will leave the floor to Marco for the introduction of the Circle box.
Thank you very much. First of all, thanks God there is no Aton here listening the Circle box. Maybe who works in the microbiome field understood what I'm meaning. Anyway, so, okay, there is the presentation. Okay, so this is a summary of the Circle box. So what I want to say now, even listening all the presentation we have today, that for some food system, we are still in the observation phase. So we are still observing the diversity and trying to understand if this diversity maybe is useful for doing something. So for instance, in aquacultures, in poultries, and also in pigs, since we have a lot of data and we are still understanding. In other situations, maybe we can do something and we can really apply the model of the Circle box. And this situation, according to what we have seen today, are both in the field of agriculture. So as we have seen from Davide Bulgarelli, we have some bioinocula that really works in shaping the spinach microbiome structure, reducing the pathogens, and also improving the productivity, since you have more yield when we collect spinach. So we have, in this case, we have a clear endpoint, which is connected to the microbiome modulator. That is something that make the microbiome modulation somehow concrete. Can work or not, but we can connect microbiomes, the Circle box, the approaches in the Circle box with something concrete, and we can try to convince people and farmers to use it. The other case in which I see the possibility to really create a Circle box is the case of Montepulciano. When we have been able to isolate strain which are specific for each area of production, for each additional geographic unit, as Georgia was saying, and that are involved in and beneficial for the plant growth, since provided a lot of growth promoting functionalities. So these strains are already there, but of course they are important, specific, and can be involved in food production and can be treated by environmental changes. So we need to know that they are. We need to know that they are part of the production process, so we can monitor their presence year by year in each place by using the Circle box. And if something happened to this strain because of the because of that, or the many global change, we can reintroduce the lost strain since can be grown, can be produced in fermentation process, and we can create some customized bioinocula that will be included in the circle box. So I can imagine for each production area its own circle box in which we monitor the presence of the specific strains as promoting and connected with the quality of the products. And if something is happening to that strain, we can reintroduce this strain as bioinocula with a circle box. So this is the two concrete applications that I see. And that say, it's so important in the context of the circle box, the auto-sampling of the food system microbiomes. For instance, in multiple channel, it's so important that the workers, the people, would sample correctly the root microbiome, the soil microbiome, and send back the sample to the circle for the analysis. So I'll leave the floor to Andrea from Well Micro that has worked a lot in producing this auto-sampling kit that let the people to sample along the microbiome, and then we will take care of the analysis and on the data interpretation.
Thank you, Marco. Thank you to everyone. It's a pleasure for me to be here. Just a very quick introduction. I represent Well Micro, which is a microbiome-based company which developed a sampling kit aimed to sequence DNA. So we collect many experience. We sequence and sampled more than 40,000 samples, okay? So we know how to sample, I think. We have just experience on this field, and so we were involved by Circos for this particular task. And yes, no, please, there should be 3-4 slides. Now I'm gonna show you, if we get the slides, the kits that we developed for both dissemination activities and also for research activity, and so data collection exploited for the data analysis. The first activity that I'm glad to present you, it was a city tour sampling, let's say, for citizen science activity initiative, where we provide to citizens for free, for sure, sampling kit for the environment, specifically for the floor of their houses, and we spread these kits through Italy, 2 spots, Bologna, which is the where Well Micro comes from as well, Mazara del Vallo, Brussels, and Helsinki. So as you can see, we got also a nice representation in terms of latitude of the environment of the samples. So we received back the we received back the samples using after try different components for the sampling. So we provided the best cutting edge components for this task. And the data analyses are like in progress because we received from the last citizen the sample a few weeks ago, but I can tell you from the excluding Brussels that we got a quite big difference between Helsinki and Italy. It seems like Helsinki has more environmental bugs in their houses. Instead, in Italy, we have more humans bugs in our houses. So also it's consistent if you think about with what we have seen during the presentation about the Western Europe dispersion of antimicrobial genes and whatever, maybe in the north of Europe are more sensible to this topic. I don't know. Then this was the kit that we built up for this activity. So This is the component with the buffer for the DNA preservation, which is the most important issue when you are facing the sample for microbiome, because your first task should be to inhibit the DNases present in the environment, and also that can be released by bugs killed or death or whatever. The second one was to develop a kit for seawater sampling, an auto-sampling kit, so it's supposed to be very plug and play, easy to use for the citizens, and this was the activity that already we have seen this morning. And here, once again, we change for sure the sampling. In this step, it was an issue for the conservation because we could not use the swab with the liquid preservation, because when you sample the seawater or water in general, it's much more poorer. It's much poorer than dust or whatever. So you should sample liters of water to have a good amount. So we have to no, sorry, the point. Okay, we told the people to filter like 20 times more than 50 mL of water, and at the end we build up also, we set up the kit for the workers dispersion tracking or sampling, because we provide a kit for sampling for feces, for skin, for domestic floor, and representing the workers' environment. We use the work shirt as a proxy of the environment of the workers. So the workers was asked to sample its work shirt after leaving the workplace, okay, after a day work. And that's it. This is a quite complicated kit because we had to sample like 5 or 6 swabs, and also providing the shirt, the work shirt, and whatever. But at the end, it was through this kit that we collect the data for the antimicrobial dispersion, considering the worker in the workplace as well. And that's it. Thank you.
Thank you, Andrea. We can move to the last presentation from Carmen Bianca Socaciu from Gate to Growth. She is a manager of EU agri-food and bioeconomy projects, and they specialize in strategy and business development for early-stage companies across various sectors. She's coached more than 150 companies, and she helped them in replicating, upscaling, and to the capacity building to identify and developing promising project outcomes. So please, Carmen.
Hello, everybody. Thank you so much. Last presentation of the day, so let's get to it and make it short. So, the whole day we've been talking about microbiome. So, we've been talking about the challenges of the farmer, low profit margins, that they need to comply with agricultural regulations, that they need to take care of the soil, the farm ecosystem, and biodiversity. So, the solution from our project is going to try to shift toward more sustainable farming. So, you saw already the box was presented in a video, and what we offer, and just like we said that before, all-in-one solution for modulation of the plant microbiome. Andrea was talking about the microbiome kit. So we will have microbiome analysis here, the smart microbiome modulators, and an intervention protocol. We're going to focus on the circle box for plants, tomato and spinach, and we're also looking at the market. How is the market? And is the market ready for our new solution? What does the market tell us? And we can see the crop nutrition and crop protection. It looks pretty good, promising. And if we look at 2024, the projection for 2029 are pretty positive. So the market is ready for us. But let's look at a bit in a business model. And so how does the business model help us? Well, this outlines how a company creates, capture values through the operation, customers, and revenue sources. So basically, how do we sell and deliver our solution to our customers and hopefully make money? So we need to think here, is the business model or the product? Which one is going to give us a competitive advantage? So you have the shop and the customer, and how is the customer going to buy? Is it via lease, purchase, pay-per-use, or we've been discussing today also potentially the farmer can pay based on its farm size, or the farmer can pay some in installment, or they can pay based on their harvest. Because we've been talking that the harvest is not something that they can rely that their harvest is going to increase a certain percentage. Now, the circle box solution explained, you saw the video, and we also tried to do the best possible to represent it. I think we need to synchronize a bit the video and our presentation, but otherwise we're good. So we have the circles kit. The farmer is going to do a soil analysis. We have some consulting services, and then the tailor box can be purchased, and the circles intervention is going to the farmer who hopefully is going to apply it on the field. Now, if we look at every orange circle, is at every step we are having a business opportunity, which means a business model in itself. So we have the circle kit where the soil analysis can be made, and we call it the circles kit business model. And we have consulting services that can be an add-on. And then we have the intervention box. Can be a business model in its own. Now, for us, and I'm happy to have the final presentation, we are combining here 6 years of research and innovation. We are combining huge collaboration to achieve the best results. We were not involved in all the hard work that the partners were doing in the lab and sampling and so on. For the farmer, it's going to be upfront cost, but potentially long-term gain. We've heard that in order to build the soil health, it requires some years. So that's really promising. Again, the gain is variable, more testing is needed, however, results are promising, and I think Davide was talking about some good results registered. So we also know that 10% approximately in yield we saw in the spinach. Well, the key learnings for here that one-size business model does not fit all. You need to adapt the business to the business conditions, talking about environment, we've been talking about that a lot. And you need partnership with local networks to have success. And as we saw before, in a supply chain, multiple businesses can develop based on product, services, and technology. Now, help us choose the right name for our box. We have, from the project, the obvious circle box name, but we thought maybe that's not the right one, and we can have the circles microbiome boost. We can have circles of growth, or circles microbial mix, or the fifth, any other. So I challenge you to a vote. Please raise your hand. Who is for the number 1 circle box? And then we have 7, 8, 9. Yeah. So 1. Okay. Number 2, the circle's microbiome boost. Okay, 10. Okay, number 3, circles of growth. 1. Still good. Okay. Number 4. 1. Okay. 5. Any ideas? Please write us. And yeah, you have a suggestion? Okay. Yeah. So circles for growth. Anybody in favor for that? Yeah, 1, 2. Okay. Yeah, thank you so much.
Can I vote for number 5 as well?
Yes, 3.
I get vote for number 3, right?
3, and yeah. As of now, we have number 2 with most votes, 10. So, yeah.
Carmen, can I argue it for my choice?
I think like we don't have more time.
No, because it was the initial name.
Yeah.
Because it sounds nice, because it's kind of contradictory. It's a circle in a box, in a square. So it makes sense. Circles box, as we said in the beginning. Yes, even better. Circles in the box. Circles in the box.
Thank you.
Okay, so 6th, yeah.
I would suggest for the second microboost instead of microbiome boost, which sounds microboost. So
Okay, so circles microboost.
yes.
Okay. Do others agree with the name? Okay. Thank you so much for being such an active participation and bearing with me the final presentation. Oh. Thank you. And thank you, Michelina, for all your hard work. And thank you for the partners being so collaborative and joining our meetings. And yeah, thank you so much. It was a pleasure.
Thanks so much, Carmen, for the perfect closing. And so we are done for today. So there is now the aperitif on the roof and the dinner. So see you later.